Testing, testing, testing.
Okay. Hello. Good morning. Okay, let's start. Okay. Thanks for coming. I know it's a big raining day. It's a surprise for Southern California. So really appreciate you be here and be here on time. Thanks for coming to the first Southern California Healthcare Innovation Symposium, joined by the third International Conference on AI in Healthcare 2022. I'm the chairperson of Southern California Mondre Science and Technology Association, Ningning Yu. Today's symposium will focus on three main areas: precision health, AI in healthcare, and telemedicine. Why? During the post-COVID era, we have experienced a paradigm shift in the model of healthcare. To prepare healthcare providers and start a company in the U.S. and Taiwan with emerging opportunities. In the U.S. and Taiwan, for the why is possible, we have invite research pioneers and business stakeholders from healthcare technology companies, healthcare providers, and major medical research institutions in both in the U.S. and Taiwan to speak and participate in the symposium. The primary goal of the first Southern California Taiwanese Symposium on Healthcare Technology, including three things. Number one, presenting the cutting-edge development in the three areas, in both U.S. and in Taiwan. Number two, identifying the synergy and opportunity between these two countries. And three, facilitating dialogues and collaborations between stakeholders and entities in the U.S. and Taiwan. Know that I hope you will stay here till the end. At the end of today, we have an exciting startup pitch contest. I really look forward to it. And the participate teams, including some of the top startups from Taiwan and some local here in U.S. And I look forward to it. And now, let me introduce Dr. Cho Wen-da, co-CEO of AHMC and our co-host for his remarks.
Thank you, Ningning. Distinguished guests, ladies and gentlemen, I would like to, on behalf of AHMC Health System, I would like to welcome all of you to come to the first SoCal Health Innovation Symposium, which is joined by third International Conference on AI in Healthcare. The president of that conference, Professor Jack Lee, is regrettably unable to join us today. As our Congress President, Ningning Yu, has already introduced the fundamentals of the whole program, I would like to share with you some future trends of in digital health. Next slide, please. May I introduce you, Professor Matthew Lin. He is Vice President of the AHMC Health and retired Deputy Assistant Secretary of the U.S. Department of Health and Human Service. Most importantly, he is the president of American Hospital Investment. So let's welcome Dr. Lin, and please stand up. Okay, thank you. Okay, I would like to introduce some of the future trend in digital health. Next slide, please. First, we found that AI and digital health publications has increased sharply after 2019. You can see that it's skyrocketed. Second, next slide, please. This is the topic. Next. Second, we need to be aware of the current development of the FDA strategy. They have defined the scope of digital health in 2020, which include mobile health, IT, wearable device, personalized medicine. Next slide, please. The FDA also suggests 5 beneficial criteria, which including efficiency, accessibility, cost, quality, and personalized medicine. Third, next slide, please. According to the American Hospital Association and HIMSS, 80% of the healthcare provider and hospitals has planned to increase the investment in technology and digital solutions over the next 5 years. It is anticipated that the growth in this area, which including telemedicine, personalized medicine, genomics, AI, cloud computing, and IoT. Next slide, please. Fourth, for innovator, we recommend the use of system thinking to improve your product using FDA's 5 established criteria. Next, please. Today, the 3 topics in this conference, including precision medicine, AI medicine, and telemedicine. These are closely related to the future strategy from the FDA, AHA, and HIMSS. So finally, ladies and gentlemen, this is a great opportunity for all of us to gather here to discuss the future of digital health. We want to welcome you again, and we hope that the conference will have a great success. Thank you for your coming. Thank you.
Thank you, Dr. Chu. So next, we will invite our sponsor, TICO office, to speak.
The chairperson of ACMJ, the chairperson of ACMJ, Yu, and the co-CEO of AHMC, Dr. Cho Wen-da, distinguished guests, ladies and gentlemen, hello, good morning. This is Jian Chen. I'm the director of the Science and Technology Division of the TICO LA, Taipei Economic and Culture Office in Los Angeles. So today, I'm honored to be invited to participate in this symposium. I want to take this opportunity to thank the team of ACMJ, especially for the chairperson, Yu, as well as the co-CEO of AHMC, Dr. Cho Wen-da, for organizing this important symposium. So if possible, I would like to start by showing you a video presented by General Louis Huang from TICO LA. So please.
Good morning. As Director General of the Taipei, Chairperson Ning Yu, distinguished speakers, ladies and gentlemen, good morning. As Director General of the Taipei Economic and Cultural Office in Los Angeles, I'm honored to welcome you to the first SoCal Healthcare Innovation Symposium. First of all, I would like to express my heartfelt gratitude to the Mount Jade Science and Technology Association in Southern California for your tireless efforts to organize this tech forum. I want to take this opportunity to thank the organizers for their hard work putting together this event to kick off the first Southern California Taiwanese Symposium on Healthcare Technologies that offers a novel forum and networking opportunity for physicians, entrepreneurs, scholars, and investors in both the U.S. and Taiwan. On July 28th, President Tsai Ing-wen addressed the opening of the 2022 BioAsia Taiwan International Conference and Exhibition. As biotechnology is key to public health and our national development, President Tsai stated that the government has introduced medical tech and precision health as one of 6 core strategic industries for long-term growth, aiming to make Taiwan a leader in precision medicine and digital public health solutions. With today's symposium topics being precision and AI medicine, it responds and supports President Tsai Ing-wen's remarks. According to the California Biotechnology Foundation report, Los Angeles County is home to 3,876 life science establishments and employees 96,644 people, generates $60.8 billion in county economic output, and increases labor income by $19 billion. Its local establishments were awarded over $1.9 billion in NIH research awards in 2020. I trust that the event will take an in-depth look at issues regarding the future trends in biomedical and life sciences. This symposium provides a platform for speakers to share their expertise and experiences with participants. I hope you will be inspired by the presentations and discussions today. Have a great forum. Thank you.
Thank you. So some of you may know that the reason I'm here is because
So may I invite Dr. Chen and Dr. Chu and Nini to the podium? We are going to have a photo.
Yes.
After photo, we will soon proceed to session 1.
Hello? I'm just checking the microphone, sorry. It's working, nothing. That's good.
Hi, good morning, everyone. This is HR Zhen from U.S.A. Medical School. So I'm the chair of the first Precision Medicine Session 1. And so we are a bit of ahead of a schedule, but I think why not I know we have our speaker have a lot to share with us. So we'll probably we'll use this time to buy a little bit flexibility for our speakers. So next slide, please. Over here, I would like to give show our special appreciation to very important staffs who make this conference possible. First one is Claire Huang, who is from AHMC. And Claire is very much instrumental with all the detail of this conference. She burned a lot of late night oils and even have a rundown down to all the details, which makes things possible over here. And Claire, if you are there, could you wave your hand to us? Claire is too shy, hide somewhere. But when you see Claire, please express your personal appreciation to her. The second one I want to recognize is Dr. Deng Kai Liu. Hi, Dr. Liu, are you around? Yeah, so Dr. Liu is from JPL, and Dr. Liu is instrumental behind all our video audio setup in the conference room. And thank you, Dr. Liu, for making all this process possible. We often joke, Dr. Liu, he used his precision sending the rocket to the Mars to control adopt the same precision for our conference. So that's part of our precision medicine over here. Okay, so next slide, please. So we're going to officially kick off Precision Medicine Session 1. Next slide, please. Okay. So when Dr. Chu and the Ning gave me the mission to structure this precision medicine session, we got to invite a speaker who will attract all the audience to come from Southern California, including people from out of the state. So Dr. Jin Lai Zhenguang probably need no introduction for his achievement. And I think I just want to highlight one point. He's a founder and the president of a BioLegend. And last year, it was acquired by PerkinElmer for $5.25 billion US. And that's really spectacular achievement in our community. And we always call Lai Zhenguang is a Taiwan Zhiguang with his Lai at the end of his name. Okay. So Dr. Lai, he's a clone antibody. Okay. And I think today's talk, he's going to lecture us, share us his experience, how he kept for monoclonal antibody and create several spectacular companies, including BioLegend, bring this cutting edge technology to the market. And I'm going to switch to Zoom because Gene, he couldn't join us because he traveled too much. Unfortunately, he was contracted with COVID. So then he's going to speak to us from San Diego. Please, streaming. Good morning, Jean. Are you there?
Yes.
Oh,
I try to share the screen. Can you see my screen?
Very well.
Very well. Okay. So I'll get ready. Okay. Let me put the view. Okay. Can I start it now?
Yes, please.
Okay. Thank you, Shenrong. You contact me to join this meeting. I'm so sorry about this last minute because I contracted COVID-19. You can hear my voice a little bit hurt. Hope everyone still able to hear clear from me. Today I want to thank you all participate in this, and I want to share with you my story. My story from a veterinarian to an entrepreneur, and the immigration story and the journey from across the ocean to life in America. I look back at the early humble beginning to the leader in life science tools of reagents. So I give a title called How I Got Here when Shannon asked me provide a title and abstract. So I thought, what can I tell to the audience here? Maybe share you all my story, how I got here. So briefly, I have 6 different sections. First is my background, and then how I get into monoclonal antibody and entrepreneurship, first company, Pharmingen, in 1987 to 1997. And then after that, I continue with the BD after acquired Pharmingen for 5 years. And then I start over my second company, BioLegend, in 2002 to present. And also I want to share with you BioLegend IPO and Alhambra acquisition in 2021, the story about that. And then after, because Shannon asked me, told me that if I can share more story after acquired by Alhambra. So I want to share with you my philanthropy and the giving back to community as well. Let's start with my background. I grew up in the countryside, Miaoli Shifu. Beginning, I was born, and then due to superstitious, I could not be raised up unless give to someone. So my parents give it to my mother's brother. So they take care of me from first year until 5 years old. So all grew up in the countryside. And after 5 years old, and then brought me back to family. So you can see that my mom and my dad took me to Taipei Zoo at that time, somewhere around 5, 6 years old. Try to comfort me as well, because I have to really recognize who are my parents. It's a kind of interesting story. And I continue to grow up. And my father is a elementary school teacher. As you can see that during my elementary school, my study performance pretty well, but it's in the country's elementary school. My father graduated from Taichung's normal school. But that time, early on, there's no normal university, just a normal school. So then, of course, my middle school and high school did not, because, and then my parents sent me to a city, Miaoli city, and try to catch up the school. But every day I would take about 1 hour walking to the train station and catch train and to school and come back again, catch train and then walk about 1 hour to home. So day after day, so I'm right behind the other students because they all after school go to the after school academy, so 补习班. So gradually, so that's why my middle school, high school did not do well. So I ended up in National Pingtung Institute of Agriculture. Today it's called National Pingtung University of Science and Technology, and major in veterinary medicine. In veterinary medicine, I got a big hit from my pathology professor. The first day in the class, he came to class and told us, I know this place may not be your first desire to come, but if you start to work hard and study hard today, you can be like me, because my pathology professor is alumni of National Pingtung Institute of Agriculture as well. The time he was an adjunct professor in National Taiwan University Veterinary School teaching pathology as well, because he's a very famous pathology professor. So of course, in my class, I have another classmate. Now he is become, he said that I have to go back to what he said, you work hard and study hard. One day you can be like him. You are not the student of National Taiwan University, but one day you can be the teacher of National University students. So that's inspired us a lot. So one of my classmates today, he's still in teaching in National Taiwan University, teaching pharmacology in veterinary school. So that I want to make a point is whenever, what time, you always can rebound and can even do better. The next one is after graduate from school, you have to serve 2 years military service. I was in Army Infantry School, platoon, the leadership training. So that become the platoon leader of MOTA after discharge from the infantry school. But I want to mention this because this really helped me my leadership training during my military service, as well as a platoon leader of MOTA. There's 4 units there, so I able to use a strategy, how to utilize my resources to concentrate on one point, one spot, or spread out, or further. So that also helped me the business as well. So how I get to know monoclonal body during my senior year in the college, I took advanced immunology. That's I learned George Kohler and Cesar Milstein's monoclonal antibody technology, and making hybridoma, and talk about the magic bullet. That is start with Paul Ehrlich. He is a Nobel Prize in Physiology of Medicine in 1908, German. And he uses imagination of there's something, if there's something can carry drug specifically to destroy any cancer or the invaded microorganism. So that's his imagination. And on the right side of the picture, that's Paul Ehrlich. And also I put the photo there. I just recently came back from German Immunology Society meeting. They present that honorary medal to me. And that big medal is also Paul Ehrlich. I'm very honored to receive that for long-standing the support of the society. And George Kohler and Cesar Milstein, not only their technology, hybridoma technology, but most important is he benefit the rest of the world today in the antibody therapeutics, because they both did not patent the hybridoma technology. So that's why hybridoma technology immediately spread out and being utilized in for research, for diagnostic, and therapeutics. Another person I like, my inspirational person is Len Herzenberg. I met the Len Herzenberg and Lee Herzenberg in 1985 while I was in Taiwan, worked for one of three biotech company called Avenue Biotech, because Ernie Huang is Len's postdoc, and he worked for Avenue Biotech as the director of research department, and both visit Taiwan, so I have a chance to talk to them. And in fact, they built their first flow cytometer, and that is the first single cell analysis tool. And this so far, I'm still very good friend with Lee, although Len passed away a couple of years ago. Let's start with my journey. First company, PharmIngen. PharmIngen founded in 1987 by Dr. Ernie Huang and myself. And the business idea is provide the antibody reagents. Because while Ernie was in postdoc in Len Herzenberg's lab, and he was always take care of around the world asking for antibody generated from Herzenberg's lab. And because as a postdoc, postdoc always want to publish more papers, but he has a lot of side work to do to fulfill all those inquiries. So he get the idea, if I can license within this cell line, start a company, we can refer them, just go to the company and then purchase from there, and the price not too expensive and will be on time as well, instead of waiting for postdoc when are available. So that's a business idea. As you can see, anything can be business possible. How I met Ernie and how I joined Ernie. So I met Ernie while I was in Taiwan, one of the three in 1980s biotech company called Avenue Biotech. And Ernie was recruited from overseas, from US back to Taiwan as the research director. So I work in the product development. And how I learned from Ernie making hybridoma and how do we screen the pairing, the antibody, is during my 3 years in Avenue Biotech. And he stayed there about 3 years later, he moved to Singapore. And because I continue to assume more responsibility and found the longer the product has some stability issue, I never able to resolve it from research lab to manufacturing. So I thought I need to catch up more advanced knowledge. So that's I decided to come to States for my further study. And of course, in Taiwan, your degree is become very important. So I want to finish my PhD and then return Taiwan. My boss very nice to me and keep my job without pay for study in US. While I was in the school, University of Louisiana at Lafayette, I came into States 1987, January 6th. And then that about the December time, almost to Christmas time, Ernie, he reconnected with me and then called me a couple of times and want to invite me to San Diego during the Christmas holidays. So that Christmas holidays, I flew to San Diego. While a connection in Dallas, Texas, due to the snowstorm, the flight was canceled and I delayed there, stayed there for 1 day. And next day, and then flew to San Diego, and then San Diego, a sunny, beautiful city. So I was so impressed, so much difference from East Coast and West Coast. And Ernie proposed to me start a company. I want to invest the company. So during that couple of days, talking with my parents for investment as well. So we both family come out total capital about 262,500. And Ernie asking for friends as well. So that's how we start PharmIngen. Of course, after we return to university, I talked to my advisor. my advisor said, well, I would be happy for you going to start a company, and asked me to just work on thesis. Impossible to graduate in first semester. So he need to organize committee and then prepare for summer break, summer section for the oral test and the thesis oral test. So as you know, that time there's no PowerPoint, no Word document, everything use typewriter. So whole semester pretty much doing all typewriting and doing the experiment, take pictures, dark room to develop the film, and so on. So luckily, that summer I graduated. So quickly give me the master degree and spent about a year and a half in Louisiana, Lafayette. So came to San Diego. When we started farming in total capital, 26 to 5,000, 262,500. That amount is terribly low. How we survived. And actually, we keep writing the NIH SBIR grants. So in the farming in town, we received the 12 NIH SBIR grant, including 2 phase 2 grants. And also started with 3 to 4 people to about 250 employees in 1997. And the company was acquired by BD Bioscience for $75 million. On the right side is my partner, Ernie Huang. And then after acquired by BD, continued with BD for another 5 years. Really, those 5 years are my real business training. I did not go to MBA, but all my training from doing work with BD. As you know, that 1999 is the dot-com bubble peak of economy. And then after 2001, bubble burst and then run into business recession. And then 2002, and just like almost all the public company during business recession, always looking for the cost reduction. What is a bigger cost is the labor. So they are going to do the layoff. As a farm engine in a small unit business in BD, a Fortune 500 company, they don't care about how business doing. It's kind of cross-bow, have to cut 5% labor. So, and then you have to name how many people are going to cut from farm engine side. I'm the senior people in farm engine. They want me to make an announcement. After I looked or submitted the name from my old colleagues, I decided not to do that. So I decided to leave the company. And then 2002, I started BioLegend in June 17. And it's a wonderful journey. Just highlight here, I received EY 2016 Entrepreneur of the Year Award in San Diego Life Science. And also same year, national finalist in the same category. And also recently came back from German Society, German Immunology Society annual meeting and received honorary medal. So when I started BioLegend, I put the mission is very important. And what, of course, why decided BioLegend name? Because if I don't have I did not have the first experience with the farm engine entrepreneurship, I probably hard to find really the legend. So when I started second company, I thought I'd like to become the legend. The company become the legend in the bioscience. And also I need to have very clear the mission. So mission cover from to enable legendary discovery from research to cure, which is to accelerate scientific discovery from research to cure. It's not only for research use, but diagnostic and therapies as well. I'll give you a quick timeline about the by legend. I start 2002, and then 2004 we already reached $1 million sales. And then 2005, second half of 2005, about a break even of finance. And then 2006 become profitable. And 2008, we extended BioLegend to global reach. So we set up BioLegend Japan subsidiary and BioLegend Europe in Amsterdam to serve all the European countries and continue to grow the business. 2010, we set up Taiwan, BioLegend Taiwan. And 2012, we set up a UK subsidiary. Some of the slides here I want to show you. I don't know whether you can see my point or not. Start 2008, we sponsor AI as a platinum sponsorship. Why I started sponsorship? Because it is very important for scientists to gather together, especially during gala or party time, easy to connect with the key opinion leaders and looking for any ideas and also what they are working on, what do they need. That's how I sponsor all the major immunology meeting and even country and local as well. And I want to show 2015. This is a BioLegend first time to reach over $100 million sales in 2015. However, it took about 13 years to get to 100 million. But from 2015 to 2018, we hit almost 200 million. That means in 3 years, we make another 100 million. It's like a snowball. Once you have a very good infrastructure, and then continue to run, it's very, very fast. Of course, 2022, we hit another mode, almost from 2018 to 2022, our revenue over at another 150 million. So the snowball is running bigger, but a lot of entrepreneurs, and they're easy to be attracted by offers. So somewhere around under 50 million, it's very sexy. A lot of companies looking for that and more affordable. So sometimes most of them are sold too early, just like a farm engine. So I want to share with everyone. And global organization. So BioLegend, before being acquired, we have in Asia, we have BioLegend Japan, BioLegend Taiwan, and China, our subsidiaries. And in Europe, we have BioLegend UK, in Netherlands, Germany, and France subsidiaries. And all those blue dots is with operations. We have people there to do the logistics center or laboratory for the new product development like Taiwan and Japan. UK and Amsterdam, Netherlands, pretty much for the logistics center and headquartered in San Diego. For this chart to show that BioLegend after 2017, from the research publications, clear we become the leader over past BD, become the leader. And 2016 received the Entrepreneur of the Year in San Diego Life Science. And this share with you some of picture is fun. And then same year, attend the EY Strategic Growth Forum in Palm Springs. Every year, they celebrate the North America, the award to the Entrepreneur of the Year. It's like a Hollywood style, and no one know who are on the final list. Only that day, that night, when you enter, it's a formal ragtime event, and then after you sit down, and then they start to work on each category, and then make announcement who are on the final list. As you can see from the screen, when I saw myself, I quickly take pictures. Oh, that's me, and And all this forum, there's a lot of celebrity there, and they give a talk and presentation. From picture, you can see I had the picture with Joe Montana and Jerry Rice. Those are the 49ers football player. And of course, Barbara, if you watch a Shark Tank, and a lot of fun and counselor as well. So it's fun when you become the winner. And unfortunately, that year, the winner of the year is the Regeneron, Regeneron Therapeutics. And the 2016, their market valuation is about the 60 billion. Today, I think probably higher than that number now, which the company was started in 1988. I'm so grateful that I be part of a finalist. And actually, Biologic 2021, we were preparing for the IPO. Tell you why we are preparing for IPO in 2021. After COVID hit, and all the healthcare becomes so important. There are a lot of hot money thrown into the healthcare industry. And there are so many companies when IPO. If I remember in the past, even the dot-com period time, all the IPO company, at least they are get into the phase 1, phase 2 clinical trial, or pretty much have some achievement, stage 1 achievement already. But 2020 and 21 is an amazing year. A lot of hard money get into. As long as you have business plan, you are able to get the plenty of money and go through the IPO. And in the 2020, after Christmas and the New Year holidays, and return to Biologic campus, there are many of our middle management managers resign from the company. The reason they resign from company because they are working for a smaller company or even start a company because a lot of money. And they seen a lot of IPO company, and their market valuation is so high, and all those employees immediately become the big fortune for them. And of course, then we think at that point, we need the equity. They're asking for equity of the company. So we think you need to give all the employees with the equity. The best way is get the company go public. So in February, we decide to go public and work on the IPO process. And we always prepare for IPO since 2016. All the financial, all follow the public company. So we just plan for any time if we decide to go public. In 6 months, we'll be able to go ring the bell. So that is exactly when we decided in February. So we plan to ring the bell in August. But normally the summertime, August is not a good time to ring the bell because many people go vacation, summer vacation. So we decided after return from September Labor Day holiday that week, they're going to ring the bell. And then we conduct the organization meeting in April. And we fire our S1 in June. And we decided to go with the NASDAQ. And also consider co-listing possibility in Asia in the future. And we have decided our ticker is BLGN. So go back here. So once we finish the organization meeting, and about a month later, I received a couple of phone calls, want to talk. They said they heard Biologic have some transaction going on. Would like very interesting to see our business collaboration. Of course, when I got that kind of phone call, I know what they mean. And so I talked to our lead banker, JP Morgan. So what should I do? It is supposed to be confidential. How come after the organization meeting and then this information leak out? They said, well, this is pretty normal if we can do the dual track. So we internally kind of we have the discussion about this because our purpose for the IPO is to try to retain our employees and talents. And secondary, we can use the money to acquire technology because we believe that this industry is extremely doing well and for the future as well. And the company doing so well. And we can use the money to acquire technology or company. We will continue to grow the company to the even billions revenue company. And third, because of some of our investors invest for 19 years, they may need some money back. So we thought these three main reasons. That's why we move on the IPO, and this is our preference. But since our banker talked about the dual track, and then we said, why not open it up for dual track? So start from June, after we fire all the S1, and open up the dual track. So let those interesting party and companies and to deal with our banker. And they start a visit and conversation. But I don't allow them to come in during business hour. I don't want to interrupt our business. So only visit during the weekend. They all agree to. And then from the S1, those lead bankers, JP Morgan and three other companies, including Bank of America, Merrill Lynch. And you can see, I want to share with you our financial statement here, because it's very important to know how BioLegend do. The most recent 3 years, from 2016 to 2020, other than 2020 due to the COVID year, that's very unusual. We never finished under double-digit growth rate. The only one is 2020, but still we are positive growth. For summary on the left side here, all 5 years normalized organic revenue growth rate about 20% on average, and gross margin about 70%. Our operating margin in the mid to upper 40%. You can see that the worst one is 2020. And our growing investment in R&D, which is double between 2016 and 2019. You can see this is the trend about the BioLegend, continue invest on R&D, new products. And all the bankers, they look at EBITDA is very important. Our EBITDA levels at over near 50%. You can see that from the financial statement. Always maintain about 50% or above. The highest could be 52%. The low probably only 49%. So it's very, very consistent. And then I share with you amazing business history here. BioLegend's capitalization table, a simple story. The founding shareholders, friends and family, employees, and former employees, because they no longer with. They joined very early on, but they left. And no other outside investor, no private equity, no venture capital, no debt at all. All growth funded by cash flow generated by operation of the company. And aggregate capital investment, so total investment to the company, only $4.885 million. So when I present to the test the water with the potential investor, a roadshow, and people asking, did you put the comma wrong place? I said, no, that's total exactly $4.885 million total investment to today. Today, BioLegend, we on campus 5 buildings and without any debts, and also still have over $200 million in cash in the bank before being acquired. And 19 years since company inception, never had the product price increased. For all those reasons, we are able to convince our bankers. In this file S1, page 135, you can see I highlight here. From the voting, you can see we are able to negotiate that dual stock structure, which is Class A common stock, will be newly released to public for 1 vote for each share. So for all those existing shareholders of the company will be Class B common stock, entitled to 20 votes for each share. It's very uncommon. Only bankers only work with those unicorn companies like Google, Facebook, Twitter. You name those unicorn companies. Why Biologic being so highly able to get this structure, dual stock structure? Because I highlighted earlier all those reasons. But BioLegend don't have any technology patent to cover the business, but BioLegend execute every single financial way they were only looking for to hit the point. And all those bankers say they never seen a company like this. So in this is PerkinElmer's announcement. So we first in touch with PerkinElmer was June 25th. And then we signed the purchase agreement in July 26th. That's exactly about 1 month. Why so quick? Because we have all those S1 filed already, and they simply just look at S1. And this is a transaction, like a transaction with a public company. So this announcement for 5.25 billion, of course, is a little bit more than this because we have a lot of cash over, and also they have some incentive program for us. And then what happened to PerkinElmer now? Why they spend such money to acquire BioLegend? Because they want to transform the company to focus on life science and diagnostic company. So recently, in August 1st, they make an announcement. They divest Applied through an enterprise service business to sell this to a private equity firm called New Mountain Capitals for 2.45 billion in cash, so they can focus on life science and the diagnostics. So I want to talk about after acquired by PerkinElmer. Although I'm still the president and CEO of BioLegend as a standalone company and focus on, we are the largest acquisition for PerkinElmer, and we're still leader in life science in the life science section. But personally, I have a philanthropy and giving back to community. Talk about giving back to community is not after this transaction. In fact, during all my tenure with BioLegend started 2008. Once we made a little more money, become positive, I started sponsor AAI, ICAIH, ECI for all those. You can see that means all immunology, either international or the regional, like a European or FIMSA Asia, or the country AI, British immunology, German immunology, and Japan immunology, Chinese Society immunology, and Taiwan as well. And it's embedded in my blood. Always want to give back to community, and by doing that, I always believe that I can learn more, and also when you give me more, I probably receive more, and that's exactly happened. And then also after close the deal, yeah, I want to quickly run, almost done. Just a Legend Foundation, and to continue to translate scientific discovery to therapy. So first one, UCSD will make an announcement soon in Xu Chen and Jinlei Department of Bioengineering. And also I established the Jinlei Institute of Immunology and the Information in Harvard. So that's why my trip to Boston, including this as well. And this will be very excited once in function. And at the end, I want to show that the role not taken. So I want to share with other people. You know, some people have many options. To me, I don't think I have many options. So that's why I choose this way and to start your own and follow your heart. And that's it. I'm done. And if any question, feel free.
Thank you, Jim, for your wonderful talk. Really inspiring. The lesson I learned is you don't need to have a PhD degree to do cutting edge science. You don't need to have an MBA degree to establish a $5 billion US company. And you actually earn your leadership experience through military training. So audience are open for audience question. And any question for Jim? Because the time limit, we have one question allowed. Yes, Judy.
Jim, very impressed by your presentation and particularly your journey. This is Judy Chow, and I particularly want to ask you a question about you chose in the industry of pharmaceutical and biology. Both are, you know, in some sense, pretty competitive in a sense that others may actually go into this field, and you end up to be very successful. What is the thing make you differentiate out of others, and how you kind of led that kind of industry to that success?
Okay, that's a very good question. Many questions like this. Why I choose it? It's very competitive business. A lot of people don't believe I can make it because they say when I started biology, it's so crowded already. Why you still work on that? I said, well, it's different. Very important is my heart. I believe that I can do much better than all those companies because I can execute everything very well. Since I start from private company and public company, I know exactly what public company will do and what they cannot do. So that's why strategy is very important. And of course, and on the other hand, the quality of the product is the most important. So building the company reputation, the product quality, and also you have to know who are your customers. So that's why I sponsor a lot of meetings. I know I attend all those meetings, talking to the people in person and looking for what their needs.
Okay, thank you, Jim. Thank you for speaking to us from San Diego. We hope you are getting well soon. Thank you, Jim.
Well, thank you everyone for listening.
Bye, Jim.
Bye-bye.
See you next time. Okay, so we're going to advance to our second keynote talk. Next slide, please. Yes, I'm going to introduce you Dr. Judy Zhou, and who is the president and CEO of a company called O2 Bio. So when I was asked to put our keynote speaker list, and many people approached me, provide me recommendation. And Judy is the one jump out of the list. And the reasoning for people who recommend is because Judy is new age of Iron Woman in the biotech industry. And I think she has been a great model, a role model for our junior student, postdoc, young fellow Taiwanese community people who has the interest to pursue career in biotech industry. And Judy actually asked me how to structure her talk last night, and she was very thoughtful. And all she tried to do today is try to share her experience over the years through many big pharma companies all the way to establish her own venture at this moment. Without much to say, Judy, podium is yours.
Okay. Let me make sure the microphone works. Good. Okay. It's an honor to be here, and thank Professor Chen for inviting me, and also for the great forum sponsor here. I'm very happy to be here to meet all of you. I named the title, actually, it was exactly my passion for the science, innovation, and for patients. It may sound pretty common, and I'd like to start my journey also a little bit. I'm very humbled by listening to Jin Lei's all his journey to get his success, and echoing that. Actually, I didn't pick on his slide, but also some maybe the start as the young picture. I will start there by talking about, actually, it happened to be this morning, I was reading an article how you make decision. No surprise, they recently published a very well study showing most of us make decision a lot of kind of back to our childhood about a lot of things we do. So back to my time, actually not try to entertain only this audience. I was at the time when during the high school days, Taiwan structured the education of this is Jiayi Bingdingzu, like 4 different categories. And you are in the school, then you'll be really aiming toward what entrance exam is all about. So I was in this school, in the class really for science engineering and nothing to do with the biomedical at all. And nowadays, probably hard to imagine because in this forum, everybody think, well, the engineer and the biomedical should be, somehow be able to merge together. But back to our day, when you are in the science engineering means the biology class, the teacher just sort of teaching you, and I guess in Chinese called feng shui, and basically just like a sort of learning biology. So, and I was very, very aiming toward, I like science. And then I like the science to get to the innovation. That's clear. That's kind of define all my career or what my rest of my life I want to do. But until then, I guess something hit me a little bit about my family and with other members of family have been established, having been the biomedical career. Hit me actually realize that helping people, touching people, be able to interact with people, to me, is a very important task. And I got a little bit worried about if I become a scientist or engineer, I may be like stay in my studio and doing my own things. And that really wouldn't be my passion. So when I add this whole 3 things together, towards the science, innovation, and helping people, and then I got persuaded about maybe I really should switch my track into the medical school. So then I ended up actually in a medical school because a lot of people ask me, and throughout my career, actually when I have majority of my organization, always a 50% engineer and 50% biomedical or biology major people. And the engineer employees of my staff tends to say, like, you are the most non-engineer boss we have, because I would be so thrilled about all those formula and this whole calculation. And maybe there's actually a good reason how I got there. And then I got deeper. Maybe today, in the interest of time, I wouldn't share the whole story about how I switched my track. And earlier, even before this conference, and people call that Ulu Qitu, because I was highly regarded as probably going to be the best fit of a medical doctor. Out of a little bit of frustration about, I feel the limit about what we can offer to the patient. And that's very profound and deep. And maybe one day I can share that story. And I did this very abrupt switch
came to US, went to graduate school, and I got graduate there, and then start my track into more research area. So a little bit about more detail about how I be humble. I cannot say how I got here because I still have a lot to explore to go further. And I, like I say, went to the medical school at National Taiwan University, and then graduated from Yale University. Did my postdoc in Germany, and luckily enough for me, my career started at Harvard Medical School as a research faculty. And then for right or wrong reason, I switched to the industry, and this was totally coincident. I was invited to give a talk and to talk about my research at Harvard, and they invite one of the vice president. I was told that they tried to hire the senior scientist or group leader having always a hard time. And then this guy actually stayed through my talk, and I heard I was the first ever he stayed through because he will be walk out through all those candidates. So then asked me if I would like to be a candidate. I never actually prepared to go into industry. So I was as unprepared as anybody, and went to thinking about meeting with the VP is probably a good idea. Something I will share with you this story. Strike me, then I stood out. They decided to give me an offer because then he asked me this question. For any one of you in the industry, this is a typical question. I ask you, where are you going to be in 5 years? Oh, that's pretty easy. I mean, as a faculty at Harvard, I can say, my research is going to impact people's life and those things. Then he asked me, where do you see yourself in 10 years? Yeah, I have no idea what industry is doing. 10 years, that's impossible. So I was embarrassed. I'm so sure my face turned red, and I was so obvious about to him, I couldn't answer the question. But he was just staring at me. So he didn't let that question go. And I feel like if I smile and look around, then he will let that question go. Because I wasn't there for the interview. So I smile, I look around, and I look at him and say, well, you have a very nice office. And he was like so shocked. I asked you where you're going to go for 10 years, and you told me you have a nice office. And I immediately, after I said, I said, gee, that really is a bad answer. But he's like, wow, okay. So I was told after he said, I like ambitious people. Like in 10 years, this person want to be a VP. But the story may be a little bit inspiring. Indeed, in 10 years, I become a VP of a big pharma. So, but that kind of goes through the journey after we launched Humira. So Humira luckily was my first project when I was at Abbott. And then I moved to U.S.A, and Genentech was impressed about what I do, both at Abbott and U.S.A, and then recruit me from East Coast to West Coast. So I love this. In my U.S. time, I have half by now, exactly half on East Coast and half on West Coast. Be able to really truly understand the different cultures, that probably helped me. I actually then after Genentech, I started my entrepreneurship. Started at 10Vex. I was employee number 1, and many people didn't know that. So in San Diego, so I was a SoCal people for quite a while, and build the company up, get it ready for IPO. It was very clear for 10Vex it should focus on biosimilar. As to me, because at that time I set it all up, and I still in my heart is still some answer yet to be some question yet to be answered. So I decided to move on to the innovative company Medivation. Medivation got acquired by Pfizer. I just come on track to Jim's story. My CEO was the 2017 the Entrepreneur of the Year. Actually, I went to even compete for the world's Entrepreneurs of the Year. So I'm familiar with this whole journey. And because of success, we got acquired by Pfizer for $14.3 billion. And with that, then I was the only executive member got retained by Pfizer to run the entire pharmaceutical operation business. This was the time then Bayer came to persuade me, which has something to do with now my current position. So allow me to take a little bit of time pause here, because then Bayer presented to me about with your research experience and also manufacturing, commercial, and all those. They want to have this once only, I think now it disappeared, unfortunately, after I left, I want to put all the biologics under one roof in a big pharma. So I basically have my biotech company under the big umbrella of Bayer, and then allowed to really from building up the pipeline quickly all the way to commercial. So I was very attracted by that model. And that's how then I got there to run a 3,000 people organization, about 1,500 in Europe, 1,500 in US, and from research build up the pipeline. Fair to say, if you Google it, before my time, Bayer has been on traditional biologics like factor 8, factor 9, and then not really migrate towards a new biologics. But nowadays, before I left, we build the whole model, and Bayer's following the whole track of the cell gene therapy, getting to the most advanced track. And that was the time, I guess then I start have the pause and thinking about, is this what I want to continue of my career? But if you using a funny way to look at that and look at the world, the place you go, where was Judy? And allow me to look at the world this way. This is very different if anybody looking at the map typically, and usually a map doesn't go this way. Taiwan usually is on the far, far east on your most right-hand side. So I like this particular map because I see Taiwan is where I start with, not what I end up with. And I'd like to bring that, be able to go back. And as I share with you the track of my career, and I start from Taiwan, go to New Haven for my education, and then continue my journey further. Sorry, I don't know. And I went to Germany for my postdoc as a faculty. Then I moved to San Diego and San Francisco. And during the time at the TenVax, probably all of you know, I built a lab in Taiwan. So, and then at my Bayer time, I end up have to be between San Francisco. I have my headquarters in Basel, Switzerland, and then 3 sites, actually 4 sites in Germany, and travel quite a bit. So I think maybe you can look at that maybe one step backward. Before connecting this, you see every place I kind of have this 3 times, and probably just what is missing is this. I need to build up further. So taking on this role, it's just a funny way to look at it, to complete the world 3 times in a sense in my words. I was presented by Dr. Peg Yan, and he reached out to me because we used to work together at Genentech. And that's where the ultraviolence idea, because this was not the name yet, it was under a formal name. And I will probably start by talking about this is how I drive my toward helping patients and hoping to have one place to truly realize that. So as we're talking about anything, start with people. I think Jin Lei's question, Dr. Jin Lei's question earlier also, his answer quite a bit of speak for that too. At the end of the day, it's all about people. The story about UltraVal, maybe before I even go into the new leadership, and some of you know, this is a company back to 2000. It was founded 100% by Taiwanese. It was back then, 22 years ago, when government started saying the 4 words are maybe not as fancy as today, 护国神山, like a new generation of the business, was already the thinking about diversify from the high tech into the biotech. And this was echoing and was found by 3 professors out of National Taiwan University Medical School. So it was actually a pretty glorious story 22 years ago, and get a lot of traction, a lot of money, a lot of Taiwanese heart into this. So the Chinese name is even called Tai-yi, kind of really like you claim that space, right? It's the Taiwan issue. It's like a Taiwanese medicine. It started really gloriously, but unfortunately through the 22 years or 20 years before I took on, and it's like went down to the degree almost like it's not just underdog. It's just not even be able to continue to survive. And this was the time, and Pat being a Taiwanese also, he has been invited to take over as the chairman to kind of savage and trying to save the company. So he approached me about this and see if there's anything I want to do. So I guess maybe, in a sense, no matter how I call myself an entrepreneur or not, if I want to live there and doing something really hard, I told myself, may as well just do the hardest one. So, and then this also allowed me to bring something back home, as I just show you the map, not like I every day mapping where I'm going, but I think maybe in my heart, this is so cool about around the world 3 times, there's still that gap about where I go back to where I start with. So Pat's idea, first want to validate the science, then he recruit and in sequence really, Sean Bohan and Steve Jusiga, two were all Genentech in the past, the former leaders. Sean ran the entire early stage research. So he did his due diligence about the ending science. It's interesting. He and I at that time starting talking about it. So I don't want to make it sound like an emotional trip about to save a company. So at the time, he and I identify what we think is the most valuable. And hopefully this will can share with anybody about the experience. And again, we are not a successful company yet. I want to humbly mention about that. Let's take this as a journey I'm sharing with you. So we identify there are many, many things at the time the company working on, but there's one novel science. Nobody else had published actually only to 2004, and Ultrabio back at that time, that research team published and took the route about worry about the world once it knowing it, it may not actually, we can continue to be competitive. You know, this kind of mindset we all sometimes having a little bit. And so let's not laugh at about this, only certain people is not doing that. And then we identify that should be the key assets of the company. So then 3 of them recruit me, and then we also recruit the rest of 2. All 5 of us, this is probably speaking for another keyword about network. We all know each other back to Genentech time. We were at the time the leaders of Genentech. But then the other common track also, we all left Genentech for 10 years. And we all have our own idea about what a company should be, what we think is the dream company should be. So we really restart the company and then give it a new life. And successfully, we get the completion of the 63 million Series A raise, and to be able to really start to realize the vision and build a team on your left-hand side. And definitely the team also is a very important point. If I'm here just brag about Judy Chow can save the world, I don't think that's possible. I luckily be able to attract other people, knowing this is an underdog company and we just got the 633 million, and they should jump onto the board, to the boat, actually, even to drive for that vision. And I managed to attract quite a bit of people. Interestingly, when we do the personality test, I didn't know that, and I didn't do that as a criteria about recruiting, but many of them have this so-called futuristic kind of feature, and that helped us to build a company further. And I'll probably dive a little bit more deeper into help you understand what is the science we're talking about. I don't want to make it sound like it's just an emotional journey about I'm a Taiwanese, I want to do this. But in first slide, hopefully showing you the people, the talent, the network are all very, very important and maybe even critical or essential. And then starting again, back to the science. This is science have a lot of this culture-driven thing, because this was back to, like I say, close to 20 years ago. Those are hard work about hybridoma screening. For any young generation, you may like never heard about this thing, because now everything can be engineered. But back then was just fundamentally ask the question. If autoimmune disease is then your immune system starting to attack you, activate at the wrong time. It's supposed to go quiescent, and then it didn't. I mean, using a funny Taiwanese expression called 老而不死. Like then it's kind of started getting aged, but it doesn't die. And then it starts keeping active to attack yourself. So this group of naive, but really, I guess the word is called a scrappy, right? So like then very naively asking the question about, if we just do the hard work and screen all the hybridoma, and then be able to identify which protein actually help to bring this whole immune system down to quiescent. So that particular question, through you can imagine that's a lot of hard work. So the team back to, when it's generates this company, a Taiwanese, and everybody's hope, did all the screening and identify one molecule, PSG01. Actually be able to not touching the young age of the immune system, but only the old activated stage. This is super important. And I want to emphasize this for anyone of you not familiar with immunology. Because if immunological drug, the key thing is about safety. I'm very proud about being my first project today is Humira. Still going to be very, very proud about this. But Humira systematically suppress the whole immune system. You compromise the time when you be able to fight for the outside attacking agent, but then in order to achieve the therapeutic outcome, that is always the key about only touching the late stage and not touch and not having the impact to the early stage. That will be what we found, or what I found at the time. Why many other reasons, I see this science hasn't been really recognized. This is almost like if we want to have the best brake of the car, the most powerful brake you won't be able to test when the car is stopped. But only when the car is driving, the powerful brake shows its power. And you only want the brake doing that when the car is moving or over moving too fast. So this is the key thing about UltraBio. So then we pivot toward the whole company, totally downplay all the other projects. Have to tell you that's hard because we have, if you don't know, we have 655 Taiwanese investors through all those years before my time. And then coming to the company, have the influence about, but I love the other project in cancer and those things. And I have to tell them about, trust us about going toward this is right. And we're still in this journey. I don't want to make it sound like we're very successful. But this data already built up, not just in vitro and in vivo in non-human primate monkeys, a lot of the investment already put in. And clinically, we also already, the next slide first. This is a very impressive, you probably never heard of any biotech company who's done this. And I think probably you can only call very scrappy and Chinese entrepreneur would do this. We already have 198 patient data on the first generation. And then if you're counting all the healthy volunteers, 244 patient data to support the science. It first comes to psoriasis, because the psoriasis is a skin disease. So you can see, observe, and take sample to prove PSG01 works. Then you go into the psoriatic arthritis that we're touching into the arthritis disease, which is still unsolved problem. And then the ulcerative colitis is a very impressive data. Frankly speaking, I got blown away by looking at data, but nobody actually say anything in the past. And then we actually now, as we speak, very pivoted toward this first generation on the SR-AGVHD. A little bit of background. So SR-AGVHD stands for steroid refractory acute graft versus host disease. This happened to hemolytic cancer patients. And when chemo and radiotherapy don't work and have to go through the bone marrow transplantation, you already see that's quite a bit, right? And not forget, this is a cancer patient. And comes through the bone marrow therapy and then starting to have a rejection and having a lot of problems that will kill you. And then you use steroid, of course. And then when you use steroid, and then you have the refractory, so you have steroid introduced problem to you. This is like a terminal. This is impossible. And if you also imagine this patient gone through therapy after therapy. So there's really no cure or no treatment. And then actually we think, okay, if all the other indication is it did work, but it really requires much more patient-friendly dosage form, and we will then find a newer molecule to do that. But then having this ground-based to the medical need, as hopefully then you ring your bell as talking about the science innovation for patients. So we're very passionately looking into that. At the same time, I'll come back to kind of introduce our second generation. Oh yeah, even the number of the project you can see is a lot of Taiwanese color, right? This is 168, the U.S.A. And then We say, okay, continue our track, let's do ALUVA. So then we have a new project called ALUVA. If you look at follow our company closely, it's not my invention. 168 has a name for aiming toward the one day when we get into the commercial. It's called Nehulizumab, and why Nehu? That's Nehu. So there's branding there. I want the whole world to know one day it's going to be well known about in Taiwan. This is such a place. And then 26A, actually the name is called Lioluzumab. And Lioluzumab, why Leo? That is Taiwanese Leo. So that's, you see the ambition of the company is one day we want to make our name on every patient, every label, and to be able to do the injection, even you don't understand them, you know there's such a thing where it's coming from. But anyway, continue that dream. For 26A, we have shown higher than at least 3 times higher efficacy. This allowed us to use less volume for injection. So then we can develop the subcutaneous. It's thinking about the patient have to be dealing with the disease in the long term. We'll be able to do this like insulin. You do this at home. Self-administration will be better. That's why the strategy is to put all those chronic disease into the second gen to use 26A. And then still pivot towards the 168 on the GVHD. I will probably just quickly go through some clinical data, but not to take too much time to get into too technical. And the reason why I say UC, so this is ulcerative colitis. It is an autoimmune disease when your immune system attack yourself, and then it really making the whole GI system kind of puncture the hole and attack the cell. Very miserable situation. The key feature of the key symptoms of the patient will need to really go to the bathroom almost nonstop. When this happens to any younger generation patient, there are a lot. Usually they just couldn't even stay at school. And I want to focus your attention on the data we have on the green bar. So this is compared to Humira. I mean, it's nothing better than Vidocell. So Humira in black, and then also Entyvio in blue. And you see in all the criteria, it looks better. So this is giving the face about once we build the 26A, we'll be able to continue. Another indication, psoriatic arthritis also. This is 168 data. You can see in all the criteria, maybe I'll make your attention just at the week 12. This is the last dose, week 16. And beyond the dosing, you can see the ACR score, and allow me not getting into too much technical. This data has been generated. And for many of those scientists and engineers here, you probably agree with me. A lot of time it's not about generating data. It's about how you read the data. So then what we bring the new life to the company is looking at this data deeper to see, hey, this has something beyond other people didn't see. At the time, also humbly as a Chinese company before my time, we say, well, you know, there's IL-17 that's on the market. We can never compete with that. Actually, by now, there are more commercial data. We're seeing on the right-hand side, the ACR score is really not bad than ICR 17. So those will be, we put on the capable hand of the 26A, our second gen. And this is what I mentioned. I only focus your attention on your most right-hand side, the one highlight, median survival. As I mentioned, those patients have no hope, but just frankly speaking, very sad. The way we recruit a patient, the doctor only need to ask the patient one question. Do you want to go to hospice now or try this drug? So that is where then this drug, we anticipate we'll close the trial September this year. We actually close in April. And we've been able to see the median survival day for the patients are higher than 140 days already. So this is really live or die and a big life-changing event. We will continue the 168 toward this direction. And for the UC, and maybe just the last couple of points, and this slide, the key thing I want to relay a little bit also is for the science and innovation, and I can talk innovation nonstop, probably quite intensively. But the key thing for innovation is about strategy. So this is just to showing you our strategy for, even I say it's very impressive data in UC. We'll be very focused on the the refractory. So the biologic refractory patient, those having almost no other drug to treat, and then trying to get back to the first line. having the right strategy is probably very important, because if I was just here naively tell you, now we have the great team, we know how to do this, and this is a novel science, we're going in, we'll still do this, I guess in Chinese, we like to say KP, and like directly just competing with that, it's not going to work. So we will be very focused humbly on the biologic refractory, meaning the patient in using other drugs that don't work yet. And then that is a medical need. So if I allow to make a mark about when I say innovation, I have this inspiration talk with my team, basically saying, let's just set anything we do. If it's not going to change the standard of care, don't even do it. So this is what, if I'm telling you, we're going to go to the non-biologic refractory, it's just adding one more drug. It's not going to change the standard of care. We are not really bringing new solutions. So what we are really aspire to do, still, you need to have the business sense about not just keeping knocking on something, not going to be very successful. It's really the whole future about the market. Why we're so passionate about this mechanism, because if you look at the most right-hand side, the whole market size is huge. Even we just humbly say we have a market penetration of 10%, we're talking about billion-dollar drug. And you can imagine with such an experience, they usually, I mean, we end board meeting, they only tell us, oh, it is going to be a blockbuster drug anyway. It just, I mean, the thing is just you don't screw up. So I think that's the pressure I have. With that, I think I want to probably wrap up. So basically for UltraBio, we focus on the first in class. Is really, that's hard, and I don't want to make it sound easy, because the PSG and so far still not a very common, well-recognized target. And we have grade 168 data to support clinically, and we're going to 268, actually very excitingly, this next year going to clinical. And we have the significant milestone upcoming for us, and good management team. As I mentioned, we do have the cash, allowed us to kind of push in this dream even further to show the world about this is it's a great target. So I will conclude my talk, and I share this vision about myself, and maybe you can call kind of always not really following the common track. Throughout all my childhood in Taiwan, and maybe all in the career, this is how I feel myself as that little red fish. I feel like I'm this happy, smiling little red fish, but all other people are doing all different directions. I want to brag about I feel good. Frankly speaking, not. Because if you can look at that little red fish, it doesn't have the defined track and path for you. So a little bit echo also what Jin was saying earlier. Don't too much worry about your past. I guess what I learned, how I be able to progress my career really is to stick to that dream. And I like to, really cannot laugh at people around me. And most of people, and for any of you entrepreneur, and we're going to, no matter it's a precision medicine or the digital medicine, they're going to sometime you'll feel this way. But if you know what you're doing, and you know your passion, and being also realistic about those things, as I share, the strategy, the team, and you're going to go somewhere. And I feel like then, lucky for me in my career, from time to time, I actually end up to be, I feel like actually other fish is following me at the end. So with that, I want to close my talk, and thank you for your attention. I'll be happy to answer your questions.
Thank you, Judy. I think we have time to allow 2 questions. Nini?
So you talk about you change your career because people ask you for your 10 years plan. So I would like to ask you, for UltraBio, what's your 10-year plan? That's number 1. Number 2 is Taiwan people usually very short-sighted, have a little patience. How do you deal with it?
Great question. So what do I see UltraBio in 10 years? We absolutely need to launch this drug. So maybe 2 visionary talk, and one on PSG01. This is going to be the one first to present the value for patients. If we be able to successfully launch, particularly 168, we will see that it will change people's life. That's what I think within 10 years we'd like to achieve. And second question, Yeah, the short-sighted, oh yeah. So the short-sighted question, that's actually really, really tough on my daily life. As I say, I have a 乡亲父老, 650 some. They all understand. I mean, like the people back at that time hoping to invest the company. Maybe allow me to extend that question even further. After the pandemic got a little bit more quiet down, I went to visit them. I really don't need to, right? Many people told me that's not your job because you didn't invite them to invest. And I really touched in my heart a lot because I went to, rural place, and also they have aggregated a lot of money for this company in the past 20 years. And then there was one woman came to me. Then she told me, I'm so glad to meet you today. My husband invests in this company and we getting to fight every day about this. I told him it's such a bad idea. Then he got our money in this company for 20 years. I was so upset. He passed away last year. So her husband passed away last year. And then she say, now I see you. I can go to his graveyard to say you're right. So my hope is to help people understand the patient has this price, and we will be able to still fulfill this dream. I do think one day if the L2 is successful, and then we can go back talking about many those 20 years of people's sweat and tears and heart and all the story. It's a turning story. If it's not such a scrappy group, being like nonstop, still stick to it and get to the point to meet us, it's probably not going to be there. So the short-sighted part is hard to address to change your mindset, but I will say two things. One, not just the longer term, but also show some early success. And in the interest of time, I will stop here, but definitely I think I learned a lot through my Bayer time as well. Bayer is a great organization, but also a very conservative environment. I would not answer this question by showing like, just building the trust. I think you need to have some early win.
Dr. Cho, I'm Edwin Posadas from Cedars-Sinai. Congratulations on your scientific work and how you're bringing it to market and to patients. More importantly, as a practicing oncologist, I'm glad that you're developing drugs for acute GVHD, especially in the steroid refractory space. But these patients rely upon the graft versus tumor response to maintain disease balance. So, as someone who is innovating in this very difficult area, finding the balance between both positive and negative effects of the immune system. One, how are you addressing the concerns about losing the graft versus tumor response? And secondly, speaking of long-term ideas, where is the sweet spot in the balance between getting to the steroid refractory point and going to the point of no return, since many of the patients with chronic GVHD have already seen a lot of systematic remodeling of their immune system and the tumor microenvironment that at some point we really can't do much about anymore.
Great question. I think that's very insightful. The reason why not a lot of people want to go into the SR-GVHD is exactly a reason, as you point out. First question first, how do we address the graft versus tumor? I think this is probably I should answer your question by like a second part, you say the answer, right? Really finding the sweet spot. If I am here telling you our drug won't have that is that's contradictory to myself. I would never want to say that. I heard other people using that answer to promote their drug. I don't think so. I think if we don't have the effect to attack the T cell to bring it down, but we don't see that. As I say, our current data supporting, as I say, this break theory. Right? We only work on the activated T cell. We don't particularly like acting on the quiescent or the resting T cell, the memory T cell. So that's what I can say so far. We need definitely more study to decipher down to that detail. And your second question about the sweet spot. Yeah, I think that's, we still need to find. Currently, our goal, and based on our interaction with the FDA, it looks like they're pretty aggressive on this. And I'm really not going to brag about this because then in our recent conversation with them, they just, at some moment pause and say, how can we help you to be your market advisor to get this to, and that's probably no surprise, we got fast track designation. So what I meant to say is then we probably need to kind of bring to the market as fast as possible first. And then we need to do some adjustment. A little bit longer term goal, and you kind of made me reveal about our thinking also more about the chronic is then in chronic stage to have patient coming to inpatient weekly IV dosage is non-realistic. So that's actually even more strategic for us if we have 268 there. This is the same mechanism. I don't get a chance to talk about it. We leverage exactly the same mechanism. Don't want to change it to have a different kind of science. So hopefully that can provide some additional hope for patients for the longer term. But absolutely very tough to address disease. And a little bit kind of bring it back to the business for the rest of the audience. This is almost like the, sadly to say, most of the investors don't want to do anything about this because this is the patient population is small and the point of low return is there. But I guess a little bit about, like in the very beginning of my talk, I mentioned about everybody go back to where you begin with. Coincidentally, as at medical school, I was trained as a hematology oncology physician. So my heart, I'm getting a little bit emotional, still thinking about that time, looking at that no hope and how got me frustrated to where I am, to want to study more, and I don't want to give up. So I guess hopefully we can still do something about it and make it still, I wouldn't call profitable, but beneficial for the patient to begin with.
Okay, thank you. Thank you, Judy, for your work on the immunological diseases. And yeah, thank you. So let's proceed to next talk. Okay, so welcome to advance to next talk. Yeah, I'm going to introduce you Dr. Ya-Zhen Zhu. And this is interesting transition. We move from two very successful company. Now we're going to step into a junior faculty at the UCLA, and she's working on developing new diagnostic technology. I think the emphasis for Dr. Zhu's work is going to be translational science for precision medicine. And briefly introduce Dr. Zhu. Dr. Zhu was a physician scientist, actually, I should say pathologist, work in the clinical laboratory analyzing tissue for 7 years. And she suddenly decided to quit her physician's job, join UCLA to start her research career over. And she is currently an assistant professor in UCLA Department of Pathology. And she will share with you her venture in translating technology developed in bench side into clinical setting. Dr. Zhu, podium is yours.
Thank you for the nice introduction. And I want to say thank you to the organizers and to the sponsors to make this happen. And thank you for the invitation. It's my great honor to be here to present our research projects on behalf of our liquid biopsy laboratory. And today, I'm just focusing on the science innovation only, no entrepreneurship at this moment, maybe in the future. And my topic will be translational research in liquid biopsy for cancer and prenatal diagnostics. Okay, first of all, I want to briefly introduce what is liquid biopsy. Currently, there are 3 liquid biopsy approaches. There are circulating tumor cells, we call it CTC, and extracellular vesicles, EVs. There are vesicles released by tumor cells and normal cells. And also cell-free DNA, there are DNA fragments released by all cells. So I want to compare these 3 liquid biopsy approaches and then give you a very clear picture why we focus on EVs. So first of all, let's look at the ctDNA, or overall we call it the cell-free DNA, and tumor-derived cell-free DNA, we call it ctDNA. So the problem for cell-free DNA is that purity is a little bit low, and the problem is that it's impossible for further purification because all of them are double-stranded DNA. There is no target we can target on that to purify. For the circulating tumor cells is a very good target in the past because the cells are have very extensive signals in that. In addition to DNA, we have RNA and proteins to target. And also it can be purified by targeting the cell surface markers. And among them, there are the third type of liquid biopsy approaches, that's EVs, extracellular vesicles. So they can also be purified by targeting the cell surface markers, very similar to CTC. And also, as you can see, the starting purity is a little bit higher than the CTC. So based on this, we focus on the EVs because it is easier to purify them, and we can start from something with higher purity and further purify them. So I want to give you a reason why we want to focus on to purify those liquid biopsy materials. So before I joined U.S.A, I was a pathologist. So I did the tissue every day. Sorry about that. So I did the tissue every day according to the SOPM guideline. So whenever you got a piece of tissue from the tumor, right, you need to ensure that the tumor percentage is very high so that your downstream molecular analysis will be very reproducible and robust. So in short, if you put something like this, you cannot guarantee that your downstream analysis is robust. However, if you put something like this with the tumor percentage up to 90%, 80%, then your downstream analysis will be very robust. So very similarly, in the liquid biopsy setting, we hope we can develop some strategy to purify those circulating materials in the patient's blood so that we will have a more robust and more reproducible assay for the downstream. With that in mind, so I joined U.S.A Liquid Biopsy Laboratory in 2017 as a full-time employee, full-time researcher. So based on Dr. Zhen's previous work, I don't know what happened for this. So based on the previous version of our technologies, in the past decades, our lab actually focused on the technology development for isolating circulating tumor cells and circulating trophoblast cells, those circulating rare cells. However, I think with that as a foundation, we can do better job to purify the extracellular vesicles using similar platform. So I probably don't need to explain too much about click chemistry because this year click chemistry won the Nobel Prize. And in briefly, the click chemistry is two click chemistry partners we call reagent 1 and reagent 2. They actually react very quickly, very highly reactive click chemistry partners. So by this click chemistry, we can just tether one click chemistry partner into our capture reagent and the other click chemistry partner onto the cell surface or the EV surface. So by taking full advantage of this highly reactive click chemistry reagents, we can use them to purify our targets in the circulation. So with that in mind, I want to give you a good example how we apply this advanced technology in clinical settings to address those unmet needs or very important clinical questions. The first question we want to address is we want to use our technology to do early detection of a liver cancer we call it HCC, because this is a big problem in clinic. So this is a busy slide, but I want to show you that if we can detect the liver cancer earlier than the estimated survival time is much, much longer. So early detection is the key for the HCC patient. Here is the detail about how we develop the assay. So on the left-hand side, we have the purification process of HCC EVs. That's the extracellular vesicles released by the tumor cells by our EV click test. So we take advantage of the click chemistry. As I mentioned, there are click chemistry partners. Reagent A here tethered onto our target EV, and reagent B tethered onto the capturing antibody. So when we load our samples into the our platform, then the click chemistry reaction will be very high efficient, and then the cells or the EVs, extracellular vesicles, can be captured onto our click test. And then with the purified material, then we can conduct some downstream analysis. Because we have high purity at this moment, then the downstream analysis will be much, much easier. For example, we just use the PCR and to quantify the purified EVs. And the first time we purify the EVs, and in the second layer, we focus on those HCC-specific genes or HCC-specific signals, so that those unwanted or not specifically captured, those background signals will not be read out here. So the second readout will ensure the specificity further. Here, we show the extracellular vesicles was captured onto the top of our tip, and also the fat wall of the silicon nanowires embedded into the chips. Here is the result. So the HCC EV-based digital scoring assay is our final assay after we optimize. As you can see here, the HCC early stage, BCLC stage 0 to A, comparing with the signals expressed on the BCLC stage 0 to A, that's early stage HCC, versus the control group, that's the liver cirrhotic group. You can see very obvious differentiation. And the area on the curve here showed that the performance of our HCC EV-based digital scoring assay is 0.93. The matched AFP, which is clinical routinely used marker AFP for HCC screening, that's the AUC is 0.69. So our HCC EV-based digital scoring assay outperformed the matched AFP assay in the same population. So we published this paper in Nature Communications. So if you are interested in this paper, and I will show you the details later. And our next step is we want to conduct a phase 2 clinical trial to enlarge the population to make sure that this is more robust and reproducible in a larger cohort. So we will publish this piece of work later. I want to emphasize that this is just an example. Actually, this is a platform technology. You can not only apply this platform into HCC to answer the very important question, we want to detect the cancer earlier, but also we can apply this technology to other disease types. You will see later on our collaborators will give you some more examples about this platform can be applied into other disease types. And another example I want to show you today is that we can not only detect circulating tumor cells for cancer diagnosis, but also we can capture the circulating fetal cells. We can also capture circulating fetal cells, and we call it CTD, circulating trophoblasts, for prenatal tests. I want to show you this disease spectrum that's called placenta accreta spectrum disorders. It's very nice that our students, our high schoolers, they have an outreach program. They will show the PAS disorders in more detail. This disease, you just go to the poster area and the kids will explain very detailed information about this disease. And basically, this is a placenta embedded into the uterine wall too deep. So this is a disease placenta. So as you can see here, normal is just very superficially attached to the uterine wall. If you go too deep, deeper, and then the placenta accreta, we call it accreta, increta, and accreta, that penetrates the uterus wall. It's very dangerous and it's the life-threatening disease. It's incredible that even in the United States, about 2/3 to about 1/3 to half of the PAS, the placenta accreta patients will be undiagnosed until it's too late, even until delivery. So this is unacceptable. So we want to develop a non-invasive early screening technology to address. So this is the first CTB, the circulating trophoblast cell cluster detected in our platform. I'm very excited to show these clusters to you. And I still remember when the first time I discovered the circulating trophoblast, the CTB clusters on our chip, I shared with Dr. Zhang, who was traveling in Taiwan. And in addition to cell enumeration, cell numbers increased in this population, we found that they attach the class, they're attaching to each other and traveling together in the circulation. And I think this is just a common sense. I was thinking why they want to stay together, why they, just travel together. So I think common sense is that because alone they are strong because they invade into the uterus wall, and together or united they are stronger. So it's kind of more severe disease type that indicated by the cell cluster. So I believe that was very important discovery for this disease. So in order to demonstrate that this is a very important discovery in this patient population, not in the normal pregnancy. So we need to conduct a clinical study. So we start to collect more patient samples, not only in the United States, but also in other sites. So as you can see, we have this clinical study design and have a training cohort, and then independently validate this assay. So again, seeing is believing. I want to show more images to you guys to kind of get a sense of this is a single CTD. So you can see just one cell, and this is the clusters, and with 2 CTDs, a clusters with 8 CTDs, and even bigger clusters with 15 or more CTDs travel together. And more pictures here. And we did a statistical analysis later. As you can see, the orange bar indicated the cluster CTDs. As you can see, the majority of the clusters located in the PAS group, and in the previous, that is at-risk group. And the normal group, they have very less cluster CTDs, and the statistical data showing here, about 86% of the cluster, about 86% of PAS patients had the clusters, and only 22% and 14% of the cluster CTDs in at-risk previous group and normal pregnancy. And in the non-pregnant female healthy donors, there's no CTDs detected. Even single CTDs cannot be detected in this population. So that's a very good control to show our to show the specificity of our assay. So this is more detailed statistical data to show that the difference is statistically significant. So here comes the most important conclusion. The area under curve for CTD ASA with or without ultrasound. We know that the pregnant people will go through the ultrasound detection anyway. And in the first trimester and or even second trimester, the ultrasound is not sensitive enough to detect that PAS. And our ASA can be very helpful in the first and the second trimester, I mean the early gestation age. So as you can see in both the training cohort and the cross-validation cohort and independent test cohort, our ASA remains very sensitive and very specific with area under curve of more than 0.946, 0.947. And it's very interesting that if you combine the ultrasound, which is readily available, you can see they can be the area under curve can be even higher with the area under curve of 0.978. And in the subgroup of the earlier gestation, only trimester one, only the first trimester and second trimester, that's the gestation age less than 24 weeks. The area under curve still remains very good. And this table also published in Nature Communication last year. And with that, I want to conclude my presentation, and I'm very happy to answer any questions. Thank you.
So, question for Dr. Zhu. Yeah.
So Dr. Zhu, Ed Posadas from Cedars-Sinai. You know me very well. You know, as a physician who has to deal with emerging technologies, I know you're working closely with Dr. Pizarska. One concern that a lot of doctors face is how to interpret new tests and how much they cost. So, you know, in the cancer center, I'm always challenged with how we're going to bring this into practice and how it will impact things, how docs will read it. You know, given the importance of this, especially to maternal fetal medicine, how for market share, for adaptation of this technology into practice, I mean, do you see barriers in terms of cost or technology that could present barriers to use of this, given the potential importance for diagnosing these you know, very morbid perinatal conditions?
Yeah, that's a very good question. So when the first time I want to develop this assay, an idea in my mind is that I want to make this assay is readily available in the future in low resource setting. Because as you can see in the United States, even in the United States, the ultrasound is readily available everywhere. And the PAS, even undiagnosed, remain undiagnosed in 1/3 to half of the patients. That's a very serious problem, and I think it's unacceptable. So in the future, if we can develop a very cost-efficient assay that will not rely on people's, I mean, the physician's expertise, for example, the ultrasound in low resource setting, probably the ultrasound expertise is not available. So this will be a very good solution in the bigger area and even globally. So in terms of the cost, I think probably Dr. Zheng is a very good person to answer how long the assay will go to the clinic. And I think it depends on the, a lot of entrepreneurs, their help, and also I hope in the future this technology can be translated into the clinical setting. Yeah.
Okay, so let's close our session at this point. And I would like to invite Ning to come with our two speakers. Judy, would you mind to the front, and Dr. Zhu? And we have a small certification and a souvenir to present to both of you. And let's go to the front. I think it's okay because we tested already. Screen won't show too much on our face. Okay, it's white, you see, right. So, Ning, so are we going to take a picture first or present the work? Okay, let's present the certificate first. Ning, it's for And souvenir. Okay. Thank you, Nini. Thank you, Judy. And for Dr. Zhu. Then you should have a photo together. And I will collect check, yeah. Yeah, we're gonna have a group photo, please. Yes. Okay, thank you. Thank you all speakers and thank you, Nini. And a friendly reminder, we have exhibition in the back from our sponsors. Go look at, check their booth. And second, we are going to shorten this break period to 5 minutes, okay? So please come back 5 minutes later. Thank you so much.
You.
That's your little one?
It is.
Will you join us? Hello, hello. If you don't mind, please get seated. We are going to start our session 2. Hello, our volunteers, please help mobilize our participants to get seated. Hello, 有人 please help mobilize our speaker to the our participant to the front. Okay, I'm going to officially start our session 2 of our precision medicine session. And so this event a lot is about networking, and it's about friendship and family, kind of in the way our doesn't matter it's a business or academic family. So I'm here to introduce you the chair of our session 2, Dr. Alan Wang. So Alan was my junior at the college time. We mingle a lot and running on Tonghai University's campus and knowing each other so well. And that is kind of the time we start to develop our commitment to each other. When I mentioned we are organizing this conference, and he decided to come to support us. It's not only support us with his presence, and also with his network link to Jerry, who is our speaker in the session. And so again, I really appreciate Alan's loyalty to friends like us and our academic family network. And to Alan, without much to say, and look at the title I put up there. He is a J&J medalist. Okay, it's more like a Nobel Prize in J&J system. And if you are a surgeon practice surgery, you definitely know Alan's invention about surgery cell. And I'm going to hand it over to Alan and let him introduce his classmate, I assume, Jerry.
And thank you, Dr. Zhang, because this is kind of unexpected, because I was asked to be a chair of this section. And so today I'm going to introduce Jerry Lee, and actually he was one of our schoolmates when we back in Hopkins in 2003-ish, maybe, yeah. So it's very glad to see him today, and almost like 20 years we haven't seen each other. And also I see, at that time, of course, he was single, and now he is a little one over here. So very glad he has a group of family. And so then I'm going to, so Jerry, he's the current associate professor at U.S.A, and he probably is going to talk about the from engineer perspectives to talk about to cover the entire what was the topic? Apologize. The interface of the science, engineering, and medicines. Okay, so let's transfer this over to Jerry, and he's going to give the talk. Thanks.
Which time should I plan for? Okay, perfect. All right, thank you, everyone. I hope you guys can hear me in the back. And thank you so much to Shenrong and to Alan for a warm welcome, and of course to the committee members and organizers of this meeting. As mentioned, I'm going to do my best to give you sort of an engineer's perspective on what it was like to support, fund, and really push cancer research over the past decade at the Cancer Institute, and also talk to you a little bit about my new organization, the Ellison Institute, over near Santa Monica, LA area. Just real quick, my disclosure statements. I'm going to talk a little bit about my work with the Department of Defense, and I do serve as a consultant to the Merck Cancer Center Director. I serve on the Scientific Advisory Board of Atlas Exomics, which I will touch upon as well. But most importantly, the Ellison Institute, we are a for-profit LLC. We are affiliated with the U.S.A, but we are our own entity. And I do serve as the Chief Science and Innovation Officer for that organization. So I think everybody had a little bit of their picture, and I think for me, I always want you guys to know, what does it mean when I say my perspective? What did I see? What did I do? And of course, as Alan mentioned, this was me in graduate school. Everybody thinks that I was 12 years old there, probably. I owe a lot of my genes to my father. Really appreciate that. But in 2006, I had the really good fortune of meeting Dr. Sang and seeing Dr. Su's presentation, what was just an idea in his mind, and then in her mind, coming to fruition 16 years later is always exciting. I'm probably not going to touch upon all of the programs that we did, especially the students, as mentioned already. I appreciate taking about a year off, really, when Dylan, who's sitting in the front, was born, and taking some time for family. Sometimes it's not something we talk about. And that is something that my boss, when I was at the Cancer Institute, I always reported to the director of the Institute. For some of you that don't know, the National Cancer Institute is the only one that has a presidential appointee. So my boss changed, every couple of years, including the one that just got sworn in a couple of weeks ago with Monica. So I was very fortunate at that time. Doug Lowy very much said, no problem, go take a break, go take a breather. And that was something new at the Institute. But then he came back and he said, well, you have a new job, because I got a call from the White House, and please go down to the White House and help former Vice President Biden, now President Biden, with the cancer moonshot. And then after that, of course, a second break. So Dylan has a little brother, Kai, who's not here. And we decided in 2018, and I'll talk a little bit about that opportunity, was asked and recruited here by Dr. Dave Vegas to serve as the Chief Science Officer at the Institute. And in the spirit of trying to be latest and greatest, I actually just got an appointment in our quantitative and biology, computational biology division. I'll talk a little bit about that for students that are interested in computer science and informatics, including the second day where we are going to talk a little bit about AI. So if we could sum up, my entire career to date, I would say my entire passion is on making data fair, findable, accessible, interoperable, and most importantly, reusable. As a chemical engineer, that's really something that we learn. How do you make a feedback loop? Because if you don't have feedback, you don't know how to optimize. And if you don't know how to optimize, you're not really building anything. You're just kind of like fitting data. And really, not just because Dylan is here, how do you make this data reusable to train the next generation that might need to use that data to continue to sort of build upon and create new innovation, 10, 15, 20 years later? And translation is very important to me. Some of you may know I was actually born in Taiwan, went to school in Taiwan. So coming here in the United States as a middle schooler, being able to understand what everyone was talking to me about is important. And some of that really comes to, as I think about translational research, everyone almost always thinks about T0. How do I get preclinical into animal, animal into human, human, and then we're done, right? We're not, as Dr. Chu mentioned as well. Taking those translational science to human studies, that gap between now taking these new interventions and changing Dr. Posada's, perception of what intervention to use, what therapy to use, this is challenging. This is challenging to measure, challenging to understand. And to Dr. Posada's question, to Dr. Zhu, how do you think about value? Value-based healthcare needs. The challenge is we don't have a feedback loop. We're not optimizing. We're doing this. We're doing this all the time, one way. And most importantly, and I really appreciate that Prasada's question as well, each of these data spheres has its own lingo. And what do? We take these little orange arrows. We have computational scientists that maybe understand a little bit of PKPD. We put them into this realm, and then they kind of learn a little bit about clinical trials. And then they learn a little bit about this, but many of them don't really get a chance to jump all the way out to the T4. They don't necessarily understand clinical utility. What is that clinical context? So maybe they are gathering great data here, but it may or may not be particularly useful if you don't have a physician scientist giving you that context. And how important is this clinical context, in my opinion? When you look at things like pre-analytical validation, analytical validation, clinical validation, and clinical utility, where we always get tripped up, having funded this type of science at the Cancer Institute for over a decade, this is where we get tripped up. We get tripped up because it takes 1 to 5 years maybe to do some great pre-analytical work. Maybe you want to take it into analytical. Maybe you're doing 10 cases and there's sort of 10 cases, 10 cases. And what do you know, our NIH grants are 5 years long. So why is it that we can't cross this sort of boundary? Sometimes it's just you run out of runway. And how do you best sort of address that, I think? And I just want to be very clear, despite all of that, and we've heard that already, cancer has really progressively been redefined over the last 20 years. Each one of these pie charts here is an actual biomarker that then drives an actionable decision. And if you're thinking about living in, 1996, 2006, and even 2016, just the amount of progress that we have seen, some of that is technology driven and some of it is not. I'm probably not going to get through all of this, so I did tell something wrong. I'm going to try to keep this to about 10 years worth of perspective. But what did it look like 22 years ago? And that's important. Everybody thinks that precision medicine, genomics medicine was like last year, 22 years ago. We all thought, here we go, we have the human genome. I'm going to walk into a pharmacist, I'm going to hand them my sequence, and this pharmacist, the only thing that I would say is probably accurate is we are generating bottles and bottles and bottles for every single subtype. How does a physician, how does a caregiver, how do you give that shared decision making when you think that piece of paper might or might not have all the answers? And then when you think about just how quickly, and when I have to teach now undergraduate students that are sort of born 18 to 20 years old, I'm thinking about that 20-year timeline because to them, I have to explain to them what a USB stick was. And we celebrated 8 megabytes of USB stick. First selfie, first camera phone probably, showing them what iTunes means. We were celebrating a 10 gigabyte iPod that had no other functionality but listening to music. Many of them know what the sound of a fax machine is. Some don't even know what a fax machine is. And that is still how we predominantly in the United States exchange healthcare data. And that's a problem. Right? There are certain things that, you're told not to talk to strangers, and now we get in cars with strangers. So how do we think through that generation? And of course, this is Dylan, this is Kai. But how do we think about building this for them? And in that same 20 years, we all, of course, celebrate things like Herceptin and Gleevec that really kicked off our, hey, could we make another magic bullet to what Paul Ehrlich and Gene sort of talked a little bit about as well. A little bit of a pause, but that's fine. I would say for me, first things first, as Alan knows, as chemical engineers, we always think about time scale, length scale. So when I got to the Cancer Institute, one of the things that I was surprised by is how little discipline sometimes we had talking about that. And whether you talk about it from a spatial perspective, that primary tumor, metastatic tumor, or regional, you can also look at numbers. We don't want to be here. I'll probably talk a little bit about that. We also want to make sure, how do we get more and more to this side, to what Dr. Zuzman showed as well. And the challenge with that is each one of these diseases has different latencies, whether it's a breast cancer, lung cancer, colorectal cancer. And the simple chemical engineer here says, well, you're going to bone, lung, brain, things that are sort of taking longer, shorter. What makes it different? How do you think about time scale? And how do you measure in time scale? And when I got there, I would say our entire community was sort of taken back a little bit. Some of you may know the Lancet Petrocoin study in 2004, where a new technology, to Patrick Sada's question at the end of the session, how do you know your latest and greatest tissue technology, radiology technology, blood technology? How do you know that you're measuring what you think you're measuring? And I think that sort of set the stage for me a little bit, looking at that time, 2006, 2000, how many of these data sets were sort of in the public domain that were single time point? I know I'm jumping ahead a little bit. Some of you probably will have used the Cancer Genome Atlas or other for-profit efforts like Orion. How many of them are only shared amongst partners? I think the scary part for me is how many are not longitudinal and how many of them are not shared. And not having this space sort of more covered, how are we going to tackle such a heterogeneous disease that's both spatial and temporal? And so for me, it has continued to be that engineering thing. How do you look at both that primary, second, those three major events, but also the wellness events in between, recurrence, prior to recurrence, monitoring? How do you think about all of these different states? And why do I care about those states? From my perspective, you know, a lot of those 20 years worth have done great for pediatric cancer, have done wonders for those in the 5th, 6th, 7th, 8th decade. I think the part that I think I'm still sneaking into this area, we've done little to nothing from my perspective in the United States for adolescent young adult cancer. They're hard to find. And on top of that, I'm happy to be guilty of that as well. There's also the group that might not take the early detection to their GPs as often. But how do we address this issue is something that I'll talk a little bit about as well. So just rewind, this is the 16th anniversary, not just of myself meeting Shenrong, but also the start of the Cancer Genome Atlas. How many of you guys use that? 1, 2, few? Okay, great. Didn't want to talk to an audience that was like, I already use it. You know, what we tried to do there, feasibility of using large-scale genome analysis technology, which seems like silly now in 2022. But some of the things we wanted to make sure it was publicly accessible. We were able to protect and respect research participants. How do you do both of that? I think that was one of those things that was like, there's no way. It's not going to be public. It's going to be behind a firewall. Never going to happen. The reason people said that, and this is a Johns Hopkins Lilly study, there's not a Lilly colleague here, published, you know, a couple of weeks before we started and said, this is it. There's a 29-person team, 11 breast cancer samples, 11 colon samples, 189 candidate targets. This is a treasure trove. We're never going to stop. We're never going to be able to finish analyzing 189. This would be, you know, rejected probably without review in 2022. All right. How do we do 3 cancers, aggressive timeline? Everybody's very skeptical about the Atlas being something achievable and doable. So for me, because I'm an engineer and I was not, you know, I worked with Bert Vogelstein. I wasn't exactly a hardcore, this is my favorite gene. And, you know, I showed up in 2006 at the Cancer Institute, hired by then Deputy Director Anna Barker. And all I saw was this, right? My gene is right, your gene is right, my protein is right, your protein is right. And what's sort of our perspective into that, and Alan will be here, so he'll share this perspective. You know, there are ways as an engineer sort of quantify things. We can either qualitatively talk about water to sort of answer this question, or we can quantitatively measure. And at least in our field, we take measurements, we generated data, but then we started seeing some disagreements. Water boiling at 92 degrees, water boiling at 100 degrees. And for us, until we said, let's standardize, let's measure water, not salt water, not other, let's use some collective unit, we could not reveal that something that we could feel but not see, called pressure, affected our measurement. Right? That somebody was measuring this in Denver, somebody this at sea level. So we could have sat there and argued for decades who got it right. But instead, we have in our field something called a steam table, in which by using this, we can generate phase diagrams, and we can say something about the different states of water. Is it going to be a single state, ice, water, water vapor? Is it going to be something that exists together, like ice water, or if you're boiling some water for pasta. And how many of you guys have seen what a triple point looks like? Right. When I talked to my stem cell biologists back at the NCI days, this is something not sped up, it's at its triple state, at its triple point. And if I was a surgeon and I was taking out a piece of tissue here, I was liquid, I would get the right answer. I would get the answer that I thought was the right answer. But something as dynamic as the human body, you've got to tell me that it's got to be more complicated than that. It's very simple stuff. So what does that mean? I think if you're going to hear a little bit of this, I'm going to keep referring back to this. We built cancer scene phase. We're still chasing that phase diagram. And with that is about a decade's worth of doing this just for that first time point. Primary, untreated late-stage tumor. People that come up to me and tell me that they think that TCGA has temporal information. I'm like, no, just one time point. And when we started this in 2006, we decided to go after 3 pilots, brain, lung, and ovarian. And we did what we could to get as much orthogonal information, clinical information about the patient without necessarily revealing the patient. This was not the easiest thing to do. I think everybody in 2022 says TCGA doesn't have enough clinical data. You got to remember we started this in 2006. Where there was a little less, hey, how might we do this appropriately? And boy, did everybody have their favorite thermometer. Everybody showed up and said, it's got to be Illumina, it's got to be a nanopore, it's got to be an ion torrent. And what we didn't do was collect samples per technology. We said, let's make the quality of something that we would not change, and let the technology continue to emerge and continue to sort of utilize. When we first started TCGA, much of this was still done on microarrays. There was no next-gen sequencing yet. There was no RNA sequencing. So when people ask how come the brain cancer set doesn't have RNA sequencing, I'm like, well, we didn't invent time machines. We couldn't go back. We couldn't reanalyze this. So again, those that have not had a chance to use TCGA, what you see on the screen here, 1 through 23 of your chromosomes, each row is one patient. I purposely put this a little bit vague so you can see this are brain cancer, ovarian cancer. I'm personally very glad that we started with brain cancer, and we were able to find some unique metabolism-based markers that people were, when they only had 1 to 10 samples, they were like, I wasn't sure. But at 200 samples, you could be a little bit more sure. The reason I say I'm glad we started with GBM is we're still, as a field, figuring out ovarian cancer, which is such a copy number-driven disease, that if we had showed our board at the Cancer Institute, this worked, and showed this, they did it great, and it didn't work. And when we went to go present this, and we started realizing that we just didn't have enough people that knew how to analyze this data, you know, I was excited that we started a program as well to bring computational scientists, engineers, mathematicians into the field that we launched in October of 2009. We made a concerted investment, 12 centers around the U.S., Hopkins, Princeton, U.S.A, and Scripps, as well others, to say, let's bring engineers, computational scientists, mathematicians, let's start teaching them what does it mean to look at cancer data. And I would argue if we had not done this, we would still be stuck with a bunch of data that was not particularly easy to functionalize. The other thing that sort of happened when our board said, hey, no problem, let's continue this pilot and let's do 10 more cancers in 2000. For those who have not met Ann Barker, she's very, very aggressive. So during the time, some of the faculty members here might not want to remember this. There was something called the Stimulus Act. Basically, everybody submitted a shovel-ready project that got denied. We put in a signature request at the suggestion of the White House at that time to say what project could, in 2 years, if given some additional resources, could really ramp up. So instead of doing 2,000 cases, 10 cancers, and put in a request to say, let's do 25 cancer types and 10,000 patients, and then proceeded to retire on me as soon as the president said, this looks like a great idea, let's do it. So I will tell you, if you look at these 2 lines here, the red and the blue, how many we told the White House that we would get versus how many we actually got. I did not sleep very much 2009 to 2011, because every single sample that came through TCGA, I had the fun and joy of looking at the quality, clearing, signing, saying yes, no, let's move. So pretty passionate about that particular program. I'm mentioning that just to say my engineering training definitely helped me set up process design a little bit easier. When we said, you know, how high should the quality of this be? Let us not accept anything where we would open up the samples and it would literally be failed samples across some of the major cancer centers. I won't name which one, not JCCC. And how many of them failed pathology when we would send them around? How many of them, once we generated and tried to generate genomic sequencing information on it, finally came all the way out the end? We averaged about 58% going through the entire pipe. And we cared about that because we knew that this type of data would be used, hopefully, in the future. We really wanted to keep that part done well. This is, of course, even now almost a decade-old version of looking at TCGA. I just wanted to give you a sense of all the different tumor types that have now been completed. Again, red is more, blue is less. Every row is still one patient. This is something that I used to generate every year because people would come and tell me that TCGA is hard to use. So I'd let them know if a federal government worker, a chemical engineer could do this, you too could do this. And when I talked to Harold Varmus about this for the very first time, he was like, why did you show the data this way? I was like, chemical engineer, head on the top, colon on the bottom. That's it. There's no extra. And he was like, what's going on in chromosome 8? And I was like, I don't know that, Varmus. You're the Nobel Prize winner. You should, I should be asking you that, right? Being able to visualize and see again what an engineer's perspective might bring doesn't mean I have the right answer. It's just, how do you think about the same problems through different lenses? And then, of course, I'll fast forward a little bit here. Some of you may know that a lot of folks then took that, generated a 143 gene panel in Oncomine, and launched a trial called the NCI Match trial. Still probably the fastest accruing, fastest completing trial. I think the NCI infographics team was a little bit more optimistic that we would actually get about 60% of the patients matched, whether it was something a molecular similar to an agent that was already approved. Unfortunately, we had a match rate at that time about 18%. Hopefully not surprising, unfortunately, because the drug developers and the diagnostic developers in the room. And on top of that, because these patients did already fail standard of care, those that wanted to enroll into, you're giving me a breast cancer drug, even though I have gastric cancer, what's going on? I don't understand what that means. But the good thing is we prepared for a little bit of this. And we did that by taking not similar cancers from TCGA, but the exact same tumors, so breast, ovarian, and colon in 2011, set those aside and said, let's finish central dogma of biology. And again, chemical engineers, so DNA, RNA, protein. I think when I asked, how do we know for sure that RNA expression of an aberrant will actually manifest itself as an aberrant protein that could be drug targeted or detected? Everyone is like, oh yeah, it's going to happen all the time. This is going to be a little audience participation. How many of you guys think TCGA RNA sequencing, aberrant protein, predicted 90% of what showed up as an aberrant protein? 70? 50? You got a couple 50s? 30%? 30%. Less than 30% is the rest of the room. That's super depressing, right? Because how many people took RNA expression data and just assumed that particular protein would be the drug to detect? And it took a chemical engineer to sort of ask this question for the field. And we wanted to do this right because Varmus at that time was the NCI director. He said, there's no way. It would be like 90%. So we went out and talked to NIST, FDA, made sure that this was done well. The answer is 33%. Super depressing. Right. This data set, again, made publicly available. I'm still looking for scientists that look at this area that does not obey central dogma of biology, where you see more protein, but it doesn't show up in expression, or vice versa. And it was at this time, we also made sure to include medical images. For every one of these patients, their surgical imaging is also made publicly available. It's not just the molecular side. The challenge that I sort of reached at that point was, this is great. I have some clinical data, I have some genetics data, some proteomics data, some medical imaging data. But unfortunately, the human cognitive capacity can only take about 5 facts per decision. And that person can be the caregiver, the patient, the physician. When we talk about shared decision-making, you're not going to hand over 10,000 bits of data. Like, hey, you should take therapy A versus therapy B. How do we simplify it? How do we begin to distill down some wonderful, wonderful science? And the challenge of being able to distill that information is that you didn't have any of the clinical context. So thankfully, at that time, my wife, Connie here, and Chen Rong knows this as well, late Sam Gambier helped me out with this. And what it was taking advantage of an ecosystem called the Veterans Hospital. I was talking to Professor Tsai a little bit about this. How many of you guys know what the VA is? How many physicians had to train there, right? This is a national network and we don't take advantage of it. And because she was a hospital administrator that was responsible for accrediting all of the 164 hospitals and 3,000 outpatient clinics, she was like, great, bring your data scientists in here. They'll try to think about big data at scale. This network serves 9.2 million veterans across the United States. And this, in addition to that year, Dylan, of course, she is pregnant here. This was our first joint presentation at the White House, proposing a program to say, let's bring data scientists into the VA and see how they fare. And many of them did okay. The program is still ongoing. But bringing those data scientists into the ecosystem to say, well, it's not because this data is a missing value, it's because the patient, the physician was trying to take care of something else. That I thought was pretty interesting and valuable. And so again, one of the things that I really, really had a headache in 2006, we did TCGA, we did our proteomics program, imaging program, ASCO had its taper, Genie, Orion, MASH trial. Like, what was next? Everybody was sort of asking, what's after TCGA? How do you do the next time point? And I'm sitting there going, well, what are we going to do? There's just no way this is going to happen. This is what I mentioned. Doug Lowy came down to my office and said, we got a call from the White House. And you have been asked by name to go down there. And apparently, folks said that I did something. So it was a really nervous time for me because for those of you who have not had a chance to work with President Biden, he's very passionate about this topic. He would spend an entire day working on what he does as VP and then come and ask, Jerry, what do you mean by raw data? Like, what do you mean by variant calling of sequences? How come Orion, TCGA, and Genie don't interoperate? Like, those are just like 3 totally different data sets. One's a clinical grade sequencing, one's research sequencing. And he had so many questions for me. But the one that I couldn't answer for him, he said, well, Jerry, we just did this 1 million cohort in precision medicine initiative, now called the All of Us initiative. We did TCGA, 10,000 patients. Couldn't we do 1 million patients? 1 million cancer patients. I'm like, no, sir, you cannot. And the reason I had to say, no, sir, you cannot, is 85% of the cancer patients in the United States are first seen, first diagnosed, first treated in community cancer studies. It took us 8 years, $500 million to get 10,000 TCGA patients in our disconnected U.S. healthcare system. There is no way we are getting to 1 million, especially since at that time it was 1.6 million diagnosed new cases. We want that number to go down. It's unfortunately going up a little bit. But each one of these places where we lose an opportunity in the United States, at the cancer center, sometimes people don't go back to the same cancer center if they recur. Cancer patients have to bring a backpack of data to their data because they only have 7 minutes with that side. It's not, hours and hours when we go and grab every possible genomics and sequencing. So what did we do for him so that he of course was like, great, that's a total downer. Can we do something? And I said, great, how do we reach out to maybe the military and not just the VA? And bring some of the same things that we did in TCGA. And instead of asking the military to recreate all of it, we reached across and started the first longitudinal prospective trial that is still ongoing as sort of the co-PI on the program that's called the Apollo program. I will try my best to talk a little bit about that, but as soon as we finished Apollo, he said, that was great. now could you do this, worldwide? And I said, well, let's give it a try. And we reached out to Australia, who is building a large proteogenomics facility at that time. And I think for us, we were really grateful that Australia reached out and said, no problem, we'll give you our program. We'll generate data the same exact way. And then he sort of left this little bit in his talking point. You're going to repeat and see it around the world. I came back and it was on my desk and said, VP wants 9 more of these so that at United Nations General Assembly, he could announce 10. I didn't know 9 other countries that were doing proteogenomics at scale. But the one that I did know was we had some places in Taiwan and Korea and others. Super proud that Taiwan stepped up and served as one of those 10 nations that not only agreed to do the data generation the same exact way, but also contributed. So I'm hoping for those of you that have raised your hand and said that you're using TCGA data, you're also using the proteogenomics data. You aren't. We of course announced this simultaneously. VP Biden announced it at UN General Assembly that day. We announced it in Taiwan, Huppo. And it was very, very exciting to see a lot of the Taiwanese colleagues, not only show up and generate and donate their data. It was also the first time my father had a chance to see me present, so I'm almost closing the loop here. Some of you that may not know him, he did work for both the former vice president of Taiwan and the current president of Taiwan in his time at TECO. But again, if you guys have not used that data, I would definitely say take a peek because this year, the release of those, this is on a US.gov website. I never thought that I would see this. But those data sets, if you are not using them, donated high quality data from Taiwan, as well as other countries, I really hope you do. That is definitely one place I'm excited about. So I've already predicted this, so I can't skip through quickly, so some of you may get whiplash, because Dr. Posada is going to talk about liquid biopsies. I just want to let you know the VP did also ask, what about liquid biopsy? He asked me that in the year of Theranos. So if you want to talk about hard problems, that's a really tough problem. You got to get everybody to the table, and we did. And this particular network continues to today, done by Lauren Lyman, who sat right next to me in the White House. All of these companies that are generating liquid biopsy tests are all, including ones that already mentioned, are generating and making those data sets publicly available. So really finishing this part out, just want to let you know what's sort of ahead. How do you generate phase diagram? And the way to generate the phase diagram, to figure out what state your patient's in, well, you need information about the patient's state. Right? Where do they live? What other comorbidities might they have? Not just their cancer. And I'm really proud, at least in the 21st Century Cures Act, that was sort of the end of the moonshot. We really did get a chance to put something into law, which if you can like anything that's in law, this is my favorite phrase out of that law. How do we eventually get to a point where we can get single longitudinal format of patient data that is easy, secure, and updated automatically? We are not yet at that point, but at least this is something that is already in law that builds upon HIPAA, high-tech, and now Cures. So how do we get out of this era of turning people into barcodes and then turning hopefully some of them from barcodes back to human? I assume some of that will be addressed tomorrow at the AI discussions as well. And I'll just let you know, for me, the moment David sort of called and said, hey, I'm about to build this institute, you got a generous donation from Larry Ellison in 2018. Would you come and be its chief science officer? It was humbling for me because this is the facility that we now currently have up and running. We've been seeing patients for about 1.5 years now. We have both translational laboratories, a CLIA testing lab on site, as well as a pharmacy. We actually have a kitchen as well, because we do care about food, and I don't mean a regular kitchen. Chef Nobu actually designed this for David so that we at least could start teaching our patients about food and food science. Just to let you know, the Apollo program came with me to U.S.A. as well. So for some of you that are interested in this particular work, I would happy to talk with you a little bit more about it. The patients are consented across all 3 cohorts for life, both their clinical grade as well as their research grade tissue samples have been followed. The cohort is now 8,000 patients, and we're going to have our first publication shortly. Let me just get through this part, unfortunately. I think the last part, I want to make sure I don't miss. I was personally very humbled to be asked to rejoin the Biden-Harris transition team in October of 2020. And some of that is not only because I got a chance for my 2 little ones to sort of see what I used to do. Again, of course, to serve him and his vision, but the person that I had to get ready was the Secretary of the VA. And the reason that is important for me so that you all get a chance to see what we're doing. The policy, hopefully some of you have already seen, like putting high quality data to the ground is an executive order I had a chance to work on. It also requested and demanded, I'm getting played off like the Oscars. It demanded a chief science officer for every single agency so that we didn't worry too much about science eroding in different agencies. But on top of that, I think what's important is some of you that may have seen there is now a moonshot of 2022. I'm excited to see that they are doubling down on the investment in the Apollo program. So now every single VA hospital and every single military facility will be done. One that is connected to this is now how might we, if we have a continuum, think about military exposures. And this particular program, when it was put together, this is the Apollo retreat that just happened a few weeks ago, and we didn't get a chance to touch too much about that. But if you're interested in Apollo, some of the work that we're doing in the environmental exposures, we were sort of asking ourselves, what about the sort of pre-disease era? Could we go back and look at VA and DoD data, their serum, their blood, and say, could we predict the patients that unfortunately showed up in Apollo and got consented? And thankfully, this is also a topic that we brought together at the highest level, the Department of Defense, highest level of the Veterans Affairs, started brainstorming about this topic in May. And I can at least share with you what I was trying to rapidly get to. A couple of weeks ago, you may have seen a second program announced by President Biden. That one, unfortunate to also co-lead. We call it Prometheus. And what we're going to do now is take some of the Apollo patients that are in fact definitively diagnosed with cancer now and look backwards into their records, look backwards into their blood. And hopefully I'll have something good to say about Prometheus shortly. I'm getting the wave, so if you ever need to, some of the things that I didn't get a chance to touch upon, Anne and myself, we actually wrote a piece for AACR this June to talk a little bit about what we see are some of the challenges for the next decade. We were sort of pushed to say, was big data real or was it sort of paralysis? And with that, I hope I leave you with, again, keeping data fair, making data fair. How do we train the next generation? And I'll leave this up just in case you're interested and you're asking, what are we doing also to sort of train that next generation? We actually did start a class. Kate is now a second-year med student, and we bring together students across these sort of freshmen to junior seniors, and actually they teach each other how to use TCGA. And it's exciting to see now the second time that we ran this course. We have 14 different students, some are first-time coders, some have some coding experience, but they're all now learning how to use this data that we tried our best to sort of put together 16 years ago. And with that, I'll just sort of close and sort of thank all the folks at the Institute, my team, as well as mentors and others. And if you want to get a hold of me, please feel free to shoot me an email at any time. And sorry again for running a little bit over. But
well, thank you, Jerry.
Thanks for your attention.
Yeah. Yeah, for the sake of the time limit, and I will encourage the audience approach to Dr. Lee and to have a more interaction during the lunch. And so, Jerry, thank you so much.
Thank you.
And so we often joke, be careful when you take a picture. So I was looking into my picture album and try to find a picture I could connect Jerry to the next speaker. So in fact, this is the one. You could see Jerry is on the far up, and this is Ed, and that was me. And so we actually stand in front of Eisenhower building, and that was the time Jerry took us to have a tour of Vice President's office. And that's one of our White House Cancer Moonshot luncheon meeting. So next slide, please. And that naturally boiled down to my introduction to Dr. Posadas, my longtime partner. We work a lot on liquid biopsy in prostate cancer. Ed's clinical work, I think, is all about prostate cancer and the urologic cancer. And the other thing, as you could show over here in the picture, Ed and I, we actually formed a program we call the TOP program, Translational Oncology Program, and in which we train Taiwanese medical doctors. I'm very proud. This was in the Jerry's wedding ceremony. Ed and I, we both attend Jeff's wedding ceremony, and Jeff was the second top student we trained over the years. I'm very proud to announce now Jeff is an assistant professor in Memorial Sloan Kettering, working in the field of urologic pathology. And Ed, podium is yours, please.
Okay, thank you, everyone. I'm a little daunted by the previous speakers, and I am also part of the Hopkins Mafia, apparently. Worse than that, I'm actually part of the Hopkins Chemical Engineering Mafia. So unlike Dr. Wong and Dr. Lee, I did actually my undergraduate training in biomedical and chemical engineering. And so I'm an engineer converted to medicine. So maybe I'm the least intelligent person since the others here have gone to do such big things, but it's my pleasure to be here as a representative for our team to talk about how we have used liquid biopsies to really move closer to the idea of precision medicine in the disease of prostate cancer. Now, before I get going, I do want to actually pay credence to a dear friend and mentor, Professor Leland Chung. He is was a very proud graduate of National Taiwan University, which I also see has quite the mafia here as well. 11 years ago, Dr. Chung sat down with Shenrong and myself and said, you know, the two of you need to work together because cancer needs innovation, and the two of you have the capacity to put something together that can transform our field. And he was a man of great vision, of drive, and importantly, and resonating with the whole theme of this meeting is he really believed that getting people together and allowing science to form bridges could actually reach across the ocean. So he spent his career bringing together the U.S. and Asia, particularly Taiwan, to form those scientific bridges in the name of advancement for science. We lost Dr. Chung last year to cancer, which is a terrible thing, but we keep his spirit alive within our group. And I think if he were here, he would be delighted that we've held a conference like this. So the first thing I want to take the opportunity to talk about is why prostate cancer. I almost had to ask myself this question back in 1995 when I came into the field, because I always thought I wanted to study what was a quote unquote important cancer, something like lung or breast or colon cancer. And I actually ended up in the field of prostate cancer, not really paying attention to the fact that prostate cancer is the most common cancer that affects men in the US, and it's the second leading cause of cancer death. Now that creates an urgency here, but what's actually becoming the case is even in Asia, as societies become more and more westernized, this has become an increasingly important problem. We have already made a lot of mistakes or hit a lot of problems in the US, and our hope is that by sharing those foibles that we've gone through, we can actually avoid those problems and further spur advancement in the field in Asia to help those men who are going to be affected by this disease. Even in Taiwan, it is now the 5th leading cause of cancer incidence in Taiwanese men. and the 7th leading cause of cancer death. It's anticipated this as, again, time moves forward and society becomes more westernized, that these numbers will get increased and look more like the United States. Our understanding of prostate cancer has been relatively primitive. So I was paying attention to this because Jerry really has a wonderful perspective on where cancer research has been. Prostate cancer was a very small bar on his sequencing data sheet because it's really been an area where really genomics hasn't impacted the field. And this is because, you know, our mindset was like this. Howard Scher, who's a luminary in our field, coined this clinical stage model. It has 4 boxes that really define how we care for prostate cancer. And this was not really a great box. It was surgery, radiation, hormones, basically then death. But fortunately, due to a number of advancements in the field, these boxes have become increasingly more complicated as we understand how to use different tools. But you know, what's become frustrating is we're still basing things on a lot of clinical state about how much disease is present, whether they're castration sensitive or resistant. And only in recent times have we begun, for example, in the areas where I have red markings, have we begun to think about the molecular alterations that can provide insight into what's happening to provide those targets and those opportunities for making a difference based on the molecular nature of the disease. Jerry gave a nice statement. Why would you use a breast cancer therapy and gastric cancer? That's because at the genomic level, at the proteomic level, these are quite similar. We've needed to do this in prostate cancer and got stuck thinking only about the androgen receptor. And I think having tools that allow us to look at prostate cancer this way is going to be very important. In fact, I would tell you the science is already getting there. So despite the small bar that you see on Jerry's graphs, there have been a lot of studies looking at molecular classifiers in prostate cancer. Felix Feng, who's a good friend of mine, and Dan Spratt have done a lot of work looking at a breast cancer classifier called PAM50, reapplying it to prostate cancer, seeing that there are actually genomically defined subsets, so not just looking at the shape of the cells, but the actual genomic content of the nuclei, and seeing these will actually predict behavior, prognosticate behavior, and predict responses to different types of drugs. Similarly, Sun Young Yu, who's an informatician at Cedars-Sinai, developed another prostate cancer-based classification system that again breaks prostate cancer into 3 genomic subtypes. Now, we haven't used this lens yet well, and I will explain that it's been a bit of a problem because these classifiers predict survival, they predict response to AR therapy, androgen receptor targeted therapy, and even potentially other hormonal therapies. But you know, what's the issue? So number one, doctors, and I want to be gentle because I'm not the only MD in the room, but we get a little pig-headed. So we kind of know what works and we do what works, and we don't think about what the next generation of questions is. So a lot of doctors are not aware of these molecular classifiers, nor do they know how to use them in practice. Secondly, genomic testing is not so simple. That's one of the reasons I asked Dr. Zhu that question, because doctors are busy in their office. They don't want to take the time to fill out paperwork, especially if they don't understand the questions that they're looking for. And so bridging that gap, make testing easier and interpretable to the practicing physician is quite serious. Prostate cancer has a long natural history. So Jerry showed a couple examples like breast, ovary, which have, and even pancreas, that's relatively fast histories. Prostate cancer has a 10 to 15-year lifespan with a lot of morbidity along the way. And so the tissue that we have is often archival. Dr. Zhu touched on this. If your tissue is garbage, you cannot analyze it, garbage in, garbage out. Biopsies are painful. If you tell a prostate cancer patient, if you tell any cancer patient, well, to understand your disease better, I need a biopsy. The first time they'll say, well, okay, don't really want to do this, and but I'll do it if it helps you. Over the period of 10 years, if you ask them to go in every few months to do a biopsy, they're going to leave your office. So they're going to tell you that's ridiculous. And bone biopsies in particular, since prostate cancer, when it metastasizes, has a predilection to bone metastases, is an even greater barrier because 50% of bone biopsies do not yield useful information. And this is probably one of the more painful biopsies that we do. The cost of testing is not cheap. The first question I'm always asked is, will my insurance cover this? Very often there's some part of it that will be covered, but insurance will only cover those things that are known to make a difference. So when you're looking at more experimental ideas, patients can get lanced with the cost of testing, which makes it undesirable to participate, and then finally slow turnaround time. So you'll do a test for someone, say, we'll have the answer back in 2 to 4 weeks. Not very satisfying, especially when you need to make critical decisions. And so this really becomes a problem in applying these things, particularly to the field of prostate cancer. So, sorry for the graphic error in color, but so one thing that really kind of came to pass was understanding, is there a way that we can classify prostate cancer without needing tissue? And one particular piece of one particular fact of medicine that was important, that tumor cells in the circulation can be shed. So I want to be careful. There are very smart people in the audience that understand shear stresses within the bloodstream. The shear force at the tumor blood interface is quite severe and will cause the shedding of both tumor cells and even subcellular vesicles, as Dr. Zhu had mentioned earlier, and even cell-free DNA. Given the integrity of several of these things is quite weak. Circulating tumor cells are very, very fragile, and it's easy to break apart. So you'll see remnants of them in the circulation. But all these analytes contain information which might be usable in a setting as an alternative to tissue-based biopsy. And so this provides an important opportunity for advancement. So there developed an interest in prostate and in other cancers at looking at circulating tumor cells. In fact, since the 1970s, scientists had hypothesized that they're there as mediators of the metastatic process. These seeds need to get out from the primary tumor to the secondary site in order to develop the actual metastatic lesion. And the question had been, is there technology that can find these things and characterize them? It really wasn't until, within the past 10, 15 years, that technologies became available and even went to market, such as what's called the cell search assay, which identified CT, because we began saying, well, we can see conversion of good and bad patients, differences between those with lots of CTCs and those with very few CTCs. But as you might guess, this has also kind of petered out because that really wasn't enough to gain insight. We're still so far away from all the fantastic molecules you've heard about. We're just counting cells at the end of the day. And this is a bit of a problem for advancing the field. It wasn't a high risk, high reward. So Dr. Zhu had shown this figure, but really 11 years ago, when I was introduced to Dr. Sen and his suite of tools, then it was only one tool with a second developing. Now we have a suite of over 5 different types of technologies based on his discovery of nano Velcro that have allowed us to dissect apart what's happening within the bloodstream. And due to the very tight interface that he created between us, literally meeting with me weekly in order to understand the problems I was facing in the clinic, we began thinking about things that we could do to change and address things. I mean, truly an engineer spirit. He asked me, what's the problem? And we came up with solutions together by addressing them like a good engineer would. That's what I learned as an undergraduate engineer. So one of the first problems that I faced was I deal with a lot of advanced prostate cancer patients. I wanted to know, is there a way we can identify these patients earlier? Because often by the time they're coming to me after 4 or 5, 6 lines of therapy, it's too late. And one of the signs that it's too late is the cancer is no longer osteotropic. In other words, it has picked up a different biology that will make it spread to other parts of the body, such as the liver. If a physician sees prostate cancer that's spread to the liver, they basically tell the guy to go home and die. It is really an end stage sign of the aggressiveness of the prostate cancer. And imaging, which is how we pick it up, there is no blood test for this phenomenon, is often too late. So it's spread like this, and you cannot resect the liver, and so often the patient will die of liver failure. So I asked Dr. Sen, is there a way we can do this? At that time, we were using the first generation chip, which allowed us to see with good definition what these CTCs looked like. Dr. Chen, whom we mentioned before, one of our early graduates, used this and began telling me one day, and Dr. Sen, I can actually tell you who's going to get liver mets. I said, Jeff, how can you do that? Because the cells change shape. So we we had compiled a database, a biorepository of specimens for men with different states of prostate cancer, visceral, non-visceral, and non-metastatic. And Jeff actually separated them out very cleanly by only looking at the shape of the blood cells. This was fantastic. We then later showed, with the help of Alex Jan and Terry Tang, two of our other graduates, Alex now at Mass Gen Hospital, and Terry Tang is a urologist over at Carbon Sian Hospital in Taiwan. We began looking at the survival of these patients and the response to therapies. And lo and behold, by looking at the blood, we could predict this without the use of additional markers. This got our scientists very excited. So this is the feedback Jerry was talking about. Because we could actually see these things happening, we began to work with Michael Freeman and his group, who were looking at nuclear shape instability, a driver of metastatic behavior and resistance in cancer, and began to see the change that he was describing in nuclear shape instability related to the loss of emrin, which is a protein that regulates nuclear shape. We could actually see that loss of emrin stability in the circulating tumor cells. And in a sense, we began to tell scientists, we can see nuclear shape instability and genomic instability in the cell, in the cancer as it develops. And so the scientists have now begun to use this technology to try to move ideas forward. A second question that came forward is, since the scientists are excited about this now, can we see other molecular prognostic information in the bloodstream? So Dr. Yu, a bioinformatician at Cedars, became a critical partner to our relationship. So we have now a triad of engineering, informatics, and medicine working together to apply molecular signatures using advanced bioinformatic technologies to refine gene signatures for the purpose of liquid biopsy, so that we can now create CTC-specific gene panels that work within the background that is loaded with white blood cells to identify molecular signals that parallel what happens with a tissue biopsy. In essence, being able to do the same thing that's being done in advanced tissue studies without the need of a solid tumor biopsy. And so we are now trying to advance this work into different clinical trials in conjunction with NCI-funded cooperative groups. So Dr. Sen and I are working with NRG in two important clinical trials to do this particular work. Even to the point where now an NCI-funded collaborative project operation is using our technologies as part of a liver metastasis PO1. Dr. Yang, who you'll meet later, is actually part of our group dealing with liver metastases in this complex environment here. Finally, as a quick additional note, since I need to get out of the way for lunch, we've been interested also not just in CTCs, but Dr. Zhu had mentioned the importance of extracellular vesicles as another byproduct that's shed into the circulation. The work that Dr. Sen and Dr. Zhu have done has been translated also into the area of prostate cancer to isolate these EVs from patients with prostate cancer in order to look at prostate cancer specific genes. This provided us a very interesting opportunity to actually address the problem of failure of salvage radiotherapy. Patients who had prostate cancer that relapsed after, with local recurrence, the standard of care is to consider them, they're either controlled or cured, or if they have disease outside the prostate, where salvage radiation to the prostate gland will not help, they will actually fail. And they'll have gone through salvage radiation only to be left with the problem of dealing with metastatic disease. And so we have been asking the question of whether we can do this with a liquid-based biopsy. Jasmine Wang, who is now our student in the laboratory, has identified a blood test that we think we can use to separate out this process. And so we're now incorporating this also into additional clinical trials. And these are examples of patients to whom we've applied this trial to actually look not just at a single blood test, but the dynamics of their disease over time by doing the same blood test over and over, which I could never do with a liquid biopsy, and so or with a solid tumor biopsy. And so I'm very excited about these applications. So in closing, this is where I see prostate cancer going, where instead of guessing, we will now use molecular guidance at various steps to identify and select optimal therapies based on a liquid biopsy, which we're already doing in the series of forthcoming studies. So these are some brief conclusions about the importance of prostate cancer and the opportunities I see with liquid biopsy. I'm excited to be part of this group. And again, this is not my work, but the group of all, but I am only the representative of our group, and have to thank greatly Dr. Sen and Dr. Zhu, who have really led the engineering efforts, Dr. Sun-Young Yu, who has put the informatics, that has put context into all of this, and the large number of folks who have been supporting our work along the way. So I will stop there and answer any questions I can before you have lunch.
So we actually have one more speaker. So for the sake of time, let's thank Dr. Posadas and Ed. Thank you so much for bringing up, mentioning both of us are a product of Dr. Li Lianqiu. Yeah. So while preparing the transition of two speakers, I'm here to introduce Dr. James Wu. So Dr. James Wu is a surgeon in our endocrine surgery program, very young, spectacular training record, and all his work is centered around precision medicine in thyroid cancer. So he will share with us his research progress. Podium is yours, James.
Hi, everyone. Man, so I'm really between you guys and lunch. Okay. Well, thanks for the opportunity to speak to you all today. And today I want to talk to you guys about the other forgotten cancer, which is thyroid cancer. So I'm an endocrine surgeon at UCLA, and this is what I deal with most of the time. All right, so the thyroid gland is the gland that sits right in the front of your neck, right above your collarbones. And this organ really only does one thing. It makes thyroid hormone. Thyroid hormone goes all over your body, has effect on all of your organs, controls your metabolism, your weight. Its little other hobby, though, is to make nodules. And the thyroid gland can make single nodules, as you can see in the bottom left, or it can make multiple nodules. Oftentimes, these nodules don't actually create any additional hormone. They're just little growths. And so thyroid nodules themselves are really common, but they're usually what drives people to come and see me is because when you see a nodule, then we start to worry about thyroid cancer, because that's the first sign of a cancer could be developing. So, I don't know how many people are in this room. If I just say 100, just to be, hip for my own convenience, if I went around and felt everybody's neck right now, I'd probably find 4 thyroid nodules somewhere in this audience. And your doctors can probably find one too. Now, the other problem is that our technology to look for these nodules has gone beyond just feeling. We can do it with neck ultrasound. Neck ultrasound is very ubiquitous. It's everywhere. I have one that fits in my pocket. It's like in my car, if anybody needs an ultrasound after this. But if I ultrasounded everybody in this room, I'd find thyroid nodules in over half of you. And this is the problem, is that this in the past would make everybody worry. They would say, oh, a thyroid nodule, what does this mean? I gotta go see my doctor, I need a biopsy. But the truth is that the number of nodules that are actually malignant or cancerous is very, very low. And I'll tell you how many in just a second. So because of this phenomenon that we have ultrasounds, everybody has nodules. And because we all want to use our technology and do ultrasounds, the incidence of thyroid cancer has been rising dramatically. And in the past 20, 30 years, it's gone up 200 or 300%, depending on who you ask. Now, people have theorized everything. They think maybe it's cell phones. There's papers that said after 9/11, there's first responders had more thyroid cancer. Maybe it's other radiation sources. But the truth is that it's correlates with how much you use the ultrasound. So in South Korea, Samsung made a bunch of ultrasounds. Everybody said, why don't you come by, stop by, get an ultrasound, just to make sure you're healthy. And they started finding all these thyroid nodules. And within all these thyroid nodules, then they started doing a bunch of biopsies, and the incidence of thyroid cancer in South Korea skyrocketed. And you can see in other developed countries like the United States, it also has gone up significantly, but not to the same extent, because we learned from that experience and didn't institute a screening protocol to say everybody get an ultrasound. So you can see here that these incidences at this peak is 120 people out of every 100,000. That's 1 in every 1,000 people. Here it's about 30. Now, the other reason why we know that this is just finding thyroid cancers by accident and not we developing serious thyroid cancers is because this whole time that the amount of death from thyroid cancer completely unchanged. So you have 3 times the amount of thyroid cancer, but nobody's dying from it. And either that's because, you know, people like me are just really good at our jobs, and we're just cutting out all the cancers that are killing people and keeping it exactly even, or we're finding things that didn't matter. We're doing a lot of biopsies, a lot of surgeries that we didn't need to do. And again, this here is 0.5 out of 100,000. So you find 120 in South Korea, only a half of one would be lethal. And more evidence. So they did an autopsy study over 6 decades. So these are people who did not know they had thyroid disease when they died. You get hit by a car, you have a heart attack, something else happens to you. If we cut your thyroid open and look through, we'll find thyroid cancer in 11%. So 100 people in this room, you know, once, you know, 100, 200 years from now, when we're all dead, 11 of us will have thyroid cancer, but you won't never know it before you die, unless you go looking for it. Okay, so that's the challenge that I want to paint, is that U.S. adult population, 209 million people. That means that 100 million people have a thyroid nodule. And at the time we all die, 23 million of us will have a thyroid cancer. But if we look at the number of deaths that are attributable to thyroid cancer in the United States over the past decade, 2,300. Very, very small. So we're not dying from this, but we can find it. But because it's called a cancer, people get really scared. This is just to hammer home the same point, that they looked at patients who are older, over 65, and they said, oh, I'm sorry, you have a thyroid cancer. But very few people die from it. And honestly, the chance of dying from something else only goes up farther and farther, where this stays relatively stable. So the problem with thyroid cancer, unlike other cancers, is that it's not that you're going to die, but it's dealing with patients' uncertainty. And patients, when it's you who has a thyroid nodule, and you don't know for sure you're going to be the one who survives, and you think, I'm going to be the one person out of 100,000 that's going to die from my thyroid cancer. They want to do everything that they can. They're going to have a lot of anxiety. They're going to start thinking about their future, their children, their finances, and they really want to have no regrets, is what people say. And so sometimes people will come to us and out of pocket pay tens of thousands of dollars for tests, surgery, things that probably wouldn't change the trajectory of their life. So to the physician, the problem is not the uncertainty, it's the overtreatment. Because the more ultrasounds we do, we're of course going to do biopsies if you come in. I tell you that I'm not. And then we do surgeries just to be safe when that biopsy is unclear. And you know, I'm a surgeon, I get paid to do surgery. We'll do more surgery and we'll take out more than we need to. And even if we walk beyond surgery, the next step in therapy is radioactive iodine, and we do that more than we need to. And there's many consequences to these surgeries, radioactive iodine. We talk about those side effects all the time, but maybe the one we don't talk about very much is the financial burden it has on patients in America. That when people get diagnosed with a thyroid cancer, 18% will borrow money to treat it or go into debt, and 1% will declare bankruptcy. And when you consider that that's like 11% of Americans who could find their thyroid cancer, it's a big number. All right, so just for context, imagine a different disease process, one that affects 39 million of the 209 million adults in America. And just in 2019 alone, 300,000 people died from it, and we don't treat it the same way. This is just diabetes. You know, we say, oh, you've been diagnosed with diabetes. Nobody gets scared. Nobody freaks out. It's a chronic disease. So we need to take back thyroid cancer and think about it and talk about it in much the same way, and probably even less so. Okay, so having said all that, what are the tools available to me as an endocrine surgeon to try to figure out whether thyroid nodule is meaningful and whether it needs treatment? So the first step is neck ultrasound. Again, I kind of painted it as the bad guy, but it's also the good guy, because you can see that with these technologies that fit in your pocket, you have full view of somebody's thyroid. There's no mystery there. And we've been able to create this scoring system by saying what we see on the ultrasound in a uniform way to communicate with each other what are the risks of a thyroid cancer and whether biopsy is necessary. Because if the ultrasound looks benign, we stop there, you're all done. Here's the problem, is that this system is the American College of Radiology. It assigns these points to give you TIRADS 1, 2, 3, 4, 5. 5 is, oh my gosh, we set the highest level of alert. 4 is maybe, 3 is maybe. And you can see that the risk of malignancy, even when you see all the risk factors, is only hovering about 30-some%. And if you're in these lower categories, TIRADS 3, TIRADS 4, when we're recommending biopsy, what is the actual risk of cancer? It's under 10%. So the ultrasound is not, it's a really good way to look at the nodules, but it's not a good way to tell if people have cancer. But when we communicate to our patients, oh, I think you need a biopsy, they're thinking, oh, I have cancer already. This is just to say that that's not the only scoring system. There's a bunch, but TIRADS is already the best. There's American Thyroid Association, a European TIRADS, Korean TIRADS, positive predictive value, 12%. If the radiology says you need a biopsy, 88 patients are gonna have a normal thyroid nodule and only 12 are gonna have cancer. The others are worse. And even when we go by these rules, the false negative rate, the chance that you actually still do have a cancer in there, even if it's wrong, and says you don't need a biopsy, still, 2 to 4%. Okay, so that was the first level of filtering we can do. Get an ultrasound, check the score. If it says it's risky, positive predictive value is low, we do a needle biopsy, figure out what's inside. We can look at the cells. People like Dr. Zhu can tell us which ones are risky and which ones are not. I don't know what these mean, but they will communicate it back to me in this category. So they'll give it a Bethesda score between 1 and 6. 6 is this is cancer, 2 is benign, 1 is not enough. And then 2, 3, and 4 is another gray area. The pathologist says these aren't normal. It's not cancer. I'm not sure what it is. And now the risk of malignancy is 10 to 30%, depending on the institution. It actually depends on how good your pathologist is. Because if you're inexperienced, you don't read these right. And of all the biopsies do, very few are clear. Very few give you a clear answer, this is cancer. Okay, so in the past, if you had a Bethesda 3 or 4, which is 30% of all biopsies, we said, well, you have this chance of cancer, better not take any chances. Let's just take everybody to surgery. So out of every 10 people I would do an operation on, only 2 would actually have cancer. And now you have 8 people who went unnecessary surgery. That's the past. Now we have molecular profiling. The biggest commercial entities are Afirma and ThyroSeq. And Afirma looks at RNA, ThyroSeq looks at DNA. And funny enough, they're kind of converging and the performance is the same. So now what we do is if it's Bethesda 3 or 4, at UCLA, we automatically will send it for one of these 2 tests. Now, here's the problem is the test is suspicious. If they see signs in molecular profile that it could be cancer, the positive predictive value is still only 60%. You just spent $5,000 on this molecular test and 4 people still have benign disease. And if the test is benign, then there's still a small chance that you have cancer and you still have to watch it. So they're not perfect. And the big problem is that it's useless without patient buy-in. Some patients, you get this test and they test benign. They're like, I don't care. I want my thyroid nodule out anyway. Well, why do we just spend this money? Some people get a suspicious test and they said only 60% chance of cancer. Maybe I'll just watch it. We just wasted money again. Okay, so that's the real challenge, right? Is that all these millions of thyroid nodules out in population, a lot of them can be seen on ultrasound. People just go to the doctor and ask for one. People just say, my neck hurts, my neck feels sore. they get an ultrasound. A lot of those patients will get a fine needle aspiration. Only a small amount will go for molecular testing because it's indeterminate. Then we do a surgery. And at the end of all that, in California in 2021, we're finding 5,000 thyroid cancers. Think about how much work went into finding those 5,000. And after all those surgeries, like they said, our malignant yield at UCLA is about 55%. So that means that there's all this surgery happening at UCLA, or this is in California, but if we use UCLA percentages, that didn't need to be done. And think about the costs. FNA, $500, molecular test, $5,000, surgery, a conservative $5,000, probably way more than that. Annual cost in California alone, $31 to $52 million. That's the impact of things we're doing because that's all we have. I'll try to speed through some of these. For surgery, there's a couple options of what to do. You can do a thyroid lobectomy, which is just taking out the half that has the cancer. There's risk to that. You can injure the nerve to the vocal cord. They may need thyroid hormone because half may not be enough. And they may need a second surgery to take out the other half. You can do a total thyroidectomy. Gives you 2 times the rate of nerve injury. Why? Because you have to do the left and right side. So naturally, twice the risk. 100% of those patients need thyroid hormone. And there's a percentage chance, 1 to 3%, that you damage the parathyroid glands and give them low calcium for life. Both cut out the tumor. So why do people ever ask for a total thyroidectomy? Well, it's because we want to give, so they do thyroglobulin monitoring, and one is radioactive iodine. So my time is up, so I'll go a little faster. Thyroglobulin helps you monitor for thyroid cancer coming back. It's a protein in the blood. It doesn't work if you have some thyroid left. And it doesn't work if you have thyroglobulin antibodies. So what happens to people if they have thyroglobulin antibodies and you can't do this test, the thyroglobulin test? Nothing. No difference in survival. So that test, it's a good test. It tells you there's thyroid protein, but doesn't change their outcome because nobody dies from thyroid cancer. And if there's something to operate on and to act on, you'll find it with ultrasound. So what does this tell us? It tells us a lot of people choose to have a total thyroidectomy to monitor this protein because they want reassurance. It's about patient anxiety and reassurance. It's not about cancer outcomes. And I don't know if that's correct. Another reason is to give radioactive iodine, but here's a table that says like how risky is your thyroid cancer. Only high risk benefit from radioactive iodine. That's when you have lymph nodes spread coming out the thyroid. Most patients live down here where there's just a unifocal small thyroid cancer under a centimeter, 1 to 2% risk of coming back, or a thyroid cancer less than 4 centimeters, 1 to 2% of coming back. If you look at this chart published recently about how often we use radioactive iodine for things that are in that bottom category, 1 to 2% chance of cancer recurrence, not even death, up to 25% at some centers. So we just do too much because patients are scared. We want to do a lot, and we take advantage of patients and spend money that we don't need to. And why? Because we don't have an ability to differentiate thyroid nodules from cancer easily. That's reliable. We don't have a way of telling which thyroid cancers are high risk and of which there are very few from the very many that are low risk. And we don't have a way to provide reassurance to thyroid cancer patients without the thyroglobulin, without doing a total thyroidectomy. So I'll just skip through these because you've seen variations of this. EVs are great. Little particles come off the thyroid cancer cells, the little pieces of the cells, and they have the DNA, RNA from inside the thyroid cancer cell. You're going to hear about that later. The results are great. And I'd love to do it with thyroid cancer because if we look at those 3 unmet needs, this would be a non-invasive blood draw to help tell nodules apart from cancer. It'd be a way for us to test DNA, RNA non-invasively, determine which cancers may be high risk or low risk. And then even with some thyroid tissue that's normal present and with thyroglobulin antibodies present, you could use it as a test to make sure there's no more disease left. Okay, I'm done. We can go to lunch.
Thank you, James. So we're going to close the morning second session in precision medicine. And I'm going to invite James and Ed to the podium. Then Dr. Chu is going to present our appreciation certificate plus our souvenir.
coming. So about 2 years ago, we started to look into the therapy for COVID. At the time, you know, as a scientist, the first thing I want to look at is that what's the mechanism of action? What is the drug target we should do? So, you know, this is old style, you know, drug discovery, right? You find a critical drug target and you say, okay, this is the right target, and you find the right molecule and you test it out. So we do confirm that 3CL protease, the main protease, is very reserved, even from the first coronavirus OC43 all the way down to SARS-1 and SARS-2. So immediately I say, this is our target. So we take our limited chemical library to test against 3CL protease. And lo and behold, we got a pretty good candidates. And we also find this candidate not only can inhibit the 3CL protease, also inhibit another protease. The protease of TMPRSS2 is situated on the host cell membrane. Somehow, this viral gene got into the human's genome and expressed a protease paradoxically welcome the virus to enter the cells. And we also find our candidate molecules can be inflammatory cytokine reductions. So mechanism of action, very important. If you want to find, I call it pan-corona, PAN, right, everything. Corona antiviral drugs, and that should be the main target, should be the main protease, 3CL protease. And we tested against OC43, against other coronavirus, and confirmed that it has strong potency. And more importantly, because of TMPRSS2, this kind of, you know, multiple target things, we realize that can be also, our drug can be also a drug for influenza. I'll explain that in a minute. Okay, sorry. So this is our phase 2 study, and of the phase 2 study on the COVID trial with our drug. I named it Pentalandia. Penta is like, you know, pen, right? Everything. Parlandia. Corona is Galen. In Latin, is Galen is crown. Via is killing the virus, so killing crown virus. Okay, Pentalandia. And Pentalandia in phase 2, you know, the clinical outcome shows clearly differentiated from placebo. We're taking mild disease patient, which means can be anyone, and we show its early differentiation and the effect lasting pretty long, even down to 56 days. But of course, in the phase 2 study, we can only ask limited questions, and the obvious burning question is how long this can be differentiated to persistent into the long term, because the concern of the long COVID now is on top on the agenda. And we also got a bonus. What I meant by that, we also see inflammatory markers is much reduced by Pentalandia treatment. And this is a marker of C-reactive protein. It's very common inflammatory markers. And more specifically is interleukin-6, which has been highlighted again and again in the study of coronavirus infections. Clear differentiations of the reduction of interleukin-6. So, you know, I just take this opportunity to show you that it's in clinical study, it's very, you can only ask limited question, you get limited answer. So, but we got a bonus here, so I immediately say, this is the drug, we should go further. All right, and we also want to take advantage of our, you know, I'm a biologic researcher, but what I learned in the past 10 years as CEO in the biotech company is to learn some chemistry. And some of the chemistry I learned is that the same drug, we can formulate as oral medicine, also can formulate as an inhaler. And the inhaler nowadays has very advanced technology. You go with the app and, you know, you can measure how much drug get in, you know, the patient take the drug, that kind of things. All right, let's talk about the status of the coronavirus treatments. All right, we know there's one oral drug which kind of stand as a standard of care around nowadays. But that drug called Paxlovid. Paxlovid is developed for serious infections. And we developed drug is for the mild infections, mild disease. So for Paxlovid, it has an issue is that, by the way, it's also a repurposed drug. The issue is that it cannot be used to everyone. You know, you only reserve that the most severe populations. And the reason we need a more clonal drug, anti-clonal drug is that, you know, like penicillin, right? The first one. Now we have like 30 penicillins type of antibiotics. And so that's very obvious because it's once the infection start to come up in human history, it's not going to go away. The only thing going away so far is which one? Smallpox. The rest is still there. Polio is still there. Everything is still there, okay? It's impossible to eradicate it. And the vaccine. I think we got a problem, right? Breakthrough infections. And we cannot catch up with the variants. We can kind of getting better protection, but we cannot, you know, the influenza vaccinations, the efficacy is around anywhere between 30 to 50%. And in the beginning, when the Pfizer BNT vaccine first came out, it's 95%, right? But, you know, in times, now it's dropping to the rate of about 30%, which is correct, following the path of the influenza vaccinations. And of course, RNA that's part of the problem. It's a breakthrough infection. We can never catch up with the new variants. I talk about COVID-19 cannot be eradicated. And, you know, Spanish flu is still here after 100 years. So in 100 years, we still have COVID infections, believe it or not. And there's this missing treatment of mild and early disease. Symptom elevations, like Tamiflu for influenza, and the household transmissions. One family member got it, you know, the rest got it, you know, that kind of thing. So we want something to not just facial mask. We also want to have some preventive measures. Paxlovid, I know you understand that it's like the long list of drug-drug interaction. It's because of molecule. Molecule is one of the two active ingredient is a small peptide. So it's kind of can binding more non-specifically. So too many drug-drug interaction. And in real world data in Israel, it's a bit troubling. A few weeks ago, the data came out. Paxlovid is good for people above 65 to reduce the hospital visit, to reduce the serious progression, ICU, that kind of thing. But I don't understand why that in real world data is not better than placebo for the age group between 40 and 64. So which is telling us the therapy we have is still very limited. Not just limited to one, but this one has some limitation. And of course, it's very expensive, right? Everything from a big pharmaceuticals is like that. All right, pandemic. How much time I have? I think I have to almost up. Okay, I'll fly through it. Pandemic, our curve is not flattened yet. Pandemic is serious. This is from JAMA, and we are facing two viruses at the same time, okay? And I just read the Washington Post like while I was sitting for my lunch. We have another year reduction of life expectancy for year 2021, okay? All right, this is, I tell you about the mechanism, TMP-ISS2, protease, and anti-inflammatory. That's why we can do both virus. And this is our influenza challenging study. We show the survival improved in the challenging study. And okay, this is our combination study. Okay, I wouldn't go into the detail, but pretty much our drug in combination with Tamiflu pretty much reduce how much drug you need to use, and there's synergistic effect. Okay, which is very important when we develop combinations. All right, probably we'll have two more to go. This is a bit. Apparently, I didn't get my military training right. Okay, so CNRS has two pillars. Number one, new chemical entity, CNS viral drug. Number two, I'm building that. What NCE can now offer, we head into proprietary Chinese medicine because we do have the science know-how to identify them. And we do have the first step of success to apply for health supplement in Singapore market. This Singapore, very nice regulatory agency in Singapore called Health Science Authority. You see how they see the therapeutics. They're not only looking at device, therapeutic product like drug or gene therapy, they also regulated traditional medicine, which is Malaya, India. They also looking at Chinese medicine. They're also looking at health supplement. And when we submitted one product for review, I was very shocked. They're asking for 6 months toxicity study, like a new drug. And that when we apply for one of the health supplements. So briefly, and I hope I didn't ruin your lunch. I just want to tell you a story that how we deal with COVID and what a science can do for all of us. Thank you very much.
Thank you, Emil. As always, Emil always give very exciting talk. And so for the sake of time, and please approach to Dr. Tsai if you have a question. And we are going to have a quick break for 10 minutes. Then we'll start from our session 3 here by Francis Su. Then the first speaker of session 3 is Dr. Linda Liao. Okay, so we'll start roughly in about 10 minutes. Thank you. Oh, yeah, a quick announcement. And while we all enjoy this very delicious lunch box, so I want to acknowledge AHMC's chairman, Dr. Johnson Wu, providing spot. Thank you so much. Okay, we will be back in 10 minutes.
Ladies and gentlemen, good afternoon. My name is Francie Xu. I'm Assistant Professor of Neurology at UCLA. I'm so honored to be here and so excited to see all of your lovely faces. Most importantly, I'm thrilled to be introducing our first keynote speaker in the afternoon. Professor and Chair of Neurosurgery and Director of the Tumor Board Program at UCLA. So many patients actually travel across the globe to see her. And after being told to have only a few months left, she gave miracles. In fact, one of the many patients after undergoing her surgery was able to pass the bar exam and become a lawyer and serve in Hawaii. I want to serve in Hawaii too. Ladies and gentlemen, Dr. Linda Liao, Dr. Liao Mingfei.
Hello. Well, thank you for having me here today, and thank you, Ms. Yu and Dr. Chu for organizing this event. The talks have actually been wonderful. It's been a pleasure to be here. I actually was supposed to be at the National Academy of Medicine this meeting this week, but I had other conflicts in my schedule, so I wound up actually having to stay here in LA. So I actually am quite excited that I would actually be able to come here today. But so what I'm going to talk about is brain cancer. As was said, that I'm a neurosurgeon. I'm the chair of neurosurgery at UCLA, but I've focused my practice and my research over the years specifically on brain tumors. And these are my disclosures. So some quick facts about brain tumors. They're, and like thyroid nodules, where there's like millions and millions of people who have them. This is actually relatively rare. We don't have millions and millions of people diagnosed with malignant brain tumors. The annual incidence in the U.S. is only about 25,000 per year. However, the difference is there are 18,200 deaths from brain tumors. So if you're diagnosed with one of these, it's inevitably almost fatal. So glioblastomas are the most common primary malignant brain tumor, and the median survival is still only 8 months. With treatment and aggressive therapy, sometimes that can be pushed out to 15 months. But even with all the treatments we have, we really don't have any curative treatment for this disease. So although there's been progress made on other cancers over the years, brain cancer is probably one of the, one or two cancers that we really don't have any definitive treatment for. I think it's, brain cancer and pancreatic cancer are the two that, if you have that diagnosis, it's inevitably, dire. And the 5-year survival rate right now is still less than 5%. Some people do survive up to 5 years, but that's, quite a rarity. So, so despite surgery, brain cancer inevitably recurs. So, regardless of how good I do in terms of taking out these tumors in the OR, with radiation and chemo, this tumor recurs. And this was the problem that I saw, gosh, almost 30 years ago when I started in this field. And this is kind of, what we still see today. So this is, a typical patient with a near Wernicke's area, which is, one of the speech areas in the brain. And, nowadays we do a good job. I could take out this tumor. It's what we call a gross total resection, and then, the tumor is no longer there. However, because we know the prognosis of this cancer is quite poor, these patients go on to radiation, and then chemotherapy. Currently, the standard of care is temozolomide, TMZ. And that was the standard of care 20 years ago, and that's still the standard of care today. So unfortunately, not a whole lot has progressed in this particular disease. But one thing that always perplexed me was that, well, why do these tumors come back? You know, why? We take out the tumor, we radiate the margins, and then give chemotherapy. The reason they come back is because there are these tumor cells that are still there in the brain. They're microscopic. I can't get to them by surgery. I can't get to them by radiation. And they still reside in the brain. And at any time, they will grow and come back and recur. So what can actually target these cells that you can't see? You can't really get to them because of the blood-brain barrier and other obstacles. You know, what kind of modality is best suited for this type of cancer? And you know, I actually, 25 years ago, I thought immunotherapy was really kind of the way to go in tackling this disease. And I could tell you when I first started in the field, everyone thought, oh, no, immunotherapy for cancer is not, you know, really, that going to be that prevalent. And we were studying EGFR inhibitors and targeted inhibitors and everything else. Now, you know, in the last 5 years or so, there have been lots of immunotherapeutic drugs that have been FDA approved for all sorts of other cancers. So I think immunotherapy is you know, kind of a future direction for cancer. But we still don't have an immunotherapeutic that's FDA approved for glioblastoma yet. But I do think, you know, one way to target these cells is, as I said, immunotherapy. So, so I set out many years ago to actually develop a vaccine because you know, after surgery, the tumor is gone. So you're really not treating the tumor, but what you're actually treating is that prevention of that tumor coming back. So what is good at preventing things from coming back? Vaccinating people against a potential you know, disease like recurrence. So so you know, when I was starting out as a junior faculty member at UCLA, I met this gentleman, Dr. Ralph Steinman, and this was, you know, I actually met him about 10 years before he won his Nobel Prize. But what he won his Nobel Prize for was the discovery of this type of cell. And I was actually quite fascinated when he came to talk about this cell, you know, because this is it's a cell called the dendritic cell, and it's an immune cell in the body. It's, you know, a natural cell that occurs in the body, and it's one of the, you know, it's what we call actually the master regulators of immunity. It does all sorts of things, you know. It actually activates T cells, it activates CD8, CD4 T cells, and it has all these different receptors that interact with the tumor microenvironment. It, you know, it sounded like it was like insulin knife of the immune system. It just did everything. So it was fascinating. And in the late 90s, that's actually, you know, is actually, even though he discovered this cell in the 70s, in the late 90s is when people discovered how to actually grow these cells outside of the body in large quantities, so that they could actually be used as a therapeutic. So the concept of, you know, this type of cell, dendritic cells, they're professional antigen presenting cells. Like I said, they're normal cells. They actually live in the bone marrow, and then they are recruited to places in the periphery to capture antigen. You know, this is their normal function. So in the skin or the gut or wherever, if you have I guess the battery went out. And can people hear me? Okay, great. So so the, you know, this cell travels. Oh, now that went out. Okay. So this cell, you know, travels to the periphery, captures the antigen, and then it migrates, you know, either via the lymphatics or the bloodstream to secondary lymphoid organs, and that's where it activates the T cells. So that's what these cells do. Normally, they're, you know, only about 2 or 4% of the immune system. But like I said, there is a way to cultivate and grow these cells outside of the body.
So, and I'll get to a little bit more about that cell later. So in tackling glioblastoma, one thing that we've learned over the years, and then, is that glioblastoma is multiforming. GBM actually stands for glioblastoma multiform. And the multiform part is has to do with the fact that it's multiple different cells. So it's not a tumor of one cell type, it's a tumor of multiple cell types. And it's not just a few cell types. It's actually, as we're learning, you can see this is basically a genetic representation of 100 different cells, 100 different tumors that we took from different patients. You can see the genetic kind of profile of these tumors are all pretty different. So you have a tumor with incredible heterogeneity within the tumor, and then that heterogeneity actually changes over time. What we're learning is actually when you treat the tumors, and the next time I go and take out the tumor, that's different. It's now a different tumor. So you can imagine how complex it is to try to devise a targeted therapy for this disease. It's like playing whack-a-mole, and so I know this is the precision medicine session that I'm talking in, but I think what we actually probably need for glioblastoma is not necessarily precision, but what I'm actually going to talk about is personalized medicine. It's something that's unique to each person. So again, getting back to the dendritic cell. So these are, cells that are in the body. They pick up antigen, and then they can activate resting T cells. And then when they activate resting T cells, those activated T cells can proliferate and migrate to other parts of the body, and then attack the antigen that it's actually, activated towards. So that's kind of the general concept. So, many years ago, we did these initial preclinical studies in animal models, and what we found at the time was that by using this paradigm, we were able to get T cells to enter into the glioblastoma tumors in animal models. This seems pretty obvious nowadays, but I could tell you 25 years ago, everyone thought there was no way you could actually induce an immune response within the immune privileged CNS. We see it in, multiple sclerosis and other types of disease states in the central nervous system. But our group was the first to actually show that these T cells can be induced by exogenous vaccination. So then, after, we did the preclinical studies, I actually met this gentleman, Kenneth Johnson. As many of you may know, our cancer center at UCLA is actually called the Johnson Cancer Center. And Kenneth Johnson actually had a nephew with a glioblastoma. So his nephew, who actually was treated at MD Anderson at the time, was looking for new treatments and new therapies. What he and his family did was actually fund our first phase 1 clinical trial. So with that, along with some NIH funding that I obtained, we did this first in-human clinical trial of a dendritic cell-based cancer vaccine. And what that entailed was, after I took out the tumor, that became the source of the antigen. What that entailed was the leukapheresis. That meant we took out the white blood cells from the patient. And then with different types of cytokines and culture mechanisms, we're able to actually mature them or develop them into dendritic cells. And then these dendritic cells were pulsed with antigens. And over the years, we've done many different types of pulsing. We've pulsed with, peptides, RNA, DNA, now computed peptides. But at the end, because of the heterogeneity of this tumor, we went forward and with tumor lysates, specifically autologous lysates from the patient, and then we inject that back into patients. So that trial, started, maybe 15 years ago or so, and it actually was, quite popular at the time. And was able to grow my clinical practice at UCLA and got on a lot of the kind of the news media, I guess programs. This is a Dateline at Keith Morrison, and this patient was one of our clinical trial patients in our first cohort. This gentleman is Meade Thompson. He was actually the Lieutenant Governor of Alaska at the time. So we were able to get funding from Alaska and Sarah Palin's group and a lot of different organizations to fund this. So that led to these initial phase 1-2 clinical trials, which showed some efficacy in terms of improved survival, median survival compared to institutional controls. We had a 3-year survival rate of 47% compared to 21% in institutional controls. And then at the time, the standard of care was this STOOP protocol, which was kind of formalized in 2015. And that led to a 3-year survival rate of 20%, which is still currently kind of the best that we get with this disease. And then at this point, after I finished this trial, I met this gentleman, and he was George Rathman. George Rathman was the founder of Amgen. And for some reason, I'm actually still not sure why, he took a liking to this concept. So one thing he asked was whether I'd leave UCLA and do a startup company that he would be my angel investor for. At the time, I had just had my second baby, and I was still an assistant professor, and I think the risk was too high to kind of go into this kind of new venture. So I said, "Oh no, thank you." But you could fund my next trial. So he gave a few $1 million to fund the next trial, but he went on to invest this money in another company that was being started by one of my colleagues at UCLA, Ari Baldegren. And Ari went on to start multiple companies. And to this day, I always wondered, maybe I should have taken him up on that offer. But anyway, we did do the next trial. Oh, and this is just some data in terms of the first trial showing that we were again able to induce T cells getting into these tumors, and the increased T cell infiltration actually correlates with increased survival. So I think that is a limiting step for glioblastomas. Glioblastomas are what we call cold tumors, so they actually don't have T cells. So that's probably why all these checkpoint inhibitor trials that have been going on, over in recent years hasn't been effective for glioblastomas, because you're not getting the T cells in there to begin with. And then we also looked at different subgroups of glioblastoma and then showed that actually this tends to be more effective in what we call the mesenchymal subgroup of glioblastomas. And those patients tend to have increased survival relative to the controls. So then we went on to a phase 3 clinical trial. Now this was a multi-center randomized clinical trial for newly diagnosed glioblastoma. And this is kind of the protocol. Patients underwent surgery, we took out the tumor. The tumor itself was the source of the antigen. And believe me, we've looked and looked for more kind of targeted therapies for this, but the autologous tumor lysate is still currently, our best source. And I think that's because of the heterogeneity of this tumor. Patients then underwent the leukapheresis to get the dendritic cells, and then we put the dendritic cells together with the antigen. So the antigen presenting cells together with the antigen outside of the body, because theoretically these antigen presenting cells never actually interact with the antigens within the brain, because they don't actually circulate in that fashion. So then we went on to do injections, and then patients then went on to follow up and adjuvant chemo and radiation. So this trial was done at 94 sites in 4 countries in the United States, UK, Germany, and Canada. It took a long time. And this was essentially the protocol again. Surgery, patients were screened a week before surgery, and then a few weeks after surgery underwent the leukapheresis. And then we did the DC manufacturing after surgery. This usually took about 6 weeks, but patients had to undergo standard radiation for 6 weeks during this time anyway. And then patients were randomized at this point to either get standard of care DC Vax. This is now called DC Vax because it was licensed to a company that gave it that name. So again, international multi-center study endpoints were progression-free survival and overall survival. We had a central MRI review with 2 separate radiologists who were blinded to the actual diagnosis of the patients, and then central pathology, and then these kind of mutational analyses. So what happened? Some challenges came up with this. One of the things is, like I said, one of the endpoints was progression-free survival. So when the trial was actually written in 2007. So when that was written, we were still using McDonald criteria, and these are the criteria by which we look at MRI scans and measure progression. So if you saw a scan like this, and then you saw that the tumor got bigger in two dimensions, then you would call this progression. So then the patients actually came off the trial and then went on to something else or crossed over. But then subsequently in 2010, 2015, 2017, they kind of rewrote the way we analyze these scans, because we began to realize with glioblastomas, there's something called pseudoprogression, particularly when given immunotherapy. So the scans actually get worse, but then they get better. So this really is not necessarily tumor progression. It could be this kind of inflammatory or immune kind of response. But it's really hard to tell when there's pseudoprogression versus real progression, and that's still currently a major challenge in our field, because we can't go in and biopsy these every day. So it's been kind of a difficulty in determining this. And then this is just another kind of view of this. So a lot of patients who actually, where we see that it looks like the tumor is growing, but in this particular patient, we actually did nothing because he was fine. So as you watched over time, this new area of enhancement actually went away. And then plotting this out, there is actually this window of pseudoprogression that typically occurs when an immune response is seen. So how did that impact the clinical trial? It's not advancing. So again, this is the trial design, and then after the radiation and chemo, patients were randomized to DC Vax or placebo, and then if there was progression, so the endpoints, like I said, progression-free survival and overall survival. So if there was progression, patients then had the opportunity to cross over to get the actual treatment. And the reason this was written in actually was because actually the FDA kind of felt that the leukapheresis was an invasive procedure, and it wouldn't necessarily be fair to do that to patients without some opportunity to get the vaccine once they came off the trial. So that sounded very ethical and reasonable. But then the problem is then 90% of the patients actually wound up getting DC Vax at recurrence. But then the problem was we didn't have a really good way of definitively defining recurrence, because if you use the McDonald criteria, what we later found out was that half of those patients actually had pseudoprogression, so they actually did not really recur. So then what happened? Oh, and this is just a timeline of how long all of this took. So I met Mr. Steinman in the late 90s, and we did our first phase 1 trial, at 2000. We did phase 2, right in here. And then initially we actually started a non-blinded randomized placebo-controlled trial, a phase 3 trial here in 2007. But one thing I learned here was that if you non-randomize, meaning the patients knew that they weren't getting the treatment, all the control patients drop out. And you can imagine if you had a glioblastoma and someone told you, oh, you randomized to placebo, the first thing you're going to do is jump off that trial. So unfortunately, we didn't get a lot of people staying on the control arm for a non-randomized trial. And then what happened was the recession of 2008 hit, and the company wasn't able to raise money to continue. So the trial was kind of on hold until, in 2012, it started up again. And this time we did it blinded, non-randomized. We made, vaccine for everybody. And then that's what led to having to have the crossover arm. So once we did that, the trial actually accrued in 3 years. So that, by the end of 2015, the trial was finished. And then we observed the patients for median survival for 3 more years after that. The initial interim data was actually very promising, so this was the intent to treat population, 28% 3-year survival. And this was, the entire population. But the problem that I wanted to get to was, so this was both arms, placebo and treated arm. But as I mentioned, the placebo arm had the opportunity to cross over, and 90% of the crossover arm then also got the vaccine. So if the endpoint was overall survival, that would be relevant if the vaccine did not have a beneficial effect at the crossover, at that recurrent point. But as it turns out, it probably does at that recurrent point. So what we then, actually the statistical analysis plan that was submitted to the FDA was to use external control arms. And I won't go into the kind of the statistics behind the propensity matching and the dynamic borrowing, but anyway, there was a statistical way to get external controls for this trial, and it's being used more now in these trials going forward, because quite frankly, very few people want to be the placebo arm of clinical trials. So the best, based on the literature, the best data source for these kind of external controls are patients from large, well-conducted randomized controlled trials, rich with baseline characteristics. So as it turns out, and this is just the statistical analysis plan that was submitted to the FDA, primary endpoint, secondary endpoint. So the primary endpoint was, it was essentially overall survival. And then there are different, proportional hazards that we looked at that could also give a signal in terms of a positive response. The secondary endpoint was actually post-recurrence survival. So basically, when patients crossed over, you imagine that pool of patients actually became a single-arm, first-time recurrent study population. So that was primary and secondary. And so as it turns out, in terms of, kind of looking for the external controls, there were, over the same concurrent time period that this trial was conducted, there were 5 very large multi-institutional studies for glioblastoma, and all their control arms had the same thing. They all had radiation and temozolomide. So there was 1,366 patients who all got the same thing, all had very extensive data collected on them. So they served as the control arm for this, for our study. And then on the recurrent side, there were a number of publications as well. And all these people had the same treatment. They all had either CCNU or bevacizumab, because, like I said, there's not a whole lot else that's approved for glioblastoma. So these control patients all pretty much had the same thing. So using these controls, again, we went back to the main study. So the main study then, as I said, initially had 331 patients, 232 randomized to DC vaccine, 99 randomized to placebo. It was a 2-to-1 randomization. And then, people these were the people who, continued on the vaccine, even though they had recurrence, because at this point, they're blinded, right? So you don't know who was in which arm. So, so later when, we found out that of the people that had recurrence, and noted recurrence could have been pseudoprogression, we actually don't know. Of those people, 120 were on this arm and 64 were on this arm. So overall survival, so we kind of did these comparisons. Overall survival was actually quite good compared to the kind of these landmark survival rates. And like 5-year survival for this control arm from the external control studies were 5.7%. Our control arm was 13% at 5 years. But interestingly, when we took the recurrent population, that was, that actually showed, increased survival, even more so with a 11% survival at recurrence. And then these are the subgroup analyses. I won't go through all of these, but I did want to mention MGMT methylation. So this is one of the markers for sensitivity to radiation and chemotherapy. And, a lot of clinical trials for glioblastoma, they actually exclude this population because people think that, oh, well, they do seem to have a response to radiation and chemotherapy. So we'll treat the unmethylated group on immunotherapy trials. But as it turns out, immunotherapy was probably more effective in the methylated group as opposed to the unmethylated group. And so there was an increase in median survival. But what was even more interesting is, so here, this tail actually, about 25% of these patients, they actually kept going on, so even past 5 years, they not only did not have recurrence, they actually, they not only survived 5 years, they actually did not have recurrence. So this tail actually went all the way down to 0 or kind of very small amount, whereas this tail kept going on. And those patients are actually doing remarkably well. So there obviously is some subgroup of these patients that tend to do better than others. And part of this could be that they're more somatic mutations related to MGMT methylation. So in conclusion, there are challenges for randomized controlled trials for glioblastoma. Patient recruitment and retention is difficult. In placebo-controlled trials, there's lack of active treatment controls. And glioblastomas are rare. It's a rare heterogeneous disease compared to other cancers. So like I said, the numbers aren't that big, even though they're, they're very fatal tumors. A lot of drug companies don't necessarily want to invest in this particular, subgroup of patients. And then even within that small subgroup, there's so much heterogeneity. So it's very hard to get a targeted treatment. And then we have, imaging issues, as I mentioned, the pseudoprogression. And then it's a rapidly progressing field where the knowledge is growing even during the course of the trial. So if the trial takes 10 years, there's knowledge that occurs throughout that course of time. So I just wanted to end by saying thank you to all my collaborators and colleagues at UCLA. And thank you for having me here.
Thank you so much, Dr. Liao, for such an informative talk. The podium is now open for the questions.
Based on what that you gave the patient the vaccine for brain cancer, because my aunt had brain cancer, is by age or by some symptoms come up that you give people the vaccine, and how effective is brain cancer vaccine? Thank you.
Oh, good question. So this vaccine, we don't have any good correlations of, like what causes brain cancer. So it's not like the, like the cervical cancer vaccine where, everyone kind of gets the vaccine because there's a virus that may predispose you to having cervical cancer. So this is more for people who are already diagnosed with brain cancer. And, we've done the surgery. but what we're fearing, and so it's used in that context. Yeah, good question. Yeah, I mean, in this clinical trial we did, because like I said, it started, about 10 years ago. I think in newer clinical trials, there are some that are enrolling patients and not, giving them Timidor. And but it's interesting. There was actually a trial recently done with a PD-1 inhibitor, an immunotherapeutic drug inhibitor. And what they did was they took the unmethylated patients because they think, well, they're not going to get benefit from Timidor, so we'll enroll those patients in that trial. And actually, it turned out that those patients did worse than the controls because we denied them Timidor. So I think there is kind of this thought that we want to give drugs other than Timidor in these trials, but until we know that one is effective, it's a little hard to really kind of do that. Okay, thank you.
Great, awesome. Thank you again. I'd just like to quickly point out that we're also very honored to have Dr. Bert Snyder joining us today. So actually, Dr. Liao's husband is Dr. Bert Snyder. If you can just wave your hand. He's a professor of the neurosurgery and also director of the pituitary tumor. So this is kind of like a buy one, get one. If you have questions about tumor, this is the family to go to. All right. Hello. Hi, it's me again. So we're very honored to have our next speaker, liver cancer clinician and researcher of epidemiology, health disparities, and biomarkers. So he is the assistant professor and director of the liver cancer program at Cedars-Sinai Medical Center. He, along with colleagues, pioneered this test of liquid biopsy, apparently theme of the day, to detect liver cancer early. So apparently it's highly accurate, cost efficient, user-friendly, fast, and ultimately life-saving. I want this test too. Ladies and gentlemen, Dr. Judong Yang.
Good afternoon, everyone. And I'm very grateful to Professor Tseng and the members and guests for this opportunity to present today. And my name is Judong Yang. I'm working as a transplant hepatologist at Cedars-Sinai Medical Center. Today, I'm going to talk about the liver cancer surveillance and early detection of the liver cancer. So this slide shows the outline of my presentation. I'll start with the clinical case that I saw during my fellowship training, and emphasizing why ACC surveillance is so important in patients at risk of the liver cancer. And then I'll go over the data on the role of the ACC surveillance. And then last part, I'll go over a few novel surveillance tests for hepatocellular carcinoma. So this is a patient, a 30-year-old man, and who had a strong family history of chronic hepatitis B. Mothers and then 3 brothers and sisters have the chronic hepatitis B. And patient was diagnosed with the cirrhotic stage liver disease when he was at his 20s, quite at an early age, and he was started on the antiviral treatment for hepatitis B. With the treatment, hepatitis B viral DNA was not detectable, and then liver disease has been well under control. So he was not seeing the physician very regularly. and of course didn't have the cancer surveillance. And the patient started developing some right upper quadrant pain and night sweat, not feeling well. So he presented to us for further evaluation. So on examination, patient was pretty healthy looking, but has the enlarged liver when I was palpating his abdomen. And also a liver test shows mild elevation of the liver enzyme, and also tumor markers, especially alpha fetoprotein L3% and DCP, two of the liver cancer tumor markers were elevated. So we went ahead and then arranged an MRI of the liver. Unfortunately, it shows the huge liver mass and with the portal vein invasion. So he was diagnosed with the advanced stage of the HCC only at the age of the 30s. And he received the Y90 treatment. Y90 treatment is one of the local regional treatment to the liver cancer, delivering radioactive material directly into the tumors using a catheter. And then, unfortunately, patient developed the liver failure, and in part due to the progression of the cancer and side effects of the treatment, and he died in 4 months after he receiving the cancer treatment. Very, very unfortunate and sad case, and at the age of the 30s, presenting with the advanced stage of the cancer in the absence of the cancer surveillance, and we lost this patient. So emphasizing the why the surveillance could potentially be important for early detection of the cancer, which could have changed his life. So HCC surveillance, the improved the survival, multiple different type of the study have demonstrated that this is one of the study from the VA, where they show that when you look at the patient with the liver cancer and those cancer patient whose cancer was diagnosed under surveillance have about 29% reduction in cancer mortality. This is one of the study that I conducted as a fellow, that patient who had the liver cancer detected under surveillance as the more likely to receive the curative treatment and then prolonged survival. And subsequent meta-analysis of the 15,000 of the HCC patients shows that it is associated with the early detection of the cancer, curative treatment received, and prolonged survival. And most of the study, however, was conducted among those who was diagnosed with the cancer and compared the survival between versus not. But there's not really strong evidence that the surveillance improved the survival in cirrhotic patient population. Cirrhotic patient who has been under surveillance versus cirrhotic patient and who has not been under surveillance, whether it affects the survival. So we conducted this Markov modeling study with the base case of the 50-year-old well-compensated cirrhotic. We modeled that the patient can die as a result of the liver failure, which is actually more common cause why people die. And also a patient can develop the cancer and cancer-related mortality, but surveillance can increase the chance of the early stage cancer detection. So when we model this, the HCC surveillance can reduce the mortality by 5.2%, any cause of the death by 5% reduction in cirrhotic patients, but reduce the mortality by 33% with the HCC surveillance. So while the HCC surveillance is good, actually improves survival, but unfortunately, the patient I showed you earlier is a very common presentation of the HCC patient in the nation. And this is the one of our study that we recently conducted using the population cancer database at Here Medicare, that we identified 5,000 of the liver cancer patients, and only 15% had the regular surveillance before the diagnosis of the HCC. And when the previous study and using the VA nationwide cohort study also showed the similar estimate, only 12% of the cirrhotic patient undergo surveillance program, which is a huge problem. So let's move on to the surveillance test itself. What tests are we using for the cancer surveillance? We use the serum alpha fetoprotein for the surveillance, and also we use the liver ultrasound for early detection of the cancer every 6 months base. And unfortunately, those are not very accurate tests. And so ultrasound, for example, can only pick up the early stage of the cancer in half of the time. And then half of the time it means early stage of cancer. So it's not a really good surveillance test. And then also it is recommended, therefore, to use the alpha fetoprotein, one of the serum protein marker, to increase the accuracy of the surveillance program. But despite adding that, the sensitivity is only 63%, meaning that a third of the early stage of the cancers are missed with the serum alpha fetoprotein and ultrasound of the liver. So which is another huge problem. First, the underutilization of the surveillance. Second, the low accuracy of the currently available surveillance tests. So there are some other blood-based biomarkers that we use in our practice to address this the unmet need. One of which is the serum alpha fetoprotein L3%, which is the physicollated isoform of the alpha fetoprotein, and it was shown to be elevated in patients with the HCC. And also DCP is the one of the immature form of the prothrombin protein that can go up in the setting of the HCC, and those are the FDA cleared for HCC risk assessment. So let me briefly introduce GALAD score that we often use for cancer surveillance. One of the most promising, widely used the surveillance test in some practice, including my own practice. So GALAD score is a model based on 2 demographic risk factors, men and old age, which are associated with the risk of the HCC, and then 3 serologic biomarkers for liver cancer: alpha fetoprotein L3%, and DCP. So based on those 5 value, you can calculate the GALAD score. It's initially proposed on about like less than 10 years ago, study from UK, Germany, and Japan, large number of the patients, 2,000 of the HCC, and close to 4,000 of the control patient population. That the GALAD score has the area under the curve way above 0.9 for early stage of the HCC detection. So we conducted the validation of the GALAD score in the US for the first time, and this is and also the first study compared the performance of the GALAD score to the ultrasound of the liver, which is a standard cancer screening test for HCC. That the accuracy area in the curve was higher with the GALAD than ultrasound, and also early stage of HCC detection too. So ultrasound the accuracy is compromised in patients with the advanced stage liver disease, like someone who had a larger ascites or the advanced cirrhosis. That area under the curve is compromised, but GALAD perform very, very nicely throughout. And then we also propose the combination of the ultrasound and then GALAD score for early stage cancer detection with the outstanding performance for early stage cancer detection. And this is one of the meta-analysis that Peter, I think Peter is here, one of our brilliant postdoc, and then postdoc of the Professor Stang's lab. And he put together this meta-analysis, and of the all publication about the last score for early stage of the cancer detection. One thing I'd like to emphasize is that the in phase 2 study, this performance is really outstanding, but unfortunately, when we summarize the result of the phase 3 study, for early stage of cancer detection, the area under the curve decreased down to 0.73, which is somewhat disappointing that many of the, as you know, the biomarker, as they advance in their phase of the biomarker study, that usually the performance decrease, and that's what happened with it a lot. So definitely we need a better biomarker study for early stage of cancer detection. And let me actually spend some time to discuss about the potential advantage of the blood-based biomarker over imaging biomarkers. And before I move on to the novel approach, blood-based biomarker for early detection of HCC. Blood-based cancer surveillance test is much more convenient to the patient because you don't need to go through the hassles of calling the imaging center to schedule radiology, ultrasound, which sometimes take a month to make an appointment, whereas the blood test is so easy to schedule, or often you don't even need a schedule. Actually, you just walk in and then get blood test done. So it's much easy. Usually cost is lower with the blood test than ultrasound. And the ultrasound has the issues with the operator dependency. So someone who is really, really good have the better performance. Someone who is not very good miss a lot of the tumors in the potential tumors in the liver. But you have the very objective result with the blood-based biomarker test. And also some of the survey study reported that the patient prefer blood-based surveillance test than imaging, so ultrasound surveillance test. And it is easy to get done, and the compliance is supposedly higher with the blood-based surveillance and imaging-based surveillance test. So now introduce the work that our team has been focusing on for the past years under the leadership of the Professor Tseng, that the HCC-EB surface protein assay, which combine the two major components, the chemistry, for the isolation of the EB, and the immuno PCR for its quantification of the subpopulation of the HCC-EB. And so we basically get plasma from the at-risk patient population, and use the liver cancer associated antibody. We use 4 of them, and at the antibody, end of the antibody, they have the structures that can TCO-grafted antibody have the click chemistry reaction with the TZ, the structures in the click bit, with the click chemistry that we can capture this HCC-EB, and later on using the DNA barcode, the antibody and the CD63 and CD9, that we can enumerate this subpopulation of the HCC-EB, and using the immuno PCR. And then again, using these 4 HCC associated EB, and time is up. Okay, I'll go quick. Okay, maybe some overlap with what Professor Zhu presented. So with that, that we calculated the HCC-ECG score, and we tested, developed the models, and then using the UCLA cohort, and then validate the performance at Cedars-Sinai cohort. And then, as you can see, that this score has the outstanding performance with the area under the curve of 0.95 at the UCLA cohort, and Cedars-Sinai cohort 0.93 for early stage of the HCC detection, showing outstanding performance. And the We did multiple subgroup analysis showing again the outstanding performance of these tests in conclusion. So, and the HCC surveillance improved the outcome of the patient with the cirrhosis, but unfortunately, it's not, it's underutilized. And also current standard, the surveillance test has very low accuracy. So we need the better markers than our team. And again, under the leadership of the Professor Sang that has been working very hard to develop their markers that we found very promising approach. Hopefully we can validate this in a larger study. Thank you so much.
Thank you so much, Dr. Yang, for this enlightening talk. Any questions? Now's your chance. All right. Thank you so much.
Thank you.
So a nanometer is 1/1000th of a meter. How small exactly is that? A strand of human hair is about 80,000 nanometers thick. So shrink that by 1,000. That's approximately how our next speaker operates at that scale. So clinician scientist of pediatric hematology oncology. Our next speaker is actually an expert and is the leader for a multidisciplinary team that tries to design new nanotechnologies to manufacture novel gene and cellular therapies. Ladies and gentlemen, Dr. Stephen Jonas.
Thank you, Francie, for that kind introduction, and obviously thank you to HR and all the organizers of this event for the opportunity to come share a little bit of our work today to hopefully apply research into the tiniest tools to help some of our smallest patients. My only disclosure is that any IP that we talk about today is assigned to the Regency University of California. Got to get that out of the way. But as a pediatric oncologist, when I think about precision medicine, I think about how we're trying to improve traditional cancer therapy, where, you know, ultimately at some point in their disease course, a patient is going to be exposed to some form or combination of either radiation, surgery, chemotherapy. Increasingly, we're thinking about targeted therapies, immunotherapies, and potentially hematopoietic stem cell transplantation. And, you know, we're fortunate enough in the pediatric space that overall cancer survival rate's actually around 85%. But, you know, that's not a good enough number for us. And ultimately, our best therapies still come with a tremendous amount of toxicity and limited access to some of the best interventions if you're not able to be diagnosed and treated at a place like UCLA. So we want to close that gap somehow. And that's really motivated the application of precision medicine approaches to hopefully personalize treatments for different types of cancers and also, you know, in general in the pediatric research away from traditional paradigms where we establish a diagnosis, formulate a treatment plan, implement that plan, and then reassess, and ultimately upfront have a therapy that's personalized to the individual. In order to do that, though, you really need to fuel that precision medicine engine, as we've kind of heard about in a lot of these talks today, with a lot of big data. You need new tools to really understand and interpret that big data, and ultimately new ways to deliver that care. And so our group is looking at the number of different ways to accomplish that and new solutions that we hope will contribute to this space. I have a PhD in material science as well, so that's kind of my weird oddity in the pediatric community. But you guys have heard a lot about liquid biopsies today. I'm going to avoid kind of talking about that, but hopefully tell you a couple brief stories about what we're thinking about in terms of hopefully moving towards some precision medicine-based therapeutics in pediatrics in terms of manufacturing different types of cell-based therapies or solutions for that. So when we think about cell-based therapies, whether it's gene therapies, where essentially you take a gene or increasingly a gene editing construct to insert or correct a disease-causing mutation in a population of cells, or in the adoptive cellular therapy space in oncology, you guys think about CAR T cells or CAR NKT cells, where you take an immune cell and you give it a set of genetic instructions that teach the immune cell to basically target and harness the immune response against the cancer. The one thread that really, in our mind, connects these areas together is that at some point, for a large population of cells, hundreds of millions, in this case for a CAR T cell dose, or up to billions of cells for some types of gene therapies, you have to deliver that gene editing cargo efficiently, hopefully safely, quickly, and ultimately cost effectively, because these therapies are expensive. A CAR T cell dose is $500,000. Some new gene therapies are going to be up to $1 million to dose. And that really limits who can gain access to that kind of care. And so we want to be able to democratize these therapies, and we hope that there are engineering solutions that will allow us to do that moving forward. That's really what our group is interested in. And the one unmet need, or one of the unmet needs that we hope will help lead to that democratization of these therapies, is what I kind of think about as a biomolecular delivery service. That's sort of what's missing, right? We think about FedEx, we think about Amazon, and in our pandemic lives, we get these boxes because those companies figured out how to put things in boxes and get those boxes where they need to be. That's the same issue in this space. We have so many amazing scientists out there who work on different types of cargoes for gene therapy, but they might not have that the means to get those cargoes where they need to be. So our group thinks about the different types of packaging and the different types of delivery vehicles that can hopefully get those cargoes where we need to be, so that we can hopefully democratize these therapies moving forward. So when we think about these cargoes, they can be any number of different either nucleic acid-based or protein and CRISPR-Cas9-based approaches. Increasingly, we think about base and prime editing based cargoes in order to make engineered cell products for cell therapies. But how do you package them into a shipping container that can get them to where they need to be? And so when we think about that, we think about different types of nanoparticle carriers. And so together with Professor Seng's group, we've looked at a number of different supramolecular-based strategies to incorporate these cargoes for different applications, and increasingly, especially in the I guess we could say the post-COVID era, maybe, I'm still wearing a mask, lipid nanoparticle-based strategies, because both of these systems provide us with a great deal of synthetic versatility, but also the versatility of being able to incorporate different types of cargoes. And HR, that TM's for you. That's hard to get. Stay with that one. But an example of that in our work right now is a collaboration that I've got with two of my colleagues and mentors in pediatrics, Dr. Bridget Gompertz and Dr. Don Cohn, and how we're thinking about delivering gene therapies for a disease called cystic fibrosis. And cystic fibrosis is a disease where you have a defective membrane transporter. And with that dysfunctional membrane transporter, patients ultimately suffer from recurrent lung injury and infections, and ultimately pass away at an early age because of these respiratory injuries. And so there are modulating medications out there that provide the ability to potentiate that dysfunctional receptor to a certain extent, but there are no definitive cures so far. And gene therapies might represent that kind of definitive cure because CF is an optimal target for that, because CF stems from a defect in a single gene. You just have to be able to target. Problem is, you just got to access the right type of cells in the airway, these airway basal stem cells. And that can be a sticky situation, kind of no pun intended, because at some point you got to get that cargo through a sticky mucus layer, through a layer of epithelial cells, and ultimately to the airway basal stem cells that you want to correct in order to deliver the cargoes that will enable the permanent gene correction for the disease. That's an interesting problem to us. And so that means we've got to modify the nanoparticles in a way that will hopefully allow them to penetrate and circumvent those barriers, the mucus barrier, and ultimately disrupt the epithelia transiently so we can get the cargoes to the stem cells without damaging the whole system and killing the lung. When we think about how we're going to engineer this, we need a way to model this. And so Dr. Gompert's lab has developed what were called air-liquid interface cultures, models of the respiratory airway, where on a cell culture insert, she cultures the airway basal stem cells, and we're able to basically recapitulate the ciliated and secretory structures of the mucociliary epithelium on this cell culture insert. And the cool part is that the cells are exposed to air, which allows us to use vibrating mesh nebulizers to basically nebulize different nanoparticle vapors, latent vapors onto the airway cells so that we can test out the delivery characteristics. And so when we do that with the two types of nanoparticles that we're talking about, I mentioned earlier, we see that the supramolecular nanoparticles are a little bit more harsh. The GFP delivery for a GFP reporter cargo that we load the nanoparticles with, sorry, yeah, thank you, that's helpful, is a little bit more stipulated. The cells look like they're coming off the plate. They're not happy. The lipid nanoparticles look a little bit more happy, and the delivery is a little bit more distributed. And so that leads us to really kind of hypothesize that, in this case, lipid nanoparticles might be a better solution. And that really emphasizes that there's going to be a specific cargo for specific applications. And that's important for what we do and what we want to engineer moving forward. We want to be able to optimize the nanoparticles to the different cargoes. And I'm not going to spend too much time on this slide, but gene editing cargoes are much bigger than GFP constructs. So we're using microfluidic-based approaches to really optimize the size distributions of these particles as we engineer them and hopefully make the synthesis more reproducible. But how do we incorporate the epithelial modulating capability? And to do that, we're trying to incorporate a molecule called polydocanol into the nanoparticle. And polydocanol is an interesting molecule because it's actually used by stem cell biologists who are interested in airway science to basically slough off the airway in vivo models to characterize airway basal stem cells in mouse models. And so we thought, well, maybe we can add that functionality to it lead to a transient disruption of the epithelia to access the stem cell. Bridget's group has also, in characterizing that this transient injury, seen increased reactive oxygen species generation with exposure to polydocanol, which we think we can leverage to have increased self-renewal and increased cell turnover from the airway basal stem cells, which we hope, if we're delivering a Cas9 directed CRISPR-Cas9 editing material that's configured for homology-directed repair for the gene correction for cystic fibrosis, we can take advantage of that because that process ultimately requires active cell division. So Ruth Foley, a really talented PhD candidate in my lab, she just passed her qual, so congrats to her. She has been exploring how to incorporate polydocanol into the nanoparticles using zeta potential analysis and dynamic light scattering. We see that she's able to incorporate these ligands onto the nanoparticles while not disrupting the size distribution of the particles adversely, and also being able to retain their delivery capabilities when they're loaded with a GFP reporter construct when using a T84 cell line as a model cell line for in vitro study. When we're testing the epithelial modulating capabilities of these types of particles, and we aerosolize polydocanol linked lipid nanoparticles onto the ALI inserts, what we see, which was interesting, is that we can get similar exposure of the stem cell populations, which are marked in red, I guess it's red, sort of purplish, by this keratin 5 stain. But after 24 hours, we get more healing and better regeneration of the particle of the ALI with LNP-linked polydocanol, as opposed to ALIs that are exposed to just neat polydocanol. And so we think that we can leverage that better or more transient disruption of the epithelium to make sure that the airway is intact and healed as we kind of move on to more clinical translational applications. You know, that's also leading to Ruth delivering, demonstrating delivery of different reporter constructs in the ALIs. And ultimately, we want to optimize the nanoparticle for particular applications. And to do that, we want to accelerate that even further. We can't just do one condition at a time. We want to do this combinatorially. So applying different types of 3D printing approaches, our group's looking at developing different types of ALI screening platforms where we make a device where we can basically distribute individual vapors of nanoparticles to a particular airway liquid interface culture in individual wells of a standard plate. We have current configurations of these devices. They snap together with MagSafe if you're an Apple fan, with in 24 well configurations and also in 96 well configurations moving forward. So we can hopefully test 96 configurations at once to really help us hone down on the right nanoparticle for the right job in this case. In the really brief time I have left, which I don't know, I just want to talk a little bit about different delivery vehicles to a certain extent, to some of the other delivery vehicles we're thinking about. That was the packaging. This is the vehicle. In classical stem cell, in classical gene therapy-based approaches, viral vectors have been kind of the workhorse and the enabling tool. However, they have limitations in terms of the size of the cargo you can deliver. They're really expensive. They have to be individualized for every particular therapy, and there's issues with immunogenicity. Existing non-viral based approaches potentially have issues with needing to have instrumentation dependence of expensive instrumentation, variable viability, and issues with scalability. So the bioengineering community, real fast, has looked at biophysical approaches because there's no ideal intracellular delivery system that meets all of these parameters. And so we're looking at a portfolio of different approaches because we don't think there's going to be a silver bullet. There's going to be certain applications that need one cell type over the other. One of those application tools in our toolkit that we're thinking about is to use sound waves to actually open up pores in the cells using something called acoustofluidics. And this is in collaboration with Tony Wong, a professor and colleague at Duke, where we take cells and we pass them through a microfluidic device that's integrated with a piezoelectric. And that piezoelectric allows us to transmit an acoustic wave into the microchannel. And that microchannel, and as the cells pass through the acoustic radiation field, the cells are stretched with shear forces and ultimately rendered porous at both their plasma and nuclear membranes. And so Jason Belling, a talented grad student, had in proof of concept studies, was able to demonstrate the ability to deliver model cargoes to model cell lines, as well as primary cell types, peripheral blood mononuclear cells, and CD34 positive human hematopoietic stem cells. These last two slides. And Yao Gong, who's since taken over the project, is now taking advantage of the supramolecular nanoparticle systems from Dr. Seng and making CAR T cells out of these using sound waves essentially. She's able to get pretty respectable transduction of these cells using Sleeping Beauty encoding plasmids. And ultimately, we're assessing the cytotoxicity. And these are kind of initial experiments that were from this week, but you can see the green cells attacking the orange cells, and hopefully we can make CAR T cells with sound waves, which we think is gonna be cool. And in that, hopefully I've been able to share with you guys that We want to make precision medicine available to all clinicians and all scientists. And to do that, you might need some engineering approaches, and hopefully these technologies are steps in that direction. So I had to thank the folks who actually do all the work, my mentors and colleagues who entertain some of these crazy ideas, and the folks who kind of pay the bills. And so thanks for that, and I'm happy to take a question if there's any time left.
Hi, Steve.
Hey.
Yeah, my name is Dan Cal. I work for JPL NASA. And you, couple of questions here. Actually, it's one question.
Yeah.
How do you do the verification and the validation? One is your model. How do you verify your model? How do you know your model is correct? Second one is you talk about transportation for certain the path.
Yeah.
How do you know every single path indeed follow exactly what you planned? How do you verify that?
Yeah, so I mean, in terms of the first part of that question, it's you verify the model using kind of the cargo, the particular reporter cargo initially. So that's what a little bit of that the GFP kind of expression is. That kind of helps us verify that cargo is getting to the cell and actually being utilized by the cell to create. You can see it with a microscope or with a flow cytometer, and ultimately, with more complex cargos for gene editing, you want to look at different sequencing approaches to really verify that your gene editing is happening at the right target with minimal off-target effect. And so there are a number of different approaches out there like TIDE or ICE that look at that. In terms of where you step through the model, in terms of that delivery, I think that's when we move towards in vivo models and really see if we're actually able to access cells in airway stem cells in a CFTR deficient rat, in the case of that example, to see if we can get the type of correction that we need. No worries.
Okay. Oh, there's one question.
And we're always looking for grad students and postdocs. So anyone out there? But sorry to go over a little bit. I blab a lot.
Yeah, I think a final question. Great research. Since the size and the charge of a nanoparticle lipid is very important, do you have a data by the time the size of a lipid particle by the time the size of aggregation? Do you have a time for that? Because sometimes you measure the dynamic scattering the sizes by a certain time.
Yeah.
Yeah.
Yeah, I mean, we try to do all
the
this.
Second, do you have a data to measure the nanoparticle charge? It's always constant. Thanks.
Sure. Yeah, we try to use these particles as fresh as possible with the synthesis at this point, just because we don't want to introduce any variability like that with aggregation. With the supramolecular nanoparticles in particular, we kind of, when we do repeat DLS and electron microscopy, we do see a lot of aggregation if they're not stored at cryogenic temperatures over an extended period after about a week, essentially. And so that's really motivated us to kind of do everything a la carte at this phase, if you will. I forgot the last part of your question. Is that charge? Yeah. So the charge, that's where we're looking at data potential in essence. We see that the charge remains relatively constant within kind of the range that we're looking for, with the particles after within the day or two that we have them before we expose them to the cell. We haven't looked at it more long term. I think that's part of the next steps of what we're moving towards.
Dr. Jonas is truly a wonderful physician scientist, and thank you so much for such an illuminating talk. And now we're delighted to invite Deng Kai from Flight Systems at JPL to present certificates of acknowledgement and souvenirs to the 3 speakers. So gonna have Deng Kai here. And Dr. Liao and Dr. Yang and Dr. Jonas should come to the stage so we can present. Certificates of appreciation and acknowledgement. So we're gonna have the certificate for Dr. Liao first.
It's not red color. It's closing up. Okay, and I really appreciate it. You know, so grateful to have you give a wonderful talk. I enjoyed it very much. Yeah. Okay, thank you. So let's have a picture here. I'm sorry. If you don't mind, can stay here just one more minute. We're gonna have a group picture here. Okay. Okay, next one. No, this one is Yang. Okay, cool. Then will be Steve here. Okay. Really appreciate your talk.
Thank you so much.
Don't tell anybody else. Finally, I understand something.
Yeah. We'll make sure to start. Right?
Yeah, exactly. Okay, so I'm going to take over from Francie. Thank you so much, Francie. Whenever Francie show up, always infuse tremendous amount of energy to our crowd. Okay, thank you, Francie. So I'm going to quickly move to sponsor talks, and it's going to be presented by 3 entities. So I'm a bit of a selfish, and also considering our previous talk was given by a pediatric oncologist, Steve. So we're going to have our junior get onto the stage. So they represent UCLA early academic research program. So they will do a 5-minute, one 5-minute presentation, one 2-minute presentation to let everybody know what the juniors are doing on UCLA campus in the research laboratory. So without much to say, the first group, please get on the stage, and Bernie, slides, please.
Hi, everyone. My name is Audrey Qian, and I'm an undergrad at UCLA. And I'm the mentor for 2 amazing high school mentees, Katharina Wang and Alex Wu. So Alex is a senior at Santa Monica High School, and he's done a lot for this project by carrying out a lot of the experiments and even coming to the lab during the weekends to extract very important samples. And Katharina is a junior at Central Jersey College Prep Charter School, and she has done a lot of organization for the logistics of this group in addition to carrying out the lab protocols. It's a pleasure for me to be able to mentor these 2 students, especially when they have sacrificed a lot of their time during the school year for this project that they have continued or started during the summer. So Alex Wu and Katharina Wang.
Hello. So we are part of the UCLA Early Academic Outreach Program.
co-directed by Dr. Tsang and Dr. Chu, that allows high schoolers like us to pursue our interests in the field of research. And this is a student-initiated project called the Fucoidan Project. So what exactly is fucoidan? Well, fucoidan is an acidic polysaccharide composed of sulfated sugars that can inhibit tumor growth, block the formation of tumor blood vessels, and activate anti-tumor immunity. And fucoidan is typically found in various brown seaweed species that can be found along the northern Pacific and Atlantic coast of the U.S. And it's typically extracted by treating raw seaweed with hot water or acidic solutions via microwaves. But we developed a new method, which is using a hot water bath. So how the project started was a combination of my interest in marine biology along with previous mentoring experience on another project. And the main goals of the project are to quantify the concentration of fucoidan in each of three components of bladderwrack. So that's the bladder, the stives, and the blades of the bladderwrack. And the second goal of the project is to test fucoidan's anti-cancer effects against lethal pediatric malignancy Ewing sarcoma through cell culture study.
So over the summer, we've been working on a couple of things. First, we read previous research articles about fucoidan and its anti-cancer effects, as well as the extraction methods used. Then we moved on to developing our own sample. So as shown in the picture, this is our extracted sample. And we extracted different parts of the bladderwrack as well as the entire plant. And then we used electrophoresis to characterize our fucoidan sample. And recently we've been working on gel permeation chromatography, or GPC, where we found out that bladderwrack stipes had the highest concentration of fucoidan, while the blades had the widest distribution in terms of fucoidan molecular weight. And we also did cell culture of urine sarcoma, specifically the SoCal1 cell line, and we used that to harvest EVs. And we are also analyzing the fucoidan beneficial effects by comparing specific gene rearrangement expressions and of urine sarcoma through digital droplet PCR. And we're also working on paper publication as well as a potential product development of our fucoidan samples. So lastly, we would like to give special thanks to our amazing mentor, Audrey Chen, who helped guided us through the project and in terms of organization, research, and how things work in the lab. So she's been really helpful throughout the project. So thank you, Audrey. And if you're interested in any further information or you guys have any questions, feel free to stand by our station, so behind the room, so like in that corner. We'll be there and we can answer any questions and provide any details of our project. Thank you.
Thank you. So we also have another student-initiated project. It's going to be presented by Lucy Shi. She is a 7th grader at Harvard Westlake School, and she's working on this project concurrently with another classmate and 2 mentors who are not here with us today. So Lucy.
Hello, everyone. Today I'm here to present the clay anatomy project, which is by me and my friend Angela Leah. I know right now that I'm standing in a room full of biologists and scientists who study organs and cells for a career, but not everyone is like that. Oftentimes, people don't usually want to pursue paths in biology because of their exposure to it, usually in textbooks, which don't exactly have the most pleasant pictures. So our solution is we made a more friendly exposure to the world inside the human body by remodeling it with clay. This gives it a much more homemade feel and makes people more open to pursuing a path in that direction. Our team and progress. Our team consists of Angela Liang and me. We are putting together a book with everything we learned in which we explain the main function of each organ, as well as the diseases it can get and what it would look like with such diseases. So just for right now, currently we have a normal liver, a liver cirrhosis, which is liver scarring from overuse of alcohol, and liver tumor, which often develops after cirrhosis. I think that's like a stage 2 tumor. Okay. In summary, our goal is to make people more open to the wonderful world that is inside the human body in a much more friendly way. And lastly, I'd like to give special thanks to our mentors, IC Liang and Patrick Zeng. They're not here with us, but IC is an undergraduate student at Brown University in Rhode Island, and Patrick Zeng is a senior currently at the University of California at San Diego.
Thank you, Lucy. Yeah. Well, I hope this presentation give everybody a sense how UCLA, how we play clays in the lab. So pretty quick, we're going to present a certification for our students. And please, we have the other 2 students in the audience that don't have a chance to join us. And Lulu, please to join. Please help us, our program, to present the certificate to our student. Yeah, and please go to the middle. Yes. Okay, thank you, our student, and thank you, Lulu. Oh, good photo, please. Okay, Dr. Chu, you want to join us? Okay, so let's enter our actually third sponsored talk, and it's going to be presented by Dr. John Schwartz. Dr. Schwartz is adjunct as a clinical professor of ophthalmology at U.S.A, and he's currently also the founder of iSonics company. And his presentation title is Therapeutic Ultrasound for Glaucoma. Thank you.
Thank you very much for that very nice introduction. It's hard to follow that terrific group of young scientists that we saw here. I'm going to talk about iSonics and its device called TUG, Therapeutic Ultrasound for Glaucoma. And the tagline for TUG is, "We save sight with sound." This is the board of advisors. Each one is very well known within glaucoma and or cataracts or ultrasound. So if I were to ask you about what glaucoma is, first of all, it is the largest cause of irreversible blindness in the world. And what is the definition of glaucoma? If I were to ask everybody here who has glaucoma or knows somebody with glaucoma, probably they would say it's elevated pressure in the eye that leads to a loss of vision. I mean, this is the common kind of statement. However, if you look at the definitions on the website for the American Academy of Ophthalmology, the World Glaucoma Association, the American Glaucoma Society, the Glaucoma Research Foundation, and the National Eye Institute, not one of them uses the word pressure in their definition. And yet all glaucoma treatments that we have today are aimed at decreasing the intraocular pressure. I'm not saying that's wrong, but there's a bit of a mismatch. And the mismatch is there are over 40 different glaucoma medications. There are laser treatments. There are minimally invasive glaucoma surgeries. There are invasive interventional surgery. And every single one of them is designed to lower the intraocular pressure, and yet the very definitions of glaucoma do not even use the word pressure. Why do we follow the intraocular pressure? Well, we follow the stability or changes in the retinal nerve fiber layer, and we alter the intraocular pressure to protect the retinal nerve fiber layer, and lowering the pressure is the only way that we have to treat glaucoma today. But tomorrow it could be better. We follow the pressure because in 1622, exactly 400 years ago, an English ophthalmologist, Dr. Richard Bannister, performed tactile tensions. He felt the eye through the closed lid and found that eye to be hard. And he said, this is glaucoma. From then on, we have been treating glaucoma by lowering the intraocular pressure. We have a number of various methods of measuring the intraocular pressure. But what we're looking at, when we look at the back of the eye, we're looking at the optic nerve and we're looking at the macula. And here, this is the innermost layer of the retina called the retinal nerve fiber layer, which gathers together and then takes a 90-degree turn, and it forms the optic nerve. And we're interested in this area right where it takes that 90-degree turn, and we measure the retinal nerve fiber layer at that point. So the imaging shows the optic nerve, and measurements of this particular layer, and a decrease in the retinal nerve fiber layer represents a worsening of glaucoma. The literature shows there's about a 3.8% loss per year in our patients who have glaucoma. And these are some images of the same eye, the left eye, in a patient taken 13 months apart. And you can see here the retinal nerve fiber layer is 0.160. Here the retinal nerve fiber layer is 0.021. You can see the significant difference in the retinal nerve fiber layer 13 months apart in this particular patient. This is slide number 13, and I'm going to get back to this. But first, I have to talk to you a little bit about glaucoma in terms of the intraocular pressure. The pressure is within the eye. The pressure within the eye is formed by the production of aqueous humor back in the ciliary process that comes forward and then leaves through what's called the trabecular meshwork and then out what's called the canal of Schlemm. This is the production and the exit of the fluid inside the eye. And it's a blockage felt to be primarily within the trabecular meshwork where the pressure is elevated. I'm going to take a little bit of a detour, but I'll get back to what I was talking about. Cataract surgery was performed up into the 1990s by making a very large incision in the cornea and then taking the lens out in one piece. This was cataract surgery up until about 1990, 1992. But in the 1990s, we began to see that there was a decrease in intraocular pressure in those patients who had cataract surgery. We didn't see this before. when we were taking out the lens in one piece. As a matter of fact, the reports indicated a slight increase in the pressure up until the 1990s. Phacoemulsification became the method adopted for cataract surgery in the 1990s. Phacoemulsification uses ultrasound in order to emulsify and break up the center core of the lens as we start the cataract surgery. So does ultrasound potentially have a beneficial effect on glaucoma or help to decrease the intraocular pressure? Well, ultrasound has a couple of specific properties that are beneficial. One is the sonomechanical or vibrational movement, and the other is hyperthermia. Hyperthermia can trigger certain cytokine cascades that potentially are analogous to a treatment with laser called SLT. SLT is a laser treatment that's aimed at the trabecular meshwork, and even though it looks in this particular diagram that you're cutting a hole through the trabecular meshwork, you're really inducing an inflammatory response that triggers certain cytokines. So the laser treatment for glaucoma triggers some beneficial cytokine activity. Phacoemulsification triggers a similar cytokine cascade. So the micro stretch that's occurring within the trabecular meshwork triggers a cytokine cascade very similar to SLT laser. So there was a paper that specifically postulated that it was this use of ultrasound inside the eye that led to a decrease in intraocular pressure. So if it's possible to develop an ultrasound specifically aimed at treating the intraocular pressure, we have to establish first the frequency and then the power. The frequency was fairly well established by the use of ultrasound inside the eye for cataract surgery at 50 kilohertz. The power is limited by the thermal effect that occurs. Greater than 45 degrees centigrade leads to cell death and pain. But between 40 degrees and 45 degrees centigrade, we have that additional benefit of a cascade of thermal-induced cytokines. So here, this was the work that I did in order to determine exactly what the power should be. I needed to have a raise in temperature of 7 degrees from baseline to level out and become a plateau. And it was between 3 and 4 watts of power to the transducer that accomplished this. This is the prototype. I have one of the prototypes in the back of the room. And a couple of studies were done with this in vivo. The first one was a pig study in order to determine the proper dosage. The second was a rabbit study in order to do histopathology verification as its non-significant risk for its use. So the device is aimed at the trabecular meshwork. It's held against the eye, and there is an energy that is localized within that anterior segment in the trabecular meshwork and the canalic limb. 4 clinical studies were done as a non-significant risk. These studies were performed in the United States. The first one was the blind eyes patients who were recruited from the Braille Institute, had no hope of any vision to see if it was safe. The second one was to determine exactly what the proper parameters were. A third one was a 2-branch study which was published, and the fourth was to see the additional effect by adding the treatment to patients who were on pharmaceutical agents. In 2019, I went to India in order to deliver a device to be used at that point. So over time, since the original studies, safety and efficacy has been shown in over 10 years of follow-up and close to 1,000 post-treatment examinations. This was the first patient that I did bilateral treatment. Most of the patients had just a single treatment. Here, the blue line is his right eye. The orange line is the left eye. And you can see, whereas I just treated the blue eye, the right eye, which was blue, there's a bilateral effect in terms of intraocular pressure that occurred. After 7 months, I treated the left eye, And this goes out for 45 months after that second treatment with a decrease in intraocular pressure of 38%. It tends to last in the total number of patients treated at least 6 months, 84% at least 6 months, and the majority lasting for a year, comparable to their pharmaceutical treatment before. So the treatment is using the device as I indicated before. It has certain lights that show whether it's been activated and tuned properly. Step on the foot pedal and it emits the ultrasound. It's held against the eye. It's held against the eye in a particular location at 12 different spots around the circumference of the cornea, which is the limbus, for 45 seconds at each spot. So the market for glaucoma, it's a huge market in terms of the United States. There are 3 million people. In the world, there are 70 million people. The number of providers who can treat glaucoma, 64,000 potentially in the United States and 450,000 around the world. So I have a device that is comfortable, it's repeatable, it eliminates a lot of problems with compliance, with allergy, with irritation, but it is simply another treatment to lower the intraocular pressure. Then the future hits. And there are recent articles that show that the red optic nerve, which has had a crush injury, is able to be regenerated by the use of mTOR, the mammalian target of rapamycin, and high-intensity visual stimuli. If it's possible to use ultrasound to trigger that same type of mTOR result, then it's possible that we may have an ultrasound that can trigger regeneration of the optic nerve. I did some work in Oregon, and I found that my TUG device actually, which is a low-power ultrasound, triggered TNF-alpha, which helped to lead to nerve regeneration. Not only that, if we look at the literature, we find that low-power ultrasound, similar if not identical to what I have, is able to trigger a whole host of very specific biochemicals that each and every one is able to lead to the potential of neuroregeneration. My people in India who were treating patients since 2019 sent me some images of patients that were treated. And at that point, we could see that there was an increase in the retinal nerve fiber layer. You may have remembered that I showed you this slide before, that 13 months difference between the left eye and the right eye, showing that significant difference. It turns out that this was flipped, because if you look at the dates, the actual treatment was a treatment between the left eye and the right eye, where the retinal nerve fiber layer, which was 0.021 prior to the treatment, ended up 0.160. This was the next patient. No, this is the same patient. So there was a significant increase in the retinal nerve fiber layer with treatment. Another patient, the same kind of thing, an increase in the retinal nerve fiber layer. And then when I went over all of the patients that I had treated, going back to the time that I had treated them back in 2009, if you look at the blue line, each patient had only one eye treated. The blue line was the patient's eye that was treated. The orange line was the eye that was non-treated. A very significant difference between the treated eye and the non-treated eye in terms of retinal nerve fiber. There were 2 or 3 that were rather significant, and these, it turned out, had multiple treatments within a year. All the others had just 1 year. This is statistical analysis of the comparison of the treated eye versus the untreated eye in those patients that only had one eye treated during that year.
I'm sorry, Dr. Schwartz. We might have to wrap up within a minute because we have a venture pitch session coming along.
All right.
I'm sorry.
This is my last slide.
Oh, sorry about that.
So what does it mean that the retinal nerve fiber layer is increasing? It means everything. So the TUG treatment not only decreases the pressure, the data and images show that the nerve fiber layer is increased. All other treatments for glaucoma only chase the intraocular pressure. And if we can stop the loss of the retinal nerve fiber layer, we have stopped glaucoma. Thank you.
So we will quickly advance to the next sponsor presentation by Dr. Julia Xu. And Dr. Julia Xu's title is New Era of Microwave Thermography for Breast Cancer Screening.
Transmit right. Oh, okay. Okay. This back, this back, this is yeah, okay. And like okay. All right, good. Okay. Thank you. Ladies and gentlemen, yeah. Because of the time strain, I have only 15 minutes or to speak about half hours material. So I just move a little bit of the my sequence. Because toward the end, I try to show you what's the accomplishment for our company's last 5 years R&D. So show this first, then I come back to tell what's the theory, what's the concept, and what's the technology we use to accomplish this product for this system. Okay. 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 The company we have the most we design called radiometer scanners or maybe radiometric scanners, microwave. Okay. Well, this is actually just receiver microwave radiation from the body. Okay, so this is very, very compact and affordable. So we already use this in the 2 pilot study in Taiwan, in Tri-Service hospitals and on my care memorial hospitals. And another thing we needed is just the template. Okay, that's the 2 systems we needed to complete the screening of breast cancers. And what else is actually there's a lot of software behind this. We collect the data, some of data from the skin, from the body, and we use a Bluetooth teeth passed to use tablet. Okay, and tablet we also collect the data through preliminary data processing, and we send this through the Wi-Fi or internet to the cloud. Okay, so when the cloud we use the download to our research center here in Torrance, and like we did a clinical trial in Taiwan. Once the major finished or measurement the patient, we already know the data, so to process data. So this is kind of build up a kind of telemedicine concept here. I'll just not develop this devices. Okay, so and we have this one here has been through the Taiwan FDA clearance for the class 1 breast cancer screening devices. Okay, that's the accomplishment so far for the last 5 years R&D effort. Okay, so that's it. All right, and then we start the topics of today. I think thermography, maybe some people not use, we're familiar with that, but I think even I found out even in medical area. Okay, so I just want to explain first what this means and how does it come from, its origin. Okay. All right, so then how is this gonna, okay. The first one I'll show you is this is some kind of, I suppose 3 parts. Introduce what's the thermography is, Now, second one is the second part I will tell you was the beneficiary of futures when you use microwave, the sonographic modalities. There are some better features than the current cancer screening devices. And third one, actually, I almost covered the third part, so I can spend a little bit more time to explain the theory and the concept here. All right. Actually, I think in the medical science, it's called thermology. Okay, thermology is, the thermology is something related to thermal heat. Okay, back to BC, 400 BC, the father of the medicine called Hippocrates, a Greek physician. He's quoted that way. In whatever part of the body, the excess of heat or cold is generated or felt, the disease is there to be discovered. This is what's quoted 2,000 years ago. I want you to know this very quickly. Is generated or felt. Okay, because we heard it as a silent killer, right? That means you don't feel any symptoms, right? Until you find it, it's too late. Okay, but it's generated a long time ago. All right, so this is from this thermology, actually from sonography from here, then go through the about the 18th century. I want to go through this just to go to the kind of very primitive one back to golden state, golden age. They use those primitive physician doctors. When somebody's sick, they put that smaller on the screen. And after they take out which part is dry or fast, then they think, okay, there's some heat over there. Okay, that's very, very primitive. Basically, we try to do the same thing, but we use modern technologies. All right. Then after the, I think since that in the 1980s, the lots of devices try to develop to measure the temperature of the body. Okay, that's another development. Now, up to about World War II, a lot of what we call the 1950s, that's overall called modern tomography. The difference between thermology and just you get the data measurement and use some data image processing. Use a visualization. That's the difference. It's the opposite thing. So the modern tomography is the two primitive one. One is infrared. Okay, infrared. Then the microwave is like a newcomer in the last 10 years, or last decades. All right. So I will kind of like focus later on in that modern one, and then go to the microwave tomography. Okay, so up to this you can see, I said some of those I think you can see here the thermology that it used a lot of many branches of medicine, like neurology, sports, and then the medicine open the breast oncology. So they already using that one. But so far, the most uses, they use the infrared. Okay, infrared is for breast cancer. It's also get a kind of FDA. I think like a computer certification back to 1982. But the reason they didn't so popular because is the sensitivity and the specificity is so low. It's about 50%. So it looks like the FDA doesn't accept that as kind of common things. And the one we use this one with better our sensitivity, everything is better than infrared. That's another advantage of these devices. Okay, you can see those. This is our infrared things. You can see that those one you can see from the COVID-19 they use a lot of those things. That's infrared. Okay, so what infrared measures the temperature? I will it's just skin temperature. Okay. The infrared measures the skin temperature. Okay, now I will tell you why. Okay, the difference between the infrared and microwave. Okay, I put the wrong part of it. All right, so when I say 100 is the modern tomography, this is the two basic one I mentioned, that infrared and the microwave on the bottom. Okay, so I don't want to go to this chronological development. Just so they are currently talking about it. Even start at the same time, and now still going on, All right. When you talk about sonography, let me compare with what we compare with the mammography. One question, mammography, and this the X-ray thing. Okay. I would say that different. The most different from you can see both sides, you can read it. But I'm going to focus one is on the thermogram. One is the early detections. Okay, I will show you a chart about the breast cancer life cycles. All right. Then the reason I say early, okay, I hope I touched that data, but I explain it a bit here. Which one? Second one. The early detection. This is what we emphasize, because you look at the let me see. Oh, sorry. Okay, the chart. This chart is back to when we start on the left-hand side of the mutation. When the cancer mutated from a normal health to a cancer cell, they start the temperature start growing in cancer cells because they need more active than metabolism activities. So you can see from the 0 to 8 years, there is no tumor, no density. So, but during this time, the cancer cells temperature already increases. Okay, so that's why we call it preclinical phase. Okay, the sonography we come back to temperature and sonography, and when you go to the theory, I can say what they are related. So during that first 8 years, okay, the current classic, okay, I call it traditional breast cancer screening methodology cannot do anything. They cannot wait until 7 or 8 years. The tumor is still forming, then they can take the picture, the images. Before that, they can do nothing. Okay, but sonography can do that. Okay, if your device is good enough, your software is good enough, your model is good enough to predict it, then you can, within the same 8 years, can detect the breast cancer. So that's the one very advantage to use the sonography, okay, sonography things. Then that's the one when I compare the okay, sorry, I just have to go back this is because the time is out of script. Okay, now we come back forward to this one now. Okay, so that's one I want to compare. So there's a lot of advantage of sonography. Sonogram, okay, I have minutes. Okay. Then let me see another thing of advantage of the. Okay, this is the X-ray mammograms. You can see that white area is different right hand side. That's called dense breast. Some women, about 40%, particularly young woman, has a dense breast. So when dense breast, the X-ray, what you look at is a white area. Similarly, if you have some tumor, showing that in the left hand side, let's stop. Why start there? So if you want this one, you can see if that tumor highlight the entire like area, you cannot differentiate it. That's what the one deficiency of X-ray. Okay, sonogram can mitigate this problem. Okay, that's the one advantage for this. All right, why? I told already the infrared has been used for same purpose, but why we need microwave? Well, this is the kind of curve. The vertical is kind of the depth of penetration. How deep this measurement can go into, and the one is frequency. Okay, so in that right hand side, that's microwave. All infrared is far away to the right here. So the reason of this is because the microwave can make the depth penetration stronger than infrared. So microwave can measure the 3D data. That's very important. Infrared so far is only can measure 2D data. Okay, so this is 2D, 3D one is very, very superior purpose for the microwave versus the infrared. Okay, so that's why we need some graph and how it works. I think I just get 3 basic theory, and then we can very quickly run it out in one time. Okay, 3 principles. One is we use our quantum physics to break bodies here.
I'm sorry, Dr. Xu.
How many?
Yes.
How many?
Your time already up.
Oh, okay.
Yeah.
All right. Okay. Now I just take this principle, then I got
Thank you.
Okay. Yeah. This is a quantum basically called break body theory. Okay. Frank. And this shows. Okay. We don't read those equations. So what I want to show you this is one very important of this one is microwave and the heat temperature is both sides of a cone. That's very important. Okay. Here it says that if any material above -273 Celsius, there will be thermal radiation. Every body and object here is above the temperature, even our body, human body. So this theory is very important. If you have heat, you have thermal radiation. Okay, that's important. So that means you have high temperature in your body, inside your breast or whatever, you have thermal radiation coming out. So that's what we use our to catch. Okay, catch. That's one and theoretical backgrounds. And the second one is the okay. This is from pathologists. Pathology, I told you on the left hand side, that is, if you lose a tumor, not tumor, the cancer cell, if you get cancer cells, then they call angiogenesis. So they turn angiogenesis. They grow a lot of small vessels, blood vessels. If you have blood vessels, the temperature there will increase. Okay, this is a medical theory. Okay, so that's the another reason we use that. Then you look at this. Here, this one, that tumor cancer cell right here. There are different ways to radiate back to the surface, and actually, the thermograph I told you that the microwave thermal can measure 3D. Okay, then make it a deep. Okay, this is deep. That depends on frequency. It shows you kind of how deep they can measure those temperature. Okay, that's the two. The third one is that we have this technology. We use it. Okay, that's the one I just showed you here. We use a microwave radiometer. Actually, we can in order with the microwave receiver. Okay, this is different from the telecommunication purpose. This is special from sensing purpose, from the remote sensing. So if this device can measure the heat source down here, down the scale, then you can catch which area has the problem. Okay. So that's just the 3 basic theory of concept behind our problem. Yeah, that's all. Thank you. Next question.
Thank you, Dr. Xu. So we're going to stop this sponsor session and quickly move to our venture pitch session. And if you don't, yes, Dr. Xu.
I just say last quote. Yeah, I heard the last of U.S.A. students here. Actually, in our team, we also 5 years before about 3-4 U.S.A. graduates and 2 U.S.A. graduates, all the local graduates as well. So we 10 young, very we a couple very experienced people. So we spent over 4 years to come out with this result, this accomplishment. So I can kind of U.S.A. and this is students very good, already. Yeah.
Thank you, Dr. Xu. Thank you again.
Okay.
So our staff is going to set up the venture pitch session. I think now I will have Nick come to the front and have our judge start to get seated. Thank you. And audience are welcome to stay to join this, to watch this venture pitch session. Thank you.
Okay, 10 minutes break and be right back. The most exciting part of the conference will start in 10 minutes. Hello? Okay, you have 1 minute. Please get into the seat. We're gonna start the contest. Okay, everyone, find a seat.
Hello, hi everyone. So our next program is about to start. The next program is our pitch competition. Hi everyone, I'm the host for the pitch competition. I'm Nick Wu. I'm currently a staff engineer at Rivian Automotive and also an active angel investor. So our first pitch competition at the first SoCal Healthcare Innovation Symposium is sponsored by Mount Jade Global Foundation for the winning awards of $15,000. And this session is, of course, is hosted by Southern California Mount Jade Science and Technology Association. And we are in collaboration with NARL, the National Applied Research Laboratory in Taiwan. So now I would like to quickly introduce our honorary pitch judges. Our first judge and the lead judge, and also the host of the pitch competition, Dr. Wenda Chu. Dr. Chu is the co-CEO of AHMC Healthcare. He was the former Minister of Health and Welfare, Taiwan, former President, Taipei Medical University, former Superintendent, Taipei Medical University Songhe Hospital. Dr. Chu is the world-renowned researcher specialized in TBI, traumatic brain injury research, and he previously held professorship in multiple research institutes and universities. He holds an MD from Zhongshan Medical College, a PhD and a master's degree in public health from University of Pittsburgh, and a doctor of medical science in neuroscience from Nihon University School of Medicine. Let's give applause for Dr. Chu. And thank you. And our second judge, our honorary judge, and also the host of the pitch competition, Howard Ko. Howard is a partner with Mobius Ventures, where he focuses on robotics, healthcare, insurtech, fintech, and proptech investments. He served as a board of directors and board observers at multiple companies. Howard was previously with Mission Ventures, where he focused on investment in software, wireless technologies, and semiconductors. Prior to Mission, he was with Solomon Smith Bernick in their technology investment banking group, where he was involved in over $3 billion public and private financings and MMAs. Howard also has served as a co-chair of the San Diego Telecom Council Wireless Infrastructure Group. He holds a bachelor's degree in electrical engineering and an MBA from U.S.A. And let's give applause for Howard. Lastly, our third honorary judges, Christopher Young. Chris is the VP of Engineering with Away, where he leads all engineering functions for the $1.4 billion D2C lifestyle brand. He was previously with Corporate Travel Management as an engineering VP, where he oversaw engineering, QA, and PM groups to build solutions that support over $1 billion business in North America. Previously, he held senior management roles at DirecTV and Ticketmaster. In addition to his career, Chris is also an executive board member of Tech Cost Angels, the most prestigious and active angel network in the US. Chris is also a member of Forbes Technology Council, where he contributes to Forbes.com on topics related to tech and entrepreneurship. Chris holds a bachelor's degree and a master's degree in computer science, a master's degree in industrial and system engineering, and an MBA from U.S.A. All the degrees from U.S.A. Welcome, Chris. Okay. Let's again have a very nice round of applause for our honorary judges. And let's welcome our lead judge, Dr. Chu, for an opening remark for our pitch competition. Welcome, Dr. Chu.
Like Steve Jobs once said, innovation distinguishes a leader and a follower. We should embrace innovation as an opportunity, not a threat. We appreciate the many innovative teams that have joined us for the venture pitch. We found that all of the teams are incredibly creative and forward-thinking, and that their ideas and products has great potential to have application in the real medical field. Thank you again for joining us. Thank you.
Yeah, thank you, Dr. Chu, for a short opening remark. And again, this is our pitch competition. And let me briefly explain our rules here. So today we will have 7 companies that present their idea or business proposal in front of our audience, and they will have 7 minutes to present precisely what they want to deliver, the message. And then after the 7-minute pitch presentation, they will have another 8 minutes with the opportunity to get feedback and Q&A sessions with our honorary judges. So today, again, that's our rules, and we are very strict on the timing. So here, I will remind the pitch team about their timing by ring the bell here. Yep. So this is the timing reminder for the pitch team. When they have 2 minutes, I will ring the bell twice. And when they have 1 minute, I will ring the bell once. And then when the time is up, I will intervene, speak up and say time's up. And then I will give the microphone to the judges for the Q&A. Okay, so I think we are almost ready for our first team to come on the stage. And again, also, as everyone knows, now we are kind of in a hybrid era. We have teams from Taiwan. Now it's like 7 a.m. They are going to pitch virtually. And we also have teams in person. They will pitch in person. So I think now it's a time we can give the first team. The first team who is going to pitch is AHMC Biosciences. Let's welcome the first team to the stage.
Thank you very much. I'm Mingyi Huang from AHMC Biosciences. It's an honor, a great honor to be here to present the AHMC story. This is a story about revolutionizing cancer care with personalized oncology on AHMC's vascularized tumor chip platform. And let me get you the PowerPoint up. Just bear with me on putting it up. So what you're looking at here is the core of the tumor chip for AHMC's vascularized tumor chip platform. Red represents living human blood vessels, and green represents tumor cells. Cancer is personal, but treatment is not. Many of you know personally friends, families, loved ones that have suffered cancer. Many of them have to go through several treatments before finding the right one that works, wasting time, money, and unnecessary sufferings. There are about 20 million new cancer patients each year, and they need a better solution. So for example, right now, even though cancer hits all genders, all races, and all ages, but you get the same standard FOLFOX treatment if you get colon cancer, just like everybody else. The solution is AHMC's oncology platform. We test your tumor on AHMC's personal tumor chip and identify which drugs will work best for you. Just imagine every cancer patient should get a personal tumor chip before a treatment. What is AHMC's tumor chip? Our CARIS tumor chip recreates a patient-specific tumor environment, so tumor drug testing is much more accurate. In human body, the drugs are delivered through blood vessels, through the artery, the capillaries, and venule for the waist. The same thing on our CARIS tumor chip. We deliver drugs through blood vessels, and living human blood vessels are why our CARIS tumor chip works better than 2D spheroids or mice animal models. We're the world's leader in vascularized tumor chips, used by 7 of the world's top 10 pharma companies. Why better? Because tumor and tissue microenvironments are very complex. You need new 3D systems to properly model cell behavior. As you can see here, we have successfully modeled many different types of tumor cells, such as slow-growing to fast-growing tumor cells. And the patient-derived tumors are also well modeled. More importantly, we already have independent validation of the reproducibility by a tissue chip testing center funded by NIH. This is the ARICARI difference. It's living human blood vessels with superior predictability of in vivo responses. Here's how the personalized oncology platform works with our tumor chip. Step 1, take the patient biopsy. Second, vascularize the tumor chip platform. Third, grow the patient-specific tumor cells in multiple devices in our tumor chip platform. 4, we test the drugs for responses in our tumor chip platform. And 5, we actually select the drugs based on the tumor chip readouts and also the mutational analysis from our CARIS data bank. Then we recommend the treatment to the patient and the clinicians. What's more powerful is our data bank, because we combine the tumor chip readouts with the sequencing and clinical outcomes to build a predictive database. What's more powerful is that we can test 30 FDA-approved drugs at the same time, compare that to the cancer treatments patients receives, and compare that to the sequencing data, dramatically improve the drug efficacy prediction. We already have 3 clinical trials underway with promising results from our first trial. There are 3 studies. One is on colon cancer, second one is on lung cancer, and third one is on nasty PC cancer cells. The results, as we said, initial results are very promising. Unfortunately, patient A expired as predicted by our platform. We could have saved some time for that patient. Maybe we could have tried different drugs that's shown efficacy in our platform, galenicidip particularly. We have a very strong IP portfolio and it's growing. We have 2 already approved and 2 more coming, including a pressure regulator, multi-layered systems for a larger platform and multi-organ system. 4th one is also pending, which is for vasoactive testing. It's very powerful for hypertension drug testing. How big is the market? By one estimate, personalized oncology market can be as big as $272 billion in 5 years in 2027. We think that's conservative because there are 1.9 million new cancer patients alone just in the US each year. Imagine each of those patients gets a personal tumor chip before they get a treatment. How big is the competition? There are 2 categories. We believe our CARIS vascular tumor chips are much more powerful predictive than spheroids, such as Xylus or Kiatic, and more personal than database-driven kind of treatment, such as Tempus and what Illumina provides. We have a plan. We have a development plan in 4 phases to market launch. The first one is platform validation with academic collaboration. Here we already have, let me see if I can get the marker up. We already have, as I said, the 3 clinical trials with the UC Cancer Center in Irvine. We also have commercial validations through commercial customers by 7 of 10 of the world's top pharma companies. We also grant recipients from National Science Foundation and also NIH. We're a phase 2 SBR grant recipient. We're in the phase 2 right now, actually, for technology transfer. We are already in process of the platform optimization. We're going to develop hospital partners. So we're looking for partners. If anyone in the audience has some good suggestions. And this is really to develop our own commercial IRBs and our AHMC clinical studies. Initially, we're thinking of 25 cancer patients. The next phase would be to really establish regulatory strategy as well as reimbursement strategy. As you know, today, any genomic testing mostly are actually paid by the insurance. It's a standard. We think we can get there as well with the right talents and expertise to support us. And ultimately in phase 3, we want to establish genomic partners for that expanding data bank, establish IRBs and also interventional clinical studies, and ready for the market launch and FDA submission. We have a highly experienced team in the field. We have 3 very world-class scientific founders, Dr. Hughes, Dr. George, and Dr. Abe Lee. They're world-class vascular biologists as well as bioengineers, all from the University of California faculty. On the right-hand side, you see the management team. Dr. Hatfield is a serial entrepreneur with many biotech startups, and myself have 40 years of experience with Fortune 50 companies on finance, risk analysis, and also business development. What's not shown here is we have a first-in-class team of 7 scientists and also technicians in our lab in in Irvine, California. To sum up, patients are waiting, and we're ready with personalized oncology on our AHMC vascularized tumor chip platform. Thank you very much.
Thank you. Okay, so I think it's time for our judges to provide feedbacks or Q&A with the company. Here is the microphone to judges. Dr. Chu.
Thank you. Very good presentation.
Thank you.
Is there any similar product in the market? And have you compared with other product?
Yes, there's several. As I said, there are 2 categories of competitions or comparable ones. Of course, we don't think we're the same. And the first one is actually, I mentioned 2 of them, ICAIH. They're active testing for personalized oncology. Xylus is also saying that they're in the personalized oncology space, but they're all based on spheroid technology. As you know, spheroids are not does not have the same complex microenvironment as the vascularized organoids that we have today. So maybe some simple signals you'll get, but how predictive is that? We question that because, as you know, animal models that are commonly used today and why it's not successful. Actually, only 10% or less are successful, even though everyone's using it. That doesn't mean it works. So we believe our vascularized tumor chip model is much more predictive because we recreate the complex tumor microenvironment just like in human, delivering drugs through blood vessels, delivering drugs, either small molecules or immune cell therapies. We can do that, and we have data for that as well.
Quick question regarding whether the model or the tumor chip is cancer specific, whether or not you need to build separate models or separate tumor chips for different cancers, whether it's lung cancer or anything like that. Is there a separate, do you have to develop separate kind of tumor chips for each of those different cancers?
Very good question. I think the answer is really no, because what's probably not clear is our technology is very biologically driven. So you might have heard of 3D printing model, scaffold model. Ours actually, because of our world-class expertise in biology, especially vascular biology, we pretty much in simplicity, we put the cells together in the happy medium. They just self-organize into a network of blood vessels. And tumor initially will survive in the suspension of the matrix in our platform, the tumor chip. But once the blood vessel network form around the tumor cells, then it just starts acting like in vivo, where the blood vessels actually can feed the nutrients and at the end, the drugs to actually to grow the tumor. So tumor actually grow much faster. So to answer your question, we also adjust the matrix. We grow the phenotype closer to the organ-specific kind of characteristics. So it's not like we have a liver model, we have a breast model. It's actually based on the same technology. It's the magic's really in the biology piece that we were able to direct the endothelial cells to grow the characteristics closer to the organ system.
Hi, great presentation. First of all, thank you.
Thank
I
you.
have two general questions.
Sure.
One is specific to your model prediction accuracy in terms of how well you're mapping the drug you're predicting to the recommendation. Do you mind sharing some more quantitative matrix around that? The second part is I'll give my question so you can understand maybe in two parts. The second part is more related to your current burn rate, how much capital you raised, and how you are thinking in terms of using the next round of capital. Thank you.
Yes, we think very big. To answer your second question first, we think big. I think to market launch, we probably need $100 million, but we also break it down in digestible pieces. We think for the second phase I mentioned, which is the technology transfer, building our own initial commercial trial, we're looking for about $5 million in funding. And the third phase, we need about $25 million, and the last phase is about $75 million. But I think to really successful launch in the market is $100 million, but initially we think $5 million will get us a quality commercial, FDA grade kind of data to really validation. The first question, can you repeat that question again?
How accurately are you predicting?
Yes. We actually have several publications to show the drug efficacy prediction on tumor cell lines. So that's very well known already. We published several papers from the academic lab. What's more important is that we are going through the clinical trials now. As I said, we have 3 trials ongoing right now with the academic lab collaboration at U.S.A. Cancer Center. And the initial result, the correlation study is very, very promising. As I said, patient A shows parallel correlated results, and we're just waiting for more result there to really validate the data further. But in terms of the foundation of the science, in terms of technology of showing the efficacy of drugs on tumor cell lines, we already have publications supporting that. Did I answer your question?
I think to that regard, I've seen, I think different companies trying to basically map out better pathways or care pathways for kind of for drug indications. How have you, what size of data set have you guys looked at, whether it's, 5, 10, 20, 30 kind of correlative kind of pathways that you've mapped, where it seems like you already had those 1, 2, and 3 kind of studies. Have you gone beyond kind of that, where you've basically tested out, hey, look, you have 10 patients, 20 patients, whatever the case may be, and they went through, a certain type of kind of care pathway from a drug indication standpoint, and you actually predicted a different pathway, and they would have potentially, let's say, bounced back to your care pathway at some point later on. Have you guys looked at that yet, or is it still fairly early, just saying, look, we would have picked pathway A versus pathway B, and a current, let's say, physician, chose pathway A, and it was not productive?
Yeah, you're saying exactly what we're trying to do with our AHMC commercial clinical trial design. What we have, as I said, we have 3 clinical studies ongoing with U.S.A. Cancer Center. We're testing 3 different kinds of cancer types. And that's exactly what we're trying to show in terms more of a correlation study rather than interventional. That's, I think, interventional is more phase 3 that requires much more resources. The reason why we're not there yet, just it's more of funding. We're bootstrapping as much as we can with what we have. What we do have shown is actually a lot of very, very specific readouts from our platform. What you can see, actually, our platform is actually very transparent, easy to use. So we can do a lot of readouts in terms of just measuring whether the drugs or the immune cell therapy can actually extravasate the blood vessel into the target tumor lines, for example. We can actually just simply observe the tumor cell growth over time. And we also, more importantly, a lot of our pharma customers love us is actually to see the pharmacology and mechanism of how the drugs actually get in and out or not getting in and out and what it's targeting in the tumor microenvironment. So all those things will already have metrics measured. So those are actually a solid foundation for us to design the interventional studies in phase 3. I think that's what you're looking for in your questions. But phase 2 and also phase 1, the studies more parallel clinical studies where we actually just compare in blind testing what patient outcomes are versus what our outcomes are. The only exception I would say is probably the third trial. You saw that's a nasty PC cancer where there are basically no treatments. And because the physicians that are really a strong advocate for our platform, they believe in our technology, I think she may try actually to use the predictive results from our platform to somehow incorporate into the treatment protocol that when she sees the results.
Okay, our time is up. And yeah, thank you, our Corey Biosciences for the presentation.
Thank you very much for your time.
Yeah, thank you for our first team. And I want to again explain a little bit. So I think we had some initial technical issue. So again, for the next and the following presenters, please just listen carefully when you present. I will ring the bell twice when the time is up, close, 2 minutes left. And when the time is only 1 minute left, I will ring the bell once. And then when the time is up, I will speak to intervene your presentation and say, time is up, and then we will transition the microphone to the judges. Okay, so I think we are pretty good on time, and I think we are almost ready for our next company. So let's welcome Jello-X Biotech for to present to our judges and the audience. Thank you.
Hello. Hey. I'm sorry. I'm sorry. The room crashed now.
Can you still hear us?
Yeah, I apologize for the issue of the computer's problem. I may need to reboot the computer again. I'm sorry. Yeah. So can I exchange the order with others?
Yes. So we do have some buffer time, but I don't want to give.
Oh, okay. I will reboot my
So
computer
if we
right
can
now.
restart as soon as possible. We do have some like 30 minutes. Sorry, 30 seconds actually. So
Okay.
30 minutes is the entire session. Sorry about that. So are you ready to present now? Okay. Yeah, he will call in. He tried to restart his laptop. Yeah. Yeah, so it's the tough time, right? So I think everyone has some sort of experience when you try to call in virtually, like communicating with your vendors, suppliers in Asia. Bandwidth and the time zone difference is always challenges, and not to mention the technical issues with virtual meeting app. So I do start the time. So whenever the presenter return to the virtual stage, we will deduct his total presentation time from the time, whatever time he left. Yeah. Okay, so I think before the presenter like comes back, we can have some interaction with our judges, just to. Okay, so I'm throwing the microphone to the judges. I mean, we can quickly talk about what the judges are looking for, right? Because our judges, they engineering background, and not to mention Dr. Chu, the medical background. I think all our judges are looking for different things. And I think our presenter is back. And are you ready to present? Or do you want to just talk? If you cannot present, you can talk. It's fine.
Okay. Yeah, I hope I can show my. Okay, yeah. There is some problem of my happen. I just tested and okay. Without slide. Yeah. I passed my my slide to your computer and just help me to show the slide.
Oh, so why, right? So do you want to
yeah,
quickly introduce?
I just passed the PDF file. Yeah.
Because we have 4 minutes left. I think we might want to start with a brief intro of your company. And once the slide is up and ready from our control station to present, we can share screen. But I think we should keep the time and try to best use the time.
Okay, no problem. Yeah, I will show it. I will try to. Actually, our company is focused on the pathology diagnosis. Now we call it the next generation pathology. Why we call it the next generation pathology? Because we challenge. We are bringing a new technology to challenge a technology already used over more than 1 century. Yeah, in the past, H&E staining already used more than 1 century. However, there is no new technology to help doctor helps scientists to investigate the tissue imaging. So it will be challenged to bring the most detailed information to the clinical study or clinical diagnosis. Yeah, so we will bring the one this technology. And what's the benefit for the oil industry is that our our technology can help each new for new drug clinical trial save 100 millions per trial. Help our technology can help their market more than 1 billion per year. So that means that accepted by several large pharma, big pharma. For example, Roche and AstraZeneca already collaborate with us, and we deliver our the most updated next generation pathology technology to them and help them to expand their market and help them to shorten and save budgets of the clinical trial schedule and the clinical trial schedule and the budgets. Yeah. By the way, we also plan to finish our FDA certifications in the 2023, and we already finalizing all the documents in the January. Yeah. And in the end, our pipelines include lung cancer, prostate cancer, breast cancer, oral cancer, and the colon cancer. Yeah, so some of validated data already published by a prestigious journal. For example, accumulate impact factor is more than 20. Yeah, so that means that we also trying to applying medical order, collaborate with our partner, the medical center in Taiwan, in North Taiwan. Yeah. And our team is interdisciplinary team. So our background include electrical engineering, chemistry engineering, and biochemistry engineering. Yeah. So this is a new challenge. We will bring the new brings new supply chain, the new industry to the biotech field. Yeah, I'm sorry for the technical issue. Yeah, because I just testing my slide in the waiting room. Yeah, everything goes well, but I'm not sure. Yeah, maybe the pitching room, my computer have some hesitate. Yeah, so our consultants include medical KOL, include Dr. Yan Zicheng and Dr. Zhou Deying. And our technology consultant also include academics from Academia Silica, Professor Zhang Anshi. And we also have a very experienced medical offer. Yeah. Okay. Yeah, thank you. I'm sorry for that. If everyone wants to know progress, please feel free to contact yyLin@jellox.com. I can show you our company's progress in person. Thanks.
Thank you. So I'm going to give the microphone to the judges for Q&A. Yep.
Okay, no problem.
I still don't know what's the major impact for healthcare in your next generation pathology. This is first question. Second question is compared to the 4 benchmark companies listed in your file, what's your strengths and opportunity? Thank you.
Okay. The major impact for the next generation pathology is that the currently pathology technology biopsy just a little bit sample. So it will be very challenging to organize the whole information together. Yeah. So you may use that biopsy to doing the next generation sequencing, but you don't know which tissue you use for mutations testing. Yeah. In our method, we can understand we can record the tissue structure firstly. After that, we can reuse our tissue to doing all of the others examinations. Yeah. So that's the major impact, because we can collect all of the diagnosis information with known tissue structure. Before that, we can know that. Is that what's the difference between us and our competitor? The most of our competitor is stay in the 2D pathology. That means that the original of their slide producing method already existing more than 100 years. So that's quite old technology. So people cannot understand the 3D information. So our advantage is that we bring the most advantage tissue imaging technologies and help doctors and the researchers to understand the 3D tissues information. So that's why we say that we can help pharmaceutical company to shrink their trial schedule and increase their available market size, because we provide more information to help them to do more judgment. Yeah, so that's the major difference. The second part is that our technology will collect new data. The new data means that the new data can help people to understand what is the best, what is the most appropriate treatment plan for patients. Yeah, so that's also very important things. Yeah, because the currently 2D pathology, everyone owns data, everyone owns different opinions, but all of this information is not consistent, so create a lot of noise. So the current pathology diagnosis is not cannot get a very conclusive information if meet a very difficult and rare case. Yeah, thank you.
Just a quick question on the cost and the cost efficiency of kind of what you guys are able to provide. Can you compare what the cost would be with your approach to, let's say, a customer of yours versus kind of the existing solutions?
Okay, I divided by two parts. If I just talk about the digital pathology, digital transformation, the conventional digital pathology company, they need to purchase the scanner, and the hospital need to purchase the scanner and all of the cloud system and the personal computer, all of the platform. So each case should be need 300 at least 300,000 per establishment. Yeah, in our case, we provide a personalized setup. So each case just only need 20,000 for the primary establishment, because we can share the scanner and share all of the technicians with our customers. Yeah, so the cost, the established cost for the digital pathology just only 1/10 of the other competitors. The second part is that we're talking about how much to do in the 3D pathologies. Yeah, for the 2D pathology, each slice maybe need cost around 300 US dollars. Yeah, for each slice. We provide 100 times slice with only 10 times cost. So that means that you can use 300 US dollar to get a single layer information. You also can choose use the 3,000 US dollar to get 100 times information and make your disease and make your patient become to reach a better treatment outcome. Yeah, thank you.
Yeah, thank you, YY. So we are time is up. So I think again, I want to give that since we might have some technical issue collecting sounds in the conference room here. So I will actually send the reminder for our virtual pitch team via the Zoom chat. So you should be able to see the pop-up at the bottom of your app, showing up the time remaining, like I'm testing now. So just a reminder for the following 3 pitch teams. Please watch out at the pop-up at the bottom of your app for the time reminder. Okay, so next team is a dot space. So let's welcome the third team to the stage. Thank you. Hello? No sound. Can you unmute yourself? Hi, Renson, can you hear us? We cannot hear you.
Do it work right now?
Yeah, we can hear now.
Yeah, great. Okay, I wait maybe 2 minutes. Okay. I'll share my screen. Do it again. Okay. We are dot space. Dot space for the critical patient, dot space offer continuous and reliable monitor systems to save their life. With the advance of technology, we use in past we use a wireless phone to discuss and talk with our friends. But right now we use a wireless phone to talk with family, right? In the health sector, the same thing is happened. In past, we used heavy, invasive, catheter-based equipment to evaluation our patient. But right now, we use a small, teeny wireless device to do the examination. But in critical care units, everything's the same. We still use the wired and the catheter-based monitor to monitor our patient. The most critical patient and fragile one. That space want to change it. We integrate now new technologies and we use a wireless technology to remove this wire and catheter from the patients. We offer a wireless monitoring system. There is another parameter we have monitored. The first one, we choose the intra-abdominal pressure. When a patient got infection, sepsis, or heart failure, burn, or trauma, it will increase the pressure of a patient's abdomen. Once the pressure above 12 millimeter mercury, it causes intra-abdominal hypertension. About 60% of patients suffer this disease, but they don't understand, and we overlook this disease. It causes a lot of sequence, and once the intra-abdominal hypertension occurs, the mortality rate will high around 50%. The treatment is very easy. Once we diagnose this and take a small operation to open abdomen, the mortality rate can down from 50% down to 5%. But the treatment is easy. Diagnosis is not so easy right now because we did not have a good tool to do so. Currently, we instill spotters into patient's bladder and measure the hydrotic pressure of the urine to replace the intra-abdominal pressure. By sense, it is a very low accuracy. It's very laborious. It's called it breaks down the sterile environment of the bladder. So most of the doctor or nurse do not like this procedure, and we do not do this measurement frequently. So in current ICU, only 6% of patients check this parameter. But everything changed since today. Our product, Pressure Dot, is automated work digital captures. Once it's sent to patient body, it can measure intra-abdominal pressure continuously for 144 hours without any labors. It's automated work and is very, very tiny to use. Our technology is very simple. We incorporate the spatial design antenna and the PC board into a biocompatible capsule and send to patient body. We have a receiver to catch the location and the signal from our capsules. We have numerous patent portfolio to protect our products. This is our capsules. Our capsules is very small and very easy to swallow. Compared to our competitors, we are the only one major laboratories passed the GI check, so we did not have the risk of infection. And we can offer continuous monitoring, and we don't need any people to do this, and we save a lot of time. Compared with other capsules on the market, our size only 1/6 of them. We do the animal study to prove our concept, and our measurement is more correct than our traditional one. Expected clinical value of continuous IIP monitoring can reduce annual expenditure of $1.6 billion because we save more times and we shorten more hospital stays. Currently, only 6% of patients measure this intra-abdominal pressure, so our market is 52 million per year. However, if the 60% of the patient in a critical unit measure IAP, our market will evaluate 10 times. If all the patients do that, our total market addressable market can raise around 1.2 billion. Our business model is very easy. We have existing CPT code. Once our product on the market, the health providers can get a pay and reimbursement. And we collecting continuous data from 144,000 hours. So collecting data, numerous data can help us to develop the algorithm to do so. And we collecting this data can help the ICAIH enterprise to develop new generation of monitoring systems. We can have a pharmacy, we can have insurance companies do right decisions. Currently, we are do our first massive production, and we will do first in human trial in this quarter. We apply the breakthrough pathway right now, and we will submit our FDA submission next year and on market at the end of next year. This is our team. I'm Renson. I'm a trauma surgeon and a fellow of American College of Surgeons. Dong is a urologist come from Stanford, and Wayman is a Google Health engineer, and he do medical device of several years. Our advisors come from Stanford, Cook County Hospital, and Taiwan famous hospitals. We have scientific advisor too. Okay, so I think we are that space, that to save lives. Thank you.
Thank you. Let's give the microphone to the judges.
Yeah.
Thank you very much. Great presentation. I have two general questions. One is related to the capture, right? So how do you get the capture out of the human body? Is there any risk involved? And what's the communication method between the device and your system? Is this RF, or do you see any risk with that as well? Thank you.
Yes. Once a patient swallow the capsules, you will pass by GI check and pass naturally. And maybe days after that. So we do not to retry that if it passes it out very smoothly. And by the communication, we use the Bluetooth communication, which is very widely used in the medical device right now. So we control and we handle this kind of technology very smooth.
Quick question on the cost of your device. What's the let's say per usage or per unit cost, and then what's the cost to kind of for the overall kind of solution? I'm assuming this is like a hospital-based solution, not a take-home type solution.
Yeah, so that's a good question. When a new device enters the market, the cost is very key point to check out is it work or not. And And for this kind of patient, we have to one of our devices around $205 to $300 per piece. For the patient who needed to use it, he stayed in the intensive care unit, it will cost maybe $18,000 per one. So using our using our device, we can shorten 2.3 days stay in the intensive care unit. So we can each patient we can save $1,560 dollars. So we can cover by shortening the hospital stay, we can cover the expenditure of our device.
Okay, it's a good idea, but you have to convince the physician to use it. Like myself, I use intracranial monitoring quite often. Sometimes with a small blood clot or something that it's cause bias a lot. It's not reliable. So I have question about the pressure dot capsule. Can you let me know who made it, which company made it, and have you tested before its reliability, and how you can prove that the pressure, especially continually monitoring, is reliable and accurate? Thank you.
Yeah, thank you for your question. And yes, it's a great challenge to prove in the clinical phase that I have to say the pressure dot is made by DustSpace. Yeah, it's our company. Our company is based in Silicon Valley and Taiwan. Our engineers is in Taiwan, and our headquarters is in California. Yeah, we do animal study to prove our product. Our feasibility and accuracy is better than traditional IAP monitoring already by animal studies, and we are due first in human this quarter, maybe next month in November, in Chang'e Memorial Hospital. Yeah, so sometimes the small sparkle will make some bias to make physicians don't like the new device, but by current result, the accuracy is better than traditional one. So our KOL from Stanford, from U.C. San Diego, and from Cook County hospitals, they like our device, so we still push it on our pathway.
Okay.
Yeah, so, okay.
DustSpace, time's up. So again, for our next team, please keep attention to the bottom of your app. I will send a time reminder through Zoom chat. And yeah, again, thank you, DustSpace, and thank you. And let's welcome the next team to the stage. The next team is from NYCU Taiwan. Let's welcome for the pitch. Thank you.
Okay, before beginning, does everyone see my PowerPoint and hear my voice clearly? Hi there. Okay, and I will start, okay? So, hi there, good afternoon, and I'm Marco Lin, the CEO of startup team of Dr. Wenjun Ko at National Yangming Chiao-Tung University, Taiwan. Today, I'm gonna introduce a novel underdeveloped product, intelligent image guiding needle puncture in laparoscopy, pneumoperitoneal establish, which aims to use in minimal invasive laparoscopy surgery. And first of all, unmet needs. In 2020, over 50 million laparoscopy are performed globally. During laparoscopy surgery, the first step of procedure is pneumoperitoneal, and at least 50% of major complications arise from this step. Hence, there are problems existing in current laparoscopy surgery. To surgeons, phase 2 personal tissue differences leading to huge stresses to patient, phase 2 4% breathing complication after surgery, to hospital, phase 2 medical malpractice lawsuit risk. Therefore, we believe a stable surgery brings a stable treatment, and we design a solution to solve the problems. Nowadays, the pneumoperitoneal procedures mainly depends on surgeons' experiences and or a simple pressure feedback tool, but none of them could provide objective evaluations. Hence, we have thought that if we could design an optical system combined with a various needle, it will provide real-time imaging just like parking sensors to get omni view in minimally invasive surgery. For this solution, we just verify this proof of concept first in human via academia IRB trial this year. And for the solution demo, let's see cartoon clip display how it works. During pneumoperitoneal processes, the optical lens combined with various needles will show real-time images, which obtain different features as penetrating into different tissue layers. Hence, it could provide standards for object evaluations. Competitor analysis, as we mentioned, the current pneumoperitoneal methods. Hansen's methods will lead to a bigger wound size. The other traditional various needle methods could have little pressure feedback add, but still depend on surgeons' experiences. For our product, we could combine the advantage from these two products and add on real-time image guiding characteristics for object evaluation. Regarding market size, we have done survey from 150 surgeons who have performed clinical laparoscopy surgery. From results, we found 74.7% surgeons think current various needle method is risky. If the traditional various needle puncture could be visualized, 82.3% surgeons will embrace this method. For global TAM market, it hits 200 to 500 million US dollars per year. For pilot market Taiwan, the SOM is about 200 to 216 million New Taiwan dollar per year. About go-to-market strategy, we divide our product system into three main components, and we've contacted potential supplier companies and downstream distributors for cooperation. In the end, we aim to form a B2B alliance with these suppliers and distributors to penetrate the market. For team members, I'm CEO Marco Lin, and I have over 6 years experience on business development, product development, and regulatory affairs in healthcare field. Our CTO Frank has over 10-year R&D experience on imaging project. Dr. Wenjuan Kuo is our technical consultant who spent over 15 years in biophonics research. And Dr. Yishou Huang is our medical consultant who assists us to clarify the pain points and validate the product application scenario. Media coverage part, this technology is recognized its innovation by Future Tech Award, and for its marketing potential, it has been supported by Taiwan government's program funding and accelerator to proceed product development. We also participate in 2021 and 2022 BioAsia Taiwan exhibition to prepare for establishment of a startup. Finally, the milestones. In the past 3 years, we've received a Tech Innovation Award and finished first generation product prototype for simple animal test and first in human academia IRB trial. In 2023, Q1, we'll have a FDAQ staff consulting meeting. Q2, we plan to open our startup. Q4, we'll try to complete the TFDA clinical trial protocol. In addition, to prepare the clinical trial execution, we are looking for $1 million US funding for that. And then we predict to execute and clinical trial and prepare registration document in 2024. Our team aims to be a better innovation creator in medical device field. If angel investor and industry company are interested in, you are always more than welcome to have a chat with us. Thank you. That will be all my presentation. Thanks.
Yeah, thank you. So I'm going to give the microphone to the judges.
Hi, it's also a very good presentation.
Thanks.
What is your instrument for image guide? Sonogram or?
Oh no, it's like the OCT image. Yeah.
OCT image.
Yeah, like using the visual, the standard way for the visual detection part. Yeah.
I see.
We use the same image system, optical system, to help us like to obtain the optical part, and it could create for a precise imaging during the puncture procedures.
Okay. Have you complete a clinical trial in Taiwan, like dividing someone who use your method and some another case control not using the?
Okay. For now, we execute our IRB academia IRB trial at TVGH Taipei Longzhou. So it led because our medical consult, who is the chief at the Division of Male Reproductive Medicine at TVGH Taiwan. So that will be the first site we designed for a clinical trial. But however, if you ask me why our team participate in this pitch, we are looking for the U.S.A. Clinical trial site resources if possible, and then we also prepare for resources. That's why we need to seek for the C-round funding.
Okay.
Is there a cost difference between kind of your approach versus current approaches? And if there is a difference, kind of, how much difference kind of is there?
Yeah, that's a good question. Yeah, for so far, the current competitor way, the cost for the cheapest is the Hansen's open surgery. It's like the surgeon just using the knife or a dissecting knife. It just use the regular open dissecting, so the cost is very low. However, for a various needle part, you would like to add US dollar like $50. But if you use our methods, I think the final product will be $300. But if you use the image guiding, the objective evaluation and the safety issue, I think would be largely increased and to decrease the risky factor to patients and to surgeon, even to the hospital. Yeah.
Thank you.
Just really quick question. You mentioned about conducting potentially next phase conducting clinical trial in Taiwan, right? Do you have any additional plan in terms of doing other experiments outside of Taiwan?
Yeah, sure. As you mentioned, besides Taiwan, I think for a medical device company, the main market is in USA, especially for startup. So I think for the second part, we are looking for is to execute clinical trial in USA if necessary, especially for the like the California part, because I think that's Taiwan government and our resources focuses on, and academia energy like Stanford or UC Berkeley or UCSF, even like in South California, I think they are also a medical resource we want to engage into. So yeah, we have the plan to execute clinical trial in USA to obtain FDA certificate.
One more question.
Yeah.
Still, I would like to ask why you choose the OCT guide instead of sono guide. You think that OCT guide is more reliable?
Yeah, the OCT guide is very reliable for sure, but however, it has its problems about for the cost part. And the other way for a storage part, it could like it need for the interface difference to have the images. So for the like the laparoscopy puncture system process, it could not provide this kind of the features for a precise positioning. And that's why we choose OCT imaging system. And then the main one problem is the cost is too expensive. But however, our R&D team has the ability to reduce the cost. So that's why we choose OCT as our final choice.
Any final questions from the judges? Okay, if not, okay. Thank you, Marco, for the presentation. Thank you.
Thank you. Thank you all.
Okay, so our next team is Taiwan Jinggu. 整蛊应该是吧 Okay, yeah. Okay, yeah, very interesting. Okay, so I think the team is here and we are 1 minute earlier, but actually we can start because after Taiwan Jinggu, the final virtual team, we will have 2 more team going to present in person. So thank you. We can start. Thanks.
Hi, thank you. Hi, everyone in South California. Hi, I'm Max from Taiwan Jianggu, and we are very honored to have the chance to introduce our product, Jianggu's the cable, which is a safety belt for spine surgeries. I am the CEO of Taiwan Jianggu, and also I'm the director of spine division in orthopedic department in Taipei Medical University Hospital. I'm also the CEO of TMU Body Design Center. Therefore, after I received my training in Stanford Biodesign, I initiated this project as a biodesign project because this is a pain point of spine surgeon and also patient, which is called a adjacent segment disease. This disease is caused by spinal fusion surgery. After spinal fusion surgery, the patient will have a fixed spine. Although it solves the pain temporarily, however, it will cause the adjacent segment have a speed, the deterioration will increase the speed. And there actually every year there's over 500,000 patients receive this type of surgery, and 30% of them will have this problem, and 10% of them will lead to reoperation afterwards. Therefore, it's a significant problem. However, current solution like a flexible rod or interspinous process device, they try to stop this kind of problem, but unfortunately, they cannot effectively prevent this problem. In these 3 years, there's a new method which is used a very simple way called stress stabilization. It's a span to reduce the stress induced by the fusion, and in the literature, it found to be successfully reduce the adjacent segment disease. However, this method can be only operate with an open method, so many surgeons actually don't like this kind of method. However, because of biodesign, we use this method and modified it into a minimally invasive solution. With a very simple tool, we modified that can be used in an open and MIS approach, and it's very easy for surgeon to use and very high compatibility for current devices, and it has been supported by clinical evidences. Therefore, many surgeons around Taiwan, including many big hospitals, they all support this kind of idea, and they all agree they will use this device in the future. And also they will help with the clinical studies and future publication as well. Not only them, they also have over 50 spine and neurosurgeons also like this idea very much, and they hope to use it in the future. Among them, there's also many famous spine surgeons, like the director of spine division in Stanford Hospital, also the director of Scoliosis Research Society, Professor Serena Hu, and she's now our team advisor. And also David Pauly and Lawrence Lanke, they are both very famous spine master in the world. They all said they don't like previous spine surgeries, but they like this kind of a nice approach, and they are willing to use this. And this gives us very good inspiration on this. And so our business model will be a disposable tool with implants. Every patient will use 2 sets, and the sale model is a B2H. The gross profit will be around 60 to 70%. And in Taiwan, we will have a direct sale from Taipei Medical University Hospital and Zhoushan Medical Hospital. And in US, we will have an indirect sale start from Stanford, from my professor. And we have a strategic partnership with a U.S.A. consulting company called Max Connect Medical. They will help us to find a distributor, group purchasing organization, and insurance companies. And also, we will invite all the KOLs to help us to promote the product, and increase the publication, and give talks in the international spine conferences. Compared to other devices, we have clear advantage that we are the only device that can use the minimally invasive approach, and the usability is very high, and also it has clinical supported evidence to reduce the adjacent segment disease, and also it can have the market that previous product cannot have, that in the minimally invasive spine surgery, and the price is also quite good compared to others. This year, we already finished our prototype and the demo and also cadaver usability testing. Now we are preparing to cooperate our company and have our first round raise at 1 million. And also we will finish our preclinical tests and submit for the FDA and TFDA next year. And we hope to clear our product in 2025 and begin the launch and also begin the post-marketing research. And our team, I'm a spine expert. I'm well-known spine surgeon in the world, and also many in the editor of many SDI journal. And also our team have experienced engineer who can develop this product. And also we can have many consultants, including accelerator and E3, and also many regulatory persons. Our team built up in 2020 and won the first place in the national competition that year, and also joined TMU Spark, DCB Accelerator, B Accelerator, and also Y Combinator Startup School, and won the first top 3 in this year's PT entrepreneurship competition. A quote from our advisor, Professor Peiyuan Li, he said, our team have a good solution to adjacent segment disease, and also we have good connection to world-class KOLs, and the whole process meet the need of clinical need, and also we can be a good example for entrepreneurship. So we hope our device, Taiwan Jianghu Cable, will help the patient to prevent second surgery. It will be a safety belt for spine surgery. Thank you so much. Thank you.
Thank you for the presentation, and it's time for the judges. Dr. Chu.
Hi, good presentation.
Thank you.
My question is, your 30% ASD, how long will it take to estimate ASD? Because of ASD is usually due to instability or losing of the instrumentation. It usually took 1 to 3 years at least. So what is your rate coming from? And the second question is, I still don't know how, probably it's a kind of confidential, but I still don't know how you do it using a minimal invasive surgery for KOL. So this is my 2 questions.
Thank you. For the first part, we have done a meta-analysis on the rate of the adjacent segment disease. There's usually very often to have a radiographic adjacent segment disease in the first 6 months, and the rate is quite high, around 15% to 40%. And for clinical adhesion segment disease can also be found in the first year around 10 to 20%. So we think although this device will need the clinical trial to get clearance, but we believe we need in the future we'll need to do a post-marketing research to further to prove this device to have better effect. Also, we will use the computer analysis like a finite element study to prove to give surgeons the data about how effective this is. And the second part, yes, thank you, Professor. Yeah, it currently is confidential, but actually it's a very simple way, very similar to repair of the meniscus. So we choose to use the MI tool, which has been already used in other surgeries, but we modify it into the repair of the tendon. So this is very already have been shown effective in animal and cadaver already. Thank you.
Hi, I just have one general question, just regarding your product life cycle, right? So, well, do you consider your product fully complete at this moment, or are you still innovating? And also, I do see that you have a prototype finished this year, right? 2022, right? So, I guess I'll let you answer first.
Thank you. Yes, we already finished our prototype and also the minimally invasive tool, and now we are already prepared for the clinical testing, like biomechanical testing, and also usability test for our clinical for our regulatory clearance. So now we are already prepared to cooperate and also prepare to some pre-stop to get to get some suggestion from FDA about our testing. Yeah, that's our current status. But we are very confident because we already test in a cadaver, and we've shown to be quite useful, and many surgeons like this idea very much.
Thank you.
Thank you.
Quick question on your FDA kind of process. Do you expect to file it as a novel or as there's a predicate device that you're kind of going against? Just curious, like the time frame that you're expecting here. It doesn't feel like you're doing on a novel 510K, or else it might be a little longer.
Yes. Thank you. Regarding to the clearance part, yes, we have found a predicate device that has been shown in previous slides, and our predicate will be implanted JCPF, and we already asked also Taiwan CD about how our device can this be a similar device to the current device. They said it seems to have similar function. So at present, we have very high confidence that we can have the chance to use this as a predicate device to pass through the class 2 device.
Thank you. And just real quick question is,
yes,
what's the data set that you have? It seems like you only have it on cadavers kind of currently.
Yes.
I'm just curious to see kind of how large the data set, even on a cadavers basis, that you've tried this kind of with, and how many do you intend to be able to kind of do in pre-tests to demonstrate, or at least prove good enough in your mind, and then subsequently good enough in the FDA's mind to kind of get this thing through?
Yes, thank you. Regarding to the clearance, actually for like JCPF, they just have a biomechanical test, and then for 1 million cycle testing to prove the longevity of the implants. So actually they didn't do any animal or cadaver study on their device. So for us, Yeah, we did usability test for animal and cadaver. And for animal, we had done actually 5. And for cadaver, we had done 3. But actually, we haven't done implanting like a cadaver biomechanical test yet. But we already done in vitro biomechanical test, which has shown this device is better than current device, around 20% to 30%. Yeah, so this is our current status. And also we believe the data set we need, yes, we are preparing for the pre-sub. So after the pre-sub, we will get the whole idea about how much we need, what's the data we need for the clearance. Thank you.
Thank you.
Any last questions from the judges? Okay, if not, yeah, thank you for the presentation.
Thank you so much. Thank you. Thank you again. Bye-bye.
Thank you. Bye-bye.
Bye.
Okay, so we are going to switch the gear because our final 2 teams will be in person, pitch. So I think we're right on time and we are good to go, right? Okay, so the next team is Elastro Medical. Thank you.
Thank you, Nick. I'll let them bring it up and then we'll take it from here. Double check. This does not advance. It does advance. Okay, great. Good afternoon, everybody. My name is Dr. Albert Huang. I'm the founder and CEO of Allotrope Medical, where, as you can see here, we're enhancing surgical precision using advanced electrical stimulation. We've had the great fortune to be backed by such luminary groups such as Y Combinator, as well as Tech Coast Angels, and even our law firm Wilson Sonsini has backed us, and we've got a number of awards and accolades from other surgical societies as well. I myself am a general surgeon by training. I've done my training at Harvard, Cornell, Penn, University of Rochester, amongst many others, and I came up with this technology in the operating room. And I've been fortunate enough to build my team out with many senior people, including those that have decades of experience in med tech, medical devices, and amongst everybody up here, there's over 11 mergers and acquisitions completed. So great team behind me bringing this to market. That's me there, what seems like a lifetime ago, performing minimally invasive surgery. This is where you're working with long thin instruments or sitting at a robotic console, looking at a video screen. And although that's become the standard of care, it's made identifying critical structures, and namely not injuring them, much more challenging and time consuming. And currently very few injury risk mitigation technologies or surgical efficiency technologies exist on the market. And if they do, they're quite expensive. Our technology focuses on preventing injury and identifying and protecting a structure called the ureters. These are the long, thin tubes that connect the kidney down to the bladder. They're not always visible, but yet verification is a key step in over 2, maybe even 3 million operations done around the United States alone, including colon resections and endometriosis cases, hysterectomies, other female reproductive organ operations as well. And up to 1/3 of the total operating time can be spent just looking for that structure. And finally, injuries to this are the leading cause of litigation for anybody operating in the pelvis. So how are we dealing with this? Our technology uses electrical stimulation that mimics the body's own signals to control the ureter to identify these critical structures like the ureter during surgery, and it's been shown to increase surgical efficiency, shorten OR time, as well as decrease hospital expenditure. And this is our device. We call it StimSight. It is a technology that lets you plug your existing surgical instruments into to deliver our electrical stimulation signal, giving surgeons a brand new feature of their instruments. And these are tools that haven't changed in over 30 years. The next video is a surgery video, so I'll warn you, it's not very bloody, but it shows how the technology works. Surgeon uses their existing instruments, sees a structure they think might be the ureter, and they can touch it, deliver our stimulation, see what it is and where it goes instantaneously under their control. This lets them move on with their operation in a matter of seconds while providing all the other benefits as well. So what does it do? It increases surgical efficiency, shortening OR time. Our data and studies have shown that an average of 5 to 15 minutes saved per case by using StimSight. OR time's expensive, $60 to $100 a minute. So already just using it saves time. On top of that, for the surgeon, they have greater visibility of the ureter throughout the whole case, meaning they have a smooth, safe case flow, benefits them as well as the patient. And a part of the operation that's done commonly by gynecologists is called a cystoscopy. They put a camera into the bladder to look to see if they've actually injured the ureter. And this takes about 17 minutes, but using StimSci during the case has taken that down to less than 1 minute. And with some surgeons even saying, I know where the ureters are at with StimSci, I don't need to do cystoscopy at all. Additional time saving, procedure saving, increased OR efficiency, all available to you now. And on top of that, because StimSci is an FDA cleared product on the market, specifically and uniquely used to identify the ureter, you can actually document your opera report that you use StimSci to identify the ureters for medical legal support and guidance. Here's a more complicated video of how it's used tied to the Da Vinci robot. So the instrument here on your left, and my microphone went out, so I'll talk louder, is used to stimulate what they think is the ureter. You can see it move there, and instantaneously the surgeon knows where to go next in the operation. At any time during the case, they can use their instrument, stimulate what they think is the ureter, know where they are in relation to it, and be able to move forward with their case. And surgeons have said, this is like I've always had this tool in my hand. We connect with everything that the surgeon already uses. The Da Vinci robot via adapters, LigaSure via adapters, any instrument the surgeon in the hospital already has, we deliver our signal through that for a totally seamless workflow experience. Shifting to the business model a little bit here and the market size, over 2.5 million operations done each year require you to identify the ureter at a key step at some point in the operation. And for us, represents over $1 billion domestic market alone. If we look at our business model, this is where we get very excited about the technology. So we don't sell that box. We place that box at no cost to the hospital, letting us get through in a very short sales cycle. And the hospital purchases this thing. It's an activation card. Think of it as like a prepaid cell phone card that plugs into the front of our box. And what it does is it allows us to generate high revenue up front and all of our profits as we sell the card. And then at the same time, this is loaded with this number of uses. The docs just use their technology and hit the foot pedal, and then we drive down the cases. So it's very user-friendly at the same time. Going deeper into the financials, we sell the cards and are selling the cards for $4,000 a piece. They cost us about $40 to make, meaning we have almost 100% profit margin. The box itself costs us $900 to make, so the math is very easy to show that from day one, we are profitable. And then after that, all you use is the card, and once that's used up, you order another one. So it's almost 100% profit margin business model that everybody is happy with. We've been on the market since April 2021. We have 18 accounts with more onboarding across the United States, as you can see here. We're in multiple health systems. Places like Ochsner are rolling us out for the full place. And we've got a wait list of over 600 engaged clinicians wanting to use this technology. So in summary, we're an FDA cleared disruptive surgical technology without direct competition. Simple to install, train, used by surgeons while providing a never before had clinical capability. And we're rapidly accelerating commercialization with near 100% profit business model that is hospital friendly. And that is why we at Allotrope and with StimSci are poised to become the standard of care for all pelvic surgery. So thank you for your time, Shishia, and I'm happy to answer any questions.
Thank you.
Okay, it's a very interesting presentation.
Thank you.
And it's smart. Really, most gynecology, our GU doctors spend much time in identifying the ureter. I think especially in a shorter time. The only question I have is, can we use nerve stimulator with low voltage to stimulate the ureter? I don't know what mechanism.
Yeah, great question. So what I'll do is I'll just use our patents to kind of answer that question. So nerve stimulators do not work to stimulate the ureter. I spent about 2 years figuring this part out because the electrical signal we make is proprietary to us. We've gotten the patents on it issued right now because it's a specific waveform that all these things are kind of a little bit of luck and timing, right? So before I came up with this idea, nobody had figured out what that electrical signal is that can make the ureter move every single time. I tried all kinds of nerve stimulators, different waveforms, power profiles. and finally figured out after 2 years and a lot of large animal models, exactly what that signal is, so that when the surgeon hits that foot pedal, they don't need to think about anything. It will make it move 100% of the time because the ureter thinks it's the body telling it to move. And so that's why they were able to give us patents on it.
That means you need your signal box.
Correct. You need the
Okay.
signal box, and we place that at no cost, and we sell only that activation card, right? And it's very small, it's this big, but then the hospital only buys this, right? And then this gives them usage of the box. And since it's already connected to the tools, in the operating room, the surgeon never has to go, oh, I need you to bring that instrument to me. It's always sitting there in the operating room. And so it's always sitting there, it's always connected to their, sorry, usually I can move, oh, there we are. You can see it's connected always to their existing tools. So they don't need to do anything. They don't need to ask for it. They literally just have the nurse push a button on our box and the tool in their hand that's already on the patient delivers our stimulation. So it's like totally seamless for them to use the technology, which is very important, right? Because when you need it right then. And so that's why I designed it this way. And then this is the business. Right? Okay. Yeah.
For estimation of the market size, what's the true rate of ureter injury?
So ureter injury is very low, right? Around 1% of actual injury, but we're not focused on injury, right? We're focused on the time spent looking for it and the time spent around cystoscopy protecting the ureters. This is where all that effort is, and that eats up a lot of time. No study for any technology has shown any ureter injury rate changes in decades, right? The study would have to be massively powered. So we don't focus on that. The safety is the undercurrent, but the surgical efficiency, the medical legal protection that using StimSci gives you, and the ability to save time during the case and have what we say the cases end on time, because now you have predictability, right? By using our technology, that is where we put the value. And then over time, all we can say right now is that it's been used in over 800 cases that we've never had a single ureter injury to. So that's not a true study, but nobody in the past has even been able to do a study with ureteral stents or anything like that yet. So we stay away from that very safely. Thank you.
Just a quick clarification question to what the doctor has asked. The signal is getting sent down through the whatever device because it's a metal device at the metal tip, right? So you're sending the signal. I'm assuming it's a very low voltage.
Very low voltage, quarter of a second impulse.
Got it. And it's meant to basically just stimulate one time.
Exactly.
It's a single pulse.
It's a single pulse. Yeah. No frequency, cannot cause any thermal injury. This was all part of the FDA clearance process as well to show no ability to injure anything. And then the other question people often ask tied to that is, can you inadvertently use your same tool to burn something when you meant to stimulate? So we have a separate foot pedal for our signal box. So you have to hit a separate foot pedal. And on the front, there's actually a push button. That's the selector switch that changes your tool. So I call it two-factor authentication. The surgeon has the foot pedal to push at any time, but somebody needs to push the button to select stimulation or the cautery. And both need to be in line for that right signal to be delivered.
And so the adapter, I don't know if the adapter's on there. The adapter has basically, connected so that you have a signal that you can go into one of the.
Yeah, it goes down. This goes to the field. This was taken just on Wednesday. I wanted to use it because it's fresh. And then you can see it says bypass right here, because right now they can use their cautery system like normal. The moment they want to use this device for stimulation, they would have the circulator push the stim button. This effectively will cut this box off from delivering signal through that tool. It will only deliver our stimulation signal with the separate foot pedal.
So it's actually doing a full cut box.
Exactly. It's super important for it to be a relay that disconnects completely. That was part of FDA's clearance process that we did, and we had to do human factor studies around that because we can't just rely on just a separate foot pedal, right? That's too risky. But at the same time, this is great because it's no different than having the circulator change a power setting on here, which they do all the time. So workflow, it's not interrupting at all because the surgeon doesn't change the tool in their hands or what they're looking at.
By the way, thank you for the presentation and also the demo too, right?
Sorry, I try to people in the surgery videos, but it's a healthcare conference. We should be okay.
Yeah, I have more business model related question, right? But by the way, I really like the activation card type of model that you can recognize revenue upfront. And it seems like everything's progressing well. And I would imagine that you're seeking capital to fuel the next level of growth at the moment. So I'm interested to learn about the market adoption rate right now. And also, what's your vision going forward on this point forward? Yes.
Sure. Yeah. So kind of a snapshot of where we are right now. We're doing a limited market release, right? So focused in key cities. We're still a small team. So what we've done is we've positioned and hired a salesperson in each one of these 3 cities. So they live there, right? Because they need to go to the hospital. We can get the product in the OR. The docs use the technology. They love the technology, but then you need to drive habit, right? So this is our focus right now is we put somebody here, here, and here. Ochsner is a great example of putting one person there. We finished our trial because they were able to camp out in the ORs every single day, get the docs to build it into their habit, and then now that unlocked the entire Ochsner health system with the 16 more hospitals that are now rolling it out. And then we're doing that with Robert Wood Johnson and Centara, which is where I was earlier this week with the same idea, right? We're not trying to go nationwide. We're really trying to be very purposeful and focused. Our spots like over here and over here are other ones like critical access hospitals that don't have resources. So they love our technology because it is the only help they have. But if we're thinking about growth, then we're looking at hospital systems and things like that. And that's why we're targeting these key cities, including Houston, where I'm from, because huge hospital systems, we start with the trial, we purposefully drive the trial, drive success, and that unlocks the entire system for us. And having somebody living in that city means that they can drive to the ORs every single day and look at the board and go, the box is already in the room. I just need that doc to use it for his 3 cases today, and there's no reason that he wouldn't. So, and then we're going to try to grow it that way, right, as we move through our subsequent financing. Yes, so the signal box is approved by the FDA. This is an FDA cleared product via 510K pathway. It was approved in November of 2020. So we've been on the market. You know, once we got the clearance, we set up all of our training for sales, got ready for the market launch, which is why April was the launch. So about 5 months after clearance is when we got it. But this is our unique indication for use that the FDA gave us at the bottom. And kind of like between the lines, next week, the FDA is going to be announcing that we're going to be allowed into the Safer Technologies Designation Program. And that is going to be the next level for these hospitals to adopt this. Now we say, it's not just us saying it's a safer technology, FDA saying it. So that's where we're going to be at now.
Okay, thank you, Albert.
Thank you all very much.
Okay, so next is our last, the 7th pitch team, AI Fab. So now the speaker is going to get the microphones and also the remote control. So let's give the team maybe 10 seconds. They will be on the stage and ready to pitch. Thank you.
Hello, everyone. Well, thank you for sticking to the end of the day. And Dr. Chu, Mr. Ko, Mr. Yang, and Mr. Wu, thank you for your help. My name is Howard Young, and I'm with AI Fab. And we are a customized AI solution house. So we work with image data and NLPs and different AI models, and we customize it. And majority of our clients are in Taiwan, and they're in the medical field, AI medical centers. And that's one of the reasons why we're here today. And we're trying to promote the solution that we already developed for the Taiwan hospitals. And company was founded in 2019, and I'm head of BD. I'm also a former Marines in the US. So that's one reason why we're engaging with some of the VA hospitals. And we also have a chief medical officer just to support the effort to sustain our growth in the medical field. He's a doctor from the Columbia Hospital. And we also have strategic partner also as part of the Mount J organization. And so some of the challenges that we experience today with the medical AI is that we know we could do better with technology, and but the pain point is the cross-domain communication. A lot of times, doctors don't talk to doctors, hospitals don't talk to hospitals, and there's just not enough AI talent in the field. And so we are the bridge to make things happen. So We are have a unique advantage. Why use us? Because we already have a proven trust. 20% of the hospitals in Taiwan is working with us. And I know the slide, I changed this slightly, and since last time we submitted it, and so it's a little bit different than what's printed. And we have a very fast time to market. We really have a lot of pre-trained models. So when we deploy, we don't have to start all over. So we can reduce the time to work with hospitals or to work with medical devices companies to get faster to the solution to the market. And cost-wise, because again, it's already there are pre-trained models, and so we can help reduce the NRE to enable broader access to the market. And talk about the business, right? What are we doing in the U.S.? We're R&D in Taiwan and BD in the U.S. And we are working with horizontal expansion. We have high retention rate with the hospital that are working with us. And we generate leads through different medical courses to talk about what we are doing through different AI models, different workshops. And we also work with different trade organizations trying to promote the AI use in the medical field. And these are some of our applications that we have developed. And so for Dr. Huang, I think that the competition is coming. So we're using AR and VR to essentially put video images to overlay with different organs to provide surgical intelligence with the different hospitals. So we're working with surgical tool companies and working with hospitals to trying to provide intelligence to trying to avoid such issues. And we also work with different images, ultrasound, pneumonia, for example, X-ray, bone fractures. We're not providing a diagnosis. We're providing a tool to enhance, to speed up the diagnosis for the doctors. We can look at Parkinson's disease with how they draw the circles, different ejection fraction ratio for the heart, how big it pumps and how small it shrinks, and different CT scan. Again, we're just doing the labeling. The doctors have to make the diagnosis, and we're not there yet to make an FDA device. And we also have patent patents in Taiwan and IP, and then we work with different hospitals to achieve that. And in the US, and that is to come yet. And because we have pre-trained model, and the model needs to be continuously improved. And since last time we talked, we changed the price a little bit. We make it a little bit more expensive. So we have a kind of like basic package. We label the data, we train the models, and we deploy it. And that's a low cost, monthly payment. And we have more advanced, including more with integration with the existing systems. And therefore, a more expensive subscription level, licensing level. Essentially, we are working with FDA partners to try to work with the device manufacturer that are working with us to try to get FDA track. So essentially, we are their AI outsourced partner, and that's kind of how we position ourselves to be. And if they want to create a medical device, we are also part of the Qualcomm innovation in Taiwan. And so essentially, we're using Qualcomm SoC system on chip to make it a similar solution to what Dr. Huang is talking about, a box in a surgical room to provide instant surgical intelligence. And so again, the benefit of having such product, it's you don't need a cloud, you have faster deploy time, and you can see the solution in a real-time space. And these are some of the partners and clients that we're working with, Qualcomm, U.C. Davis Health, VA hospitals, and we also work with Stanford, and a lot of Taiwanese hospitals are using us. And in terms of the business model, like how big is this market? Fractional AI, licensing AI, however you want to call it, outsourced AI. We're looking at about $21.5 million a year if we can get into 20% of the labs. And then if we can get into majority of the hospitals in Taiwan and US, we're looking at about $788 million. And so we are in a pre-A status for the startup. We are looking for money for about $2 million to help us get into certain markets. And yeah, and that's kind of where we are. And we're using the safe agreement for the future equity. And if you Anybody today have any questions about how to AI enable your medical device, or what are we looking in terms of angel investment or seed investment? Please come talk to me after the show. Thank you.
Thank you. Right on time.
Start in the middle. Who actually owns the AI model when you work with some of these hospitals or divisions, right? You develop a model, you ingest whatever data they have to ingest, right? The model you develop, who owns that, right? Do you own it, or does the hospital own it?
Well, I mean, so the subscription model that I was talking about is really to help them get to that first, so they can look at a kind of proof of concept. But then if they really like it, they want to own it, we train them with their own data, and then they can own the AI model that trained with their data, and but that's with the premium fee.
So you can't take that AI model you developed
And
right?
fine-tuned it.
Well, my question is more, can you use it in other instances? Let's say, for example, everybody wants to do the same thing with an ingestible kind of data set, right? Sure. Can you use that model you developed, right, in partnership with that hospital and sell it to some other hospital that has pretty much the exact same need? And the data set, training set data is all about training set data, right? Is already, even potentially more robust, right? Now you can refine that model to be even better in theory, right? Can you do that, or is that like not part of the equation currently, and you're pretty much doing a one-shot NRE type approach with each of these hospitals?
So to answer your question, yes, that can be done, but that's not on our roadmap at the moment. So we are doing training to enhance our models, and when we are ready to launch kind of like a product like Dr. Huang, then we can take a software as a medical device to the FDA 510K process, and then we can have start to all the different hospitals say, well, this is FDA approved product, use it. So, but at the moment, we're really providing an outsourced AI services to device manufacturer hospital, research hospitals that wants to enhance and to make less mistakes at the moment. I hope that answers your question.
Sort of. I think it's more, can you use, because it's all about training set data, right? So you got one training set data, whether or not you can use the output of the training set data, right? Call it, you can obscure it, whatever the case may be, right? But you can't, can you utilize the knowledge you gain from that training set data in that model, right? And use it in a different model, in a different set of data. Does that make sense?
Yeah, we can do that right now.
Right, and then utilize that to refine, so you don't start at zero, right?
Yeah, exactly. So we do that right now. That's kind of what I was talking about earlier. We can speed up the training process with the because we already have the model developed, but then we just fine tune it with the customers their own data sets.
I want to kind of build on that question a little bit, and also a couple other questions as well, right? So. You talk about deploying this at edge, right? So what Howard mentioned was that whether you can leverage the same data set if there are multiple edge devices within the same client that you're trying to utilize, right? So how is that being accomplished? And also as a related question, right? What biometrics are you collecting as part of this experiment or many experiments? And my doubt is that whether how many pre-trained models you have, because on your side you're trying to adjust at least 6 or 7 different type of I would say applications in the hospitals. Right. And just, can you elaborate on that a little bit?
Yeah. So essentially to deploy it on the edge, we work with a device manufacturer who does have video input, voice input, and we enhance the products. But then that's kind of we working in the background to not so much deployed, is really more on the R&D phase. So for proof of concept, for feasibility tests, and so we are the outsourced AI team to help them achieve what they're trying to look for. And because to do so in the U.S., to build an AI team, to do that is really expensive. So that's one of the approach that we're doing for revenue purpose. And for the AI models, all the different fields, we work with outside partners that have data label experience in the medical field. So that way, we don't have all the data. I mean, in general, medical data are small data, not really big data. 10,000 images, 100,000 images, that's still small data. So, but we don't have access to all that. So, but we work with partners like you to help train the model.
I see. And in terms of utilizing, are you thinking about using federated learning across different devices, or is this more like you deploy once and just use it without any sort of reinforcement learning?
We do reinforcement learning, and then we also wanted to continue training the models to make it more, I guess, more accurate. And right now, our accuracy is about 92%, 95%, and we want to get higher than that. So we want to continue to improve our efficiency and also the accuracy. So that way, then we'll go to becoming a software-as-a-medical device. Yeah.
A final question. Going back to your final slide, you mentioned raising 100K at a 2 mil evaluation, right? Is 100K going to be enough? How many data scientists are you thinking to employ? And you also mentioned about utilizing an outsourced labeling company. What will be the cost related to that? Thank you.
Oh, we're actually looking for about $2 million, and minimum is like 100,000 because we don't want to make it. Yeah. So yeah. And then for the U.S., we're doing mostly business development and also. So we just, strategic placement. Yeah, thank you. Hi, hello.
Hi, it's a good model, but up to now, we only can learn the structure and the process. As you have so many partners, could you show us some outcome data, like which partner, hospital partner, had quality improvement, or which company has cost down or improved efficiency? Could you show us that?
I didn't provide data for today's purpose because due to the time constraint. We have a lot of videos, and then but in general, I mean, for example, like I use ejection pressure for the heart. We identify the edges of the when the heart expands and when it contracts, and we can quickly do a calculation within minutes. And then instead of having the doctors do a snapshot and then draw the line and then do the calculation, so that we improve efficiency by at least 5x, and higher accuracy. In fact, I think the paper that I was talking about is related to that. Find it? Yeah. So this is with the Chime Hospital in Taiwan, and they're talking about different, the same idea, just how we do it, how do we do it efficiently, and so the doctors, when they look at it, they could quickly know what they're looking for instead of having to look at the black and white images. I hope that answers part of your question. Thank you.
Thank you. I think we are right on time.
All right, thank you.
Any other final questions? If no, yeah, thank you,
thank
Howard. Okay, so I think this concludes our program today, and also thank you all the 7 companies that present today and pitch to the judges, and of course, thank you our honorary judges today for your precious time evaluating those companies. And last but not least, thank you our host, Southern California Mount Jay Technology Association. Thank you, and thanks.